maximum number of people that. And this is what I was. Lower right, second one, second down, the top line. It's open. Okay. Around. Possibly. And equal. Okay. Then this ground. And okay. Man down. The nine, 7, perfect. And machine please. My God. Not all.
again, first round. And this a diamond ball. Please notice I am can see the fingers, the night Egyptian. Then I'm covered back. Anything? Today I'm thinking for
Otherwise, if you read over, begin.
external. See, see, see the gingival tissue, the gingival tissue, but this is the gingival tissue that yourself lies. Just be comfortable. Have you been comfortable, Sarah? Yes, yes, gingival tissue. And the water is see the meeting point. This is the party and this is round. Right? Until and this is how We practice how fast and this contradiction. This is a country. Right? It is being very how very how you are what I think what I and thank you now now well now I already I what I should say for the India credit not on the something and comfortable. So that one and on the and underway and done on down and down and God and that I and God there was the right but the on and time by day and has the time that now and how the put of it. Can you see there is no feelings and on and you see the ground the feeling and also there is peace. And God by that something continuous. I am trying to say and talking on the radial side, then see how the side looks. If this career is saliva, here, but and the research what now I thought that the one is my best and thought that I just want them world over that I need some content. One kind of content around outcome project. Number two, offer the thinking, just living, offer intention, number four, and compassion. I have tried. Abolish all the things I have my own and and I would, but for that time, it's fast work. And I think that is fast. Then I every fast. If you get it, then the time and the damage. The time. And it and then the influence market and does not have it does not have anything from the and I after full text of the text now in we think so that someone said there I thought it was I I had saliva and on the saliva and it has sometimes from it what that is maybe now for saliva after having done this what good one no money and the I system was on by technique of the what would be what and saying so and patients, there were most about 400 plus ladies and only 250 around males. So there is a disparity almost 1:2 ratio. But I throw it open to any, I would love the pathologist, especially because I want the pathologist to study what is the tissue condition with my technique and what is the tissue condition after using a scalpel as to what happens, which is a cleaner cut and which is a better tissue. Secondly, I dedicate this not only to dentistry, but as I told in my, I'm sorry, yesterday we couldn't show the video, but I have another presentation abstract in which I said it could be used in many lines of medicine as well. So I request all the doctors to try it out. We have a neuropathologist also over here. And maybe it could be tried on other branches of medicine where they may not need as the only thing why I request you all to make less use of anesthesia is because if the body can welcome such a technique, then we do not need to put a foreign medicine, chemical inside the body and avoid the reaction, even the simplest of the reaction. As I said yesterday in Pune, India, a 3-year-old girl died in the dental chair because she was given a local anesthetic injection for her root canal, and then she had to be rushed to a pediatrician and to a hospital, and she died. And the doctor was called as a killer dentist, and I helped him out because I couldn't tolerate doctors being unnecessarily blamed for things which are not in their control. So all I request the entire global world is, especially in poor countries, I'm ready to go to, as I said yesterday, 10 most poor countries in the world, free of charge, my service will live in, and teach them how to do these things so that they do not incur more expenses in using lasers and others. Thank you. I hope UN and WHO are listening. Thank you. Thank you.
Thank you very much.
Again, she gave me yesterday. It's already given yesterday.
So right now, I would like to introduce the first speaker of day 2, Mr. Mahmoud Ali from United Arab Emirates. I'm passing him the stage to deliver his speech on pharmaceuticals, managing turbulence and securing a competitive advantage. Thank you.
Thank you, Dr. Hani. It's our chairperson, and a very good morning, ladies and gentlemen. My talk today, it will be about pharmaceutical industry and how pharmaceutical industry as one of the cornerstones in the healthcare structure. And it was obvious, and that healthcare structure before COVID was in little comfort zone. They not take any risk management procedures. But during COVID, I think everyone witnessed that there is a lot of challenges facing supply chain, facing the pharma industry, facing about even the supplies for the hospitals. Let us have a journey about pharmaceutical industry. Yeah, okay. So, okay. Okay. About the Middle East, we will talk about the Middle East Africa pharmaceutical market insights. I was living in UAE since 16 years ago, and in GCC around 23 years old, 3 years ago. And actually, I'm a pharmacist. I was taking a lot of position in different pharmaceutical company. And finally, I'm a general manager for Saja Pharmaceutical, which is a joint venture for Saudi Arabian and Japanese company, Astellas and Daiichi. Thank you. We have a lot of product, around 46. Okay. So let us have a look for Middle East Africa pharmaceutical market insights. Market site snapshot about Mat quarter 3, 2022. This is very recent, which is published in December 2022. Many large markets have witnessed high growth. Overall, Middle East market reached to around $31 billion in value and around 8.7 in units. Value sales growth has been 11% growth over the last year and 7 CAGR, while the volume sales growth has a 5% BPG and 3% CAGR, which indicates that the product of high value is more growing rather than the number of units. Retail channels dominate the Middle East market with 70%, which means that not all the institutional sector and public cover all the population. Multinational dominates the market with 57% share in total value. Sales local companies have growth of 15% BPG. Systemic antibacterial is the top therapy area, holding 8% of value. KSA is contributing 27 and Egypt 25, which means that there is a lot of empirical treatment and without over OTC or over the counter, which is not advised actually. Lantus and Prevenar is the most selling group. If we go among the key markets, Egypt show a growth of around 17%. However, KSA contribute to the largest market, accounting for 31 from the sales of Middle East region today. But the growth pattern in Egypt is higher, and the unit-wise sales is higher than the value in Egypt. UAE growing increased by 11%, according to $3.2 billion. Regarding also South Africa, have a 6% BPG, accounting for $3.4 billion, which is almost same like UAE. Saudi Arabia have a 14% BPG and accounting for around $9.6 billion, so it's 3 times like UAE sales. Egypt have a 15% BPG in the Middle East region, driven by growth of hospital channel, which is 26% BPG, and the health insurance improve. Also, the same pattern is applied for Algeria as a bigger market, Francophonic West Africa, which is talking about French language, and Morocco and Kuwait market. By 2025, this is about projection and prognosis for the pharmaceutical market, expected to grow to reach $1.6 trillion. This is global. Middle East and Africa to collectively contribute $56 billion out of $1.6 trillion. As you see in Middle East, it will around 25 billion. CAGR, this combined compound average annual growth rate, which is from 21-25, this 4 years, 4.8% growth. Middle East Africa is forecasted to reach 47 and is collectively growing 2.5, 16 from 16 to 2022, which is around average of 5 years growth. As you see, North America is contributing the highest all over the world. This is the blue color, and after this, Europe. Second, Southeast and East Asia is the third one, and Japan is the 4th biggest market in the world. Middle East is around contribute as a CAGR. Projection is higher in the growth, but the last 4 years was 0.7 only. It's almost plateau. There is a lot of initiatives during the mini crises like COVID and after COVID. A lot of initiatives in pharma industry, starting from omni-channels, smarter IC plan setup. It's considered like incentive plan setup, which integrate for the face-to-face and the digital representation activity and motivate pharma sector for effective omni-channel promotion activity. Second initiatives is empowering self-serving capability. And third is motivating future interactions and connected artificial intelligence. This all is around digitalization. This is a benefit actually after post-COVID, we will get this from it. A pharmacy level analysis helps to address demand fulfillment, challenges to ensure availability of potential pharmacists. This a lot of countries suffering from shortage during COVID, But after COVID, many authorities and public sectors, they start to have like a protection or a strategic stock from pharmaceutical product. And also in UAE, they have a lot of great initiative about Tatmin. This following is a box in everywhere. So Ministry of Health, they can know the box where, which in pharmacy, and how the cycle from the cold chain, from the start from manufacturer to the distributor until reaching the patient, pharmacy and the patient. This will decrease the counterfeit, smuggled product, and also the abuse of any medicine, or taking about controlling of temperature, so like a data logger that you will keep the temperature from the cascade, from the cold chain, from manufacturer until the end user. Regarding digital adoption landscape, a focus on Middle East region. Both healthcare professional and pharmaceutical company in the Middle East region have started recognizing the value of digital engagement for better reach. It has been found that 54% of healthcare professionals are willing to consider hybrid engagement model, and I think we apply this in our conference, that we have a physical interaction and also as well as the online interaction. About regional summary and analysis, all channels grow in the value growth in BBG. As you see, this about retail, this is the blue retail sector, and institutional and LBO is considered like a public sector, which is a dark blue and white. So as you see, it's dominating, the retail sector is dominating. Okay, and even it is growing and the projection is higher. So it's a matter of privatization, and start to have the retail have more stronger role than the institution and the LBO market. The dominant channel is the region, accounting 70% value sales in the region. LBO channel continue to have a stable contribution in the region, with Kuwait and UAE and Saudi Arabia growing by 10, 15, and 16 respectively. Most Middle East countries have contributed to grow in value, except Kuwait and Algeria, which saw slight low in growth over last month. As you see that about PSA 14.7, Egypt 17, this RSA 6%, UAE 11%, which is showing a very strong attractiveness in UAE, especially as a higher growth battle more than the other countries in the region. If we will go for a therapy area or a therapeutic classes, we will go through a different therapeutic class. What is the most selling therapy area? Is alimentary tract treatment, systemic anti-infective, this including antiviral, antibacterial, antiviral, and antifungal and antibacterial, nervous system, cardio product, anti-neoplastic, and respiratory. This about leadership in the turbulent time is very, very important. Dealing with leadership challenges and how to manage the turbulence or the challenges we are facing in pharma or healthcare structure in general. Dealing with leadership challenges, recognizing inclusiveness and diversity, leadership as a positive sum game. This is about challenges for the leadership and the organization facing, about external as a turbulent environment, since a lot of new rules or a new system and a new trend. And now it's going to account management rather than it will be a general or a flat treatment. You have to deal with every chain of pharmacy or every hospital in a different way. And about internal turbulent organization, maybe embedded into organizational institution through factional conflict, staff turnover, conflicting role, internal reform, and complex operation. Second, about priority, make a priority about leadership challenges and how we will make a priority for which one is the highest influencing the healthcare structure, and what is second one there, and give allocating of resources and time devoted to manage these cases. Third one, a types of leadership challenges. It is will be from a team to critical and weight challenges. This about COVID, during COVID impact on GC and the pharmaceutical industry. We'll go about the impact. It's impact of most of all sectors. Some sectors affected negatively, and some sector limited impact, and some positive impact. We will may go for the positive impact about digital services, telecom and internet, digital media and the payment portals, food, e-retail and e-commerce, healthcare and education. This is a positive affected during COVID. Pandemic challenge on healthcare infrastructure. This about last 3 years, we have to review, rethink, and reshape operations and how to manage these issues. Scenario is expected to greatly impact the generic drug maker in the region, with increased dependence on India and China for API. Actually, China is the most contributing around 65 from raw material and API all over the world. But India now is growing a double digit, showing a huge growth in API in the last period. We'll offer value-based service as a patient assistance program, like a patient support program, like here in UAE, there is Axios or Manzil or IQVIA, which are supporting the patient journey from the start of diagnosis and algorithm until getting the medicine in the end, and how to be convert from volume to value, and how this the cost for the healthcare structure to minimize, or by other word, optimize. COVID-19 impact on healthcare to do social distancing measure, lockdown, and fear of catch infection in hospitals. The healthcare sector is facing several impacts, such as cancellation of regular care. This is the impact all over the world. It is about a big study about source of IQVIA. 50% regular care is cancelled, 8 million fewer patients per week. This is all over the world. About reduced visit to emergency department for public sector is 50% drop, private is 40%. Outpatient and ambulatory care, public 80% drop, and private 50% drop. Inpatient admission 50 for public and private 40. Regarding estimated drop per segment, cosmetic 100% drop, dental minus 95% drop, and primary care minus 70, allied health and secondary care. Cancellation of surgery also, about new hip, cataract, incontinence, varicose vein, plastic surgery. So it's affecting almost all the healthcare structure. A major decline in respiratory and primary care and allied health, despite outstanding growth for primary care product during 18-19, following a new trend of insurance amended policy about starting basic plan, like you will hear about the Dubai Healthcare Authority and the Healthcare Abu Dhabi. Also, they start to apply a new system for a basic plan to cover all the patients, even the labor and blue collar. The hospital solution was increased abruptly due to absorb a huge number of hospitalized patients during COVID. This market started to rebound along with the East COVID pandemic restriction. Challenging faced by the healthcare industry become alarming, as government is supposed to be ready for possible shortage in supply within 5 to 6 months. Drug shortage and happened as a result of an temporary ban imported from India, China, and even GCC country. So it's about shortage of product. Region facing an ability to meet the sudden demand for medical devices like ventilator and the patient monitoring kits. GCC authority were very smart by giving exceptional importation permits, approved drug for compassionate use, and support the local drug maker like Jorfar Saudi, based drug maker, and Kuwait Saudi and Kuwait. The challenges faced by the healthcare industry, patient enrollment issue, inability to engage patients virtually and manage real time, created delays for completing many ongoing projects. Elective surgery, non-emergency surgery, which can be delayed or cancelled without jeopardize the patient health. Launching a new pharmaceutical product become very risky due to the lack of supply and challenging access to healthcare professionals. This is about post-pandemic era and what's the future and the opportunity. About the e-health, now starting many medical hospitals and medical centers, they start to have consultation even online, and this will support the healthcare structure, and he can even make a refilling for his medicine while he's in the home. And there's a lot of initiative from Dr. Ali Sayed about Dubai Healthcare Authority, that's the delivery to the patient in home. It's a medicine. Virtual hospitals should be a new focus for growth. Look down online pharmacies like in Nahdi Pharmacy, Aster, here in UAE, Medicina, etc. in the region. The era of online webinars and virtual conferences become increased as alternative for physical traveling. 5 trend in e-health in GC, artificial intelligence augmenting healthcare workflow and decision making, massive investment by government, especially Saudi Arabia and United Arab Emirates. Diagnosis through multiple technical solutions, wearable to improve patient experience with biosensors. About digital transformation, pre-pandemic, all pharma companies employed the same go-to-market model. Sales representative visits doctor in person and outlines the value of their company. Now, following this, rejects the product use. This model was common practice for decades. After the outbreak of COVID, it changes the matter to start to have a less face-to-face and start electronic or omni-channel rep. This is about digital, and now we have a hybrid. Most of the conferences now start to be hybrid. About telediagnostics and moving forward to a surge of innovation in remote diagnostic. Also believes that 5G will play a major role in operational success for all these innovations. Seamless distribution, the effect of COVID highlights supply chain about disruption and vulnerabilities, and now they start to have a strategic stock in distributor, and Ministry of Health start to make a safe stock for all important medicine, especially the chronic. Here is a very important point about UAE, about located within 8-hour flight, that UAE starts to be a hub, can be a hub for all the region around and all countries, and to be distributing for other countries in the region, especially they are very centrally located and have a very flexible and agile system, which is allowing more resilience, and how they manage to be an excellent hub for pharmaceutical in the Middle East. This about growth opportunity and what happened after COVID. There's a lot about Arabio, and this actually is my holding company. They start to be a manufacturer for vaccines and biopharmaceutical, and this cover a lot of vaccine supply, around 1.1 billion Emirate dirham distribution in the GCC region, and also about Hayat Vax and from also Abu Dhabi. So KC and UE started seriously to minimize the drug importation and to localize the industry even for biotech and high potent medication, and this by Yes Holding, Abu Dhabi ADQ, Abu Dhabi Qabda, and also G42. Thank you.
Thank you very much for your presentation. Any questions?
Thank you for all that information. This is important. I think it's you specifically spoke about digital adoption somewhere in your slides. I just wanted to ask you, and this is a very specific question. How is the Middle East solving the issue of counterfeit drugs with technology, with the digital adoption? Has there been any advent in the use of blockchain technology recently, like the West?
Yes, many thanks for your question. First, about this, they start to make a serialization and QR code in every box. So you can monitor this box, it is original or not, from the start. And before you don't know which is this medicine in X pharmacy, from where you get it. And you can't monitor. Now every pharmacy have a system about that this about serialization project, and how the UE here applying this since 3 months, and it is restricted. And even the medicine which came from outside, they will not accept unless they have a serialization on it and matching with that. So any machine can make a reader or anything. He know very well that where is this medicine located exactly. in which store, in the distributor or in the pharmacy. It is transferred from the borders by air cargo or by sea or by land and reaching the distributor in UAE. And after this, how this will come to go to the pharmacy and tracking all this process. And should be there's a data logger that to maintain a specific temperature as per the specs. Yeah, for example, this should be stored below 20 degrees centigrade. So anything above 20, it will be rejected the batch. It will be out, not for use, because affects the stability or the product. And also it will control the counterfeit and smuggled medication, which actually it's contribute like a second trade all over the world after weapon and military issues.
Thank you.
Thank you.
To be self-sufficient in production of the pharmacological products is a good thing for any country.
Yes.
But in case of this COVID pandemic-like situation, where there is shortage of the vaccines or medicines in any country in the world,
yeah,
India has donated a lot. So if it is a standardized way, so why not to accept? Why you are banning India, China for accepting the medicines? In the benefit of patient also, in such a critical situation, every country has to help other country if they are
sure,
self-sufficient in their own way.
Yes.
Because one of the statements you told like that,
yep,
it is banned from to import the medicines from India, China, etc.
No, actually, let me clarify. It's not about banning, rather than it is increasing the contribution of API. Majority of around 87% from total raw material all over the world, it is from China and India. China was contributing since 5 years ago. It's contributing around 72% from API. And now it is decreased the contribution of China. It's become 60 plus, and India started to double-digit growing in supplying raw material and API for active pharmaceutical ingredient for all over the world now. So India is playing a lot of big role now about supplying of raw material. Is the issue about, for example, European countries and US, they start to ask about it should be CEB. CEB, this should be CEB certified to pass and to accept the raw material. It is a specific quality measures they have to accept about the impurities, about grading and like this. But India now, they are supplying to even Europe now and supplying for all Middle East. And here in UAE, we have around 16 pharmaceutical company operating in UAE. And some of them, they have a local manufacturing in collaboration with private equity here in UAE.
Background also.
Yes.
Forget about the borders and the political problems and everything. But in such crisis,
yeah,
we are calling in Indian language. Yesterday, Doctor also mentioned Vasudhaiva Kutumbakam. That's a Sanskrit language.
Yeah.
The whole world is my family.
Yes, sure.
Yeah,
sure.
that is the motto of the saying. So in future also, if it is palatable and acceptable, we should help each other.
Yes, sure.
Sure.
Yes, but for your info, for example, if there is some surge or increase in demand, okay, can be managed from country to country as per the requirement or the need. But during COVID, it was unprecedented, and there is a huge substantial surge in the demand, in the requirement, and the demand is very high and the supply is very low. So how you manage this from country to country, but there is a lot of WHO and FDA say, yeah. Exactly. Yes. Yeah. Yes. And also it is not about availability of API only. It's also another issue that even the labor and the human who make this about manufacturer for API raw materials. You can't. Yes. But you can't leave the people come inside. Thank you.
With your permission. Sir, that was a very intensive presentation. And as I've been saying since yesterday, it's time for us to cut the ball as doctors. we should be a borderless world. In 80s and 90s, in my young days, end of the 20th century, I used to see a lot of patients from Middle East coming to Mumbai. Especially, I used to work with Tata Memorial Hospital.
Yes, exactly.
The cancer patients, and I always felt very sad for them because they have to spend not only on their cancer treatment. I'm talking only of oncology. So, have you become self-sufficient now? Has the Middle East in total become self-sufficient in cancer medication?
Yes, I will come to you. About healthcare structure, actually, for the GCC and the Middle East start to have a local, well-developed oncology centers. Even here in UAE, for example, in Tawam hospitals and some in SKMC, Sheikh Khalifa Medical City, they have a very high standard and big team for management of oncology. And also they are covering now the oncology product as a treatment for them for a long period. So now even for expats, they are supporting also. And healthcare insurance, the healthcare insurance companies started to give advantage in different tiers, especially, for example, here in Abu Dhabi, for example, a SICA tier. This is the highest tier. The patient will not carry any, almost no any fees or co-payment. They are covered fully. And also Egypt, they start to localize, for example, some oncology product to be produced inside the country. And even KSA now, they have some pharmaceutical company, they start to produce the oncology product. But API also, we are depending on India and China by majorly. So this is the reason about before there is no, during 80s, there is no oncology center, but now, alhamdulillah, there is a lot of developed oncology center with a very excellent local and expatriates professionals. So that's why. But still there is many traveling to India for transplantation or knee replacement, or there is a lot of issues also still. We have a room of development.
Thank you. But sir, have you become self-sufficient in chemotherapy product?
Not yet. We are depending majorly on branded multinational companies still, because a lot of the guidelines and about first line treatment, there is a new product which came with a less side effect. And this product is patency, actually, it is patency protected, and we can't produce. So we import from outside, and even UAE here, the product, if approved as from Food and Drug Association, FDA, or from WHO, they start to second months, it will be available in the country. Even under what called the name deviation program. Name deviation program, even it's not registered in the country or it's not complete the papers or the dossier and the files of this product is imported direct from outside to the country. So UAE from the first countries and the GCC from the first countries, they make the product available after approval from the WHO and the FDA.
Well, how can we make Middle East and UAE like countries self-sufficient? Can we not open some manufacturing units?
Yes, it started.
Like India or other countries, USA,
yes,
and or UK, which are the giants in pharma industry.
Yes,
and can we not establish there at least branches or something in Middle East so that the local people do not have to depend every time on the export?
Yes, exactly. Yes.
If I am an Arab national and I need those drugs, it should be readily available.
Yes. I live in UAE since around 16 years. Before we have only 2 companies, local manufacturing. Now we have more than 20 local manufacturing facility. Okay, not all working in oncology because oncology is a chemical hazard and it needs a dedicated area separate from normal or from conventional medicine production. So, but around 2 or 3 now they have well-established units and they start to make a localization for these issues. So they try to increase the local content, and the government and the authorities, they start to give incentives and the privilege for this company who make a manufacturing facility on and support an investment to support and promote more facility for high-tech product or what called high potent product in the landing.
Sir, have you ever given a thought to Ayurveda? Because Ayurveda, the Indian system of medicine, has been specially effective in anti-cancer treatment and very effective.
Yes, I just not.
No,
I'm
I'm just putting a thought in
Yes.
the global medical community.
Yeah.
Is whatever treatments, and we are now, I've been doing holistic dentistry. So I basically try and avoid, as you can see
Yeah.
from my technique, I
Yes.
try and avoid as many chemicals going into the body. But Ayurveda, the natural way of healing, can be of helpful since you are a senior person from the pharma industries in the Middle East. I just wanted to put that thought in your mind.
Yes.
Thank you very much, sir.
Thank you, sir.
Answering all my questions.
Thank you. Thank you.
12 years back, we worked for 10 years at Saudi Arabia.
Yes, amazing.
Not Riyadh itself.
Yes.
So we don't have talented doctors they are hiring. Every patient, whatever is not available, Riyadh, means go to Riyadh
Yes,
for the
yes.
treatment. So I think even such a big hospitals, they are establishing in province places. The pharmacy should give a pharmaceutical product should be given to the province hospitals also.
Yep.
Because I have seen, particularly being female doctor, I came across only female patients and
Yeah.
started below 12 years. They find lots of difficulty.
Yes. You're right.
Without anybody driving, they cannot come. So really, and I have seen my patients crying. The son dies of a leukemia in Riyadh. The mother is not been taken to see the face also.
Yes.
So your pharmacy company should try to involve yourself at the smaller places also.
Yeah, we supply for traditional and conventional medicine, but regarding high specialty product like and a very expensive product, so it's not reach until prescribed by a specialized doctor or oncology specialist, and should be from the big major hospitals. Unfortunately, but they can't make oncology hospital in everywhere, like a medical center, normal general specialty. Yes, exactly. Yes, you are right. Yes.
Ajnabis like expatriates don't get justice. That's what was our experience
Yes.
12 years back.
Yes, they encouraging now the private sector also to support the public sector and to be like a balance between public and the private sectors, and start to make a lot of beds, a number of hospitals, huge capacity for beds. Now, and even for UAE, you have around more than 19,000 bed capacity in the country, which is even it is around 60% for private. So it's supporting from time to time, a lot of improvements and substantial increase in the healthcare sector. Even UAE now, they are encouraging to have like what called medical tourism, even for the country from other countries.
So just last sentence, so like without borders, why don't you try pharmaceutical without borders?
Without borders.
On Facebook, we have a group called Doctors Without Borders. Any doctor from any country, irrespective, goes to that country which needs doctors. We don't even, I'm an Indian. Today, if Pakistan needs me, it's my duty first to go and treat patients in Pakistan. Whatever the political issue, I am not concerned.
Yes, you are right.
Pakistani person is my brother, my sister. Same regional country, I'm saying. So why don't you start a movement like that? It's already existing, Doctors Without Borders. Why don't you take a lead and start pharmaceuticals without borders? Let everybody in the world with big countries like USA, UK guidance, let them get and see the supply chain is maintained all over the world, as Madam said. Even the remotest villages in the Middle East should get the same treatment as you get in Dubai or any other.
And also for UAE here, they create about golden visa now for the doctors and the high specialty and the highly educated person to attract the talent and they make talent retention in the country, and how this will support the public or the population. Okay.
Because your son is my son.
Yes, sure. It's very good idea. Yes. Thank you, sir. Thank you, Dr. الله يخليك. Thank you. Thank you. Thank you. Thank you very much.
So now come to our next speaker, Dr. Sidra Arshad from Pakistan. I'm passing her the stage to deliver a speech on a 5-year prospective study on the spectrum of histologic diagnosis in renal allograft biopsies from a tertiary care hospital.
So you can just press this one. Okay. I don't know.
So good morning, ladies and gentlemen. I'm Dr. Sidra Arshad, Associate Pathologist at Aga Khan Hospital, Karachi, Pakistan. So I'll be presenting a 5-year retrospective study of spectrum of histological diagnosis in renal transplant allograft biopsies which were received in our center.
It's okay. Yes.
Okay. So not to forget this contribution that the first successful renal transplant was done in a Massachusetts hospital by a doctor at Peter Brigham Hospital at Boston, Massachusetts. So this was the first successful renal transplant done. So despite significant developments in the diagnostic modalities for immune injury in renal allograft biopsy remains the gold standard for evaluation of graft dysfunction. On an average, biopsy findings change the clinical diagnosis in 36% and therapeutic management in 59% of the cases. Of renal allograft pathology was developed in an attempt to standardize renal allograft biopsy interpretation and reporting. The goal of the use of this classification is to be able to give a diagnostic biopsy grading that will provide both prognostic and therapeutic tools. So talking a little about the evolution of Banff classification, until early 19s, there was no standardized classification of renal allograft biopsies, resulting in considerable heterogeneity in the reporting. Kim Solis, along with pathologists from Johns Hopkins Universities, met in Alberta in 1991, which led to the creation of the first standardized international classification for biopsies of transplanted kidneys. So this is where the first BAF meeting was held at Alberta in 1991. So every 2 years, now the transplant pathologists meet clinicians, surgeons, immunologists, and researchers from more than 30 countries get together for BAF meetings. BAF working groups were established in 2009 meeting with the aim to conducting multi-central trials to evaluate the clinical relevance, practical feasibility, and reproducibility of potential evidence-based changes in the BAF classification. Now, BAF working group, every 2 years they meet and they modify their criteria, and then they recommend certain diagnosis and reporting patterns. So the last meeting was held in September 2022, and in which they have also actually developed a digital pathology working group, and they are also going to now establish certain criteria and just to standardize the artificial intelligence and digitalization in now renal transplant biopsies as well. Now coming towards our study, it was a retrospective 5-year study, which was done over a period of June 2017 to May 2022. And we actually included all the relevant clinical details, including age, gender, the biopsy duration, post-transplant, pre-transplant renal functions were also recorded, and which were compared to the post-transplant renal function tests. So these were studied, all biopsies were studied according to the updated BAF 2019 diagnostic categories and frequencies of various etiologies. These are the 6 categories in which we divided our findings. So category number 1 is a normal or non-specific biopsy. Then we have antibody mediated rejection. Then we have suspicious or borderline for T-cell mediated. When we have T-cell mediated rejection, which is going to be further subdivided into further categories. We can have also interstitial fibrosis, IFTA, and tubular atrophy, which is known as IFTA, and then other causes, which was a miscellaneous category. Now, talking about the adequacy of this specimen, this is what the BAMF has given, that the specimen within at least 10 non-sclerotic glomeruli and 2 arteries. A minimal acceptable biopsy should have seven glomeruli and 1 artery, and the specimen with less than 7 glomeruli or no arteries or with only medulla are considered non-diagnostic. So this is one of the image of the H&E taken from a renal transplant biopsy. We analyzed the changes in the renal cortex only. The number of fields are counted at 40x, and we actually see the infiltrate in the renal cortex only, right? So this is just an overview of the BAMF categories again. So there were out of 301 patients, 242 were male, 59 were females. The ratio was 4:1. The mean male age was 37.4. The mean female age was 31.7. And the biopsy duration post-transplant, they were from 0 days to 9 years. Serum creatinine level range was 1.2 to 11.84. The most common clinical presentation was the rise in the serum creatinine, followed by a decrease in urine output and proteinuria. So this is actually the gist of the study, but I'll be further telling you in detail. So this is how we divided the categories according to the BAMF. Like I said, normal, antibody mediated, borderline, T-cell mediated, IFTA, and the miscellaneous. So the most common cause of rejection was T-cell mediated rejection, followed by antibody mediated rejection, and we had a few different cases in the miscellaneous group. There were a few cases which had combined T-cell mediated and antibody mediated rejections, and also a few of them were also associated with infections. So breaking down the T-cell mediated rejection, they were acute T-cell mediated rejection, which was further graded into 1A, 1B, and 2A, and also chronic changes were also seen. So this actually tells you that the most common T-cell mediated rejection was acute phase, and which it was graded as 1B. And in the miscellaneous category, we saw a number of cases which had infections, certain glomerulopathies, the pyelonephritis, which was the most common in the miscellaneous category. And these were the different glomerulopathies which we saw. The most common was IgA nephropathy, which actually shows that it is one of the diseases which comes back very easily, and patients usually they don't respond well to transplant even. So talking about T-cell mediated rejection in detail, as I said, it is further subdivided into type 1A, B, 2A, and 3, which how do we grade it, right? So it has certain criteria. We need to see inflammation, and we then also grade the inflammation. So by 1I, I means that the inflammation is in less than 10% of the cortex. I3 means that the inflammation is in 50% of the cortex, right? So then it's the inflammation, then it's tubulitis. So we scored tubulitis as well, depending on the number of lymphocytes entering into these tubules, right? So a foci of more than 10 cells in the tubular section was said to be T3, and if it was T1, if it's 1 to 4, and T0 if it's no inflammatory cells within the tubules. So just to explain this further, this is T1, and this is one of the lymphocytes, this is another lymphocyte, so it's 1 to 4 lymphocytes. So this is why it's T1. Why is it T2? Because it's more than 4 lymphocytes in this cross section, like 1, 2, 3, 4. So it's more than 4. So talking about T3, it's more than 10, as I said. So this is how you further rate or score tubulitis in the diffuse. So then you can also have criteria of seeing a vascular arthritis. So by we score it by V0 to V3. V0 mean no arthritis, and V3 mean transmural arthritis or fibrinoid necrosis. That the inflammation involves the entire vessel wall. So we're talking about, now telling inflammation, it's actually, as I said, I2, I3, which is more than 50%. So this is I2, this is T2, which is involving, it's less than 10, but more than 4 lymphocytes within the tubules. And acute vascular lesion type 2A. So we actually then, what do? We grade all of these: inflammation, tubulitis, vasculitis, and then further subdivided into T-cell mediated type 1A, type 2A, and so on, right? So this is type 2B because the vessel is involved. This is why it's type 2B. So acute vascular rejection, another image showing inflammation within the vessel wall. This is a V3 lesion. It is actually involving the entire vessel wall. So is this. If you see chronic changes, like if you have intimal fibrosis and foamhistiocytes, these are the foamhistiocytes, then you call it chronic active T-cell mediated rejection, right? And in acute antibody mediated rejections, we can have histologically, we can see evidence of glomerulitis. So you can see polymorph within the globe. This is a glomeruli. You can see polymorph within the glomeruli, and then you can see PTC element of PTC. That is, there is tubular capillary, the small peripheral tubular capillaritis, and then what is important is the C4D immunostain. So this is a positive immunostain which is staining all these. This is staining positive. So in addition to this, you can also correlate with the serum donors antigen levels, and this is one of the cases which showed antibody mediated rejection. Now, talking about chronic allograft failure, you can have a number of conditions, like you can have tubular atrophy, you can have interstitial fibrosis, glomerulosclerosis, and obliterative arthropathy. Going into detail of interstitial fibrosis and tubular atrophy, this is also further graded. It has a 3-tier grading system, and of grade 3 is more than, if it's more than 50% of the cortical area is involved. This is one of the image showing fibrosis within the tubule, and also atrophic fibrosis in the stroma, and also atrophic tubules, right? So as I said, that if this category is known as IFTA, and this is also graded on the basis of interstitial fibrosis and atrophy seen in the renal biopsy. So more than 90% of the case of the renal transplant patients have infections in the first month, which can be bacterial, viral, and fungal. Infections occurring later are usually viral and fungal opportunistic infection, which include CMV, BK virus, and it can also have aspergillus fungal infection. So this is acute pyelonephritis. You can see a number of mixed inflammatory cells, lymphocytes, plasma cell, neutrophils, which are present in the stroma as well as in this tubule. So this is a very good picture of acute pyelonephritis, which we see in renal biopsies. So this is one of the case showing a large cytomegalovirus inclusion, which is also highlighted on immunostain CMV. So this is also another inclusion, viral inclusion seen within the tubule of a transplant patient. BK polyoma virus, you can see these viral inclusions within the tubules here also. And this is SV40 immunostain, which is highlighting these viral inclusions. This brown stain shows all positive. This is a positive stain showing that these are all viral inclusions. So polyoma recently, when there was this meeting, there was is a working group on PVN has developed a morphological classification a comparative to actually increase the comparative outcome analysis. So two independent histological variables were found to be the most significant predictive for graft dysfunction and graft survival. Number one was the intraparenchymal polyomavirus load. Then there was this BAM interstitial fibrosis scoring as well. So recently, they've actually really developed this scoring system, which further enhanced the standardized reporting as well. So this is what we did was we did actually further characterize our polyomavirus cases on the basis of this BAM scoring system. So post-transplant lymphoproliferative disorder, in this EBV plays a central role in the pathogenesis of post-transplant lymphoproliferative disorder. It is usually commonly extranodal and most are B cell origin. However, T cell post-transplant lymphoproliferative disorder has also been reported in 10 to 15% of the cases. This is one of the miscellaneous group category, which is showing acute calcineurin inhibitor toxicity. What we see is this isometric vacuolation within the tubular epithelium. But it's different from and then sometimes it can also present as thrombotic microangiopathy. You can see this fibrin deposits within the glomeruli. But chronic calcineurin inhibitor toxicity is somewhat different. What we see is you usually see a stripe pattern of fibrosis. You can see a normal kidney, you can have fibrosis, then you can have tubules within, then fibrosis. So this is a stripe pattern of fibrosis characteristic for chronic calcineurin inhibitor toxicity. This is another case showing chronic calcineurin inhibitor toxicity with nodular hyalinosis of the arterioles. So acute tubular necrosis was also seen in many of the cases. It is the most common form of acute tubular injury that results from prolonged war or cold ischemic time during the transplant. It usually manifests an immediate post-operative period. So what we usually see is, if you see this tubule, it has these beautiful columnar cells with these round cells, but what happened is if there's acute injury, these cells get flattened up, right? They're all flattened up, and there's flattening of the lining epithelium. So this is called acute tubular necrosis. So coming towards the results, the most common transplant pathology which was seen was acute T-cell mediated rejection, and our study it was comparable to different studies conducted in national and international forums internationally. So this was another study which was performed at which was done at Force Institute of Urology. They also concluded that the cell-mediated rejection or T-cell mediated rejection was the most common renal dysfunction, and however, it can also be in association with other pathologies. Another study which was conducted at Shaukat Khanum Hospital also showed similar results. They also had T-cell mediated rejection as a most common pathology seen in the post-transplant patients. So this was another study which was done in India, which also showed similar results, and they also had acute cell rejection in the most common cause, and some of them had both acute and T-cell rejection and also mixed antibody-mediated rejection. So concluding, T-cell mediated rejection still remains the major cause for graft dysfunction, which was seen in 48.5% of the cases, followed by antibody mediated rejection seen in 31.2% of the cases, and IFTA seen in 33% of the cases. Recurrent glomerulopathy constitutes 5% of the graft dysfunction, while viral infection was present in 3.6% of our cases. So biopsy still remains the gold standard for diagnosing the exact cause of graft dysfunction and in planning further management. Thank you.
Thank you very much for this presentation. More questions?
Do you have any strategy for post-transplant renal biopsy? That you biopsize all the patients after transplantation, or you wait for rejection to take place and then you take biopsy?
Okay, so I am a pathologist, right? So this is what the nephrologist has to decide. So when is he going to do the biopsy? This is what he decides, and it really depends on the clinical symptoms of the patient. So it depends if the creatinine levels he does monitor, right? So if the creatinine levels are not coming down, so sometimes he does hydrate the patient and see. But if the creatinine levels are not coming down, and he sees that the biopsy has to be done, this is what he decides. I don't decide that.
What if you have some strategy that every renal transplant patient should have biopsy after 1 year or 2 years, 3 years, whatever? I mean, I'm not.
No, I don't think so. They have a certain strategy.
Please listen to my question. Will that help in preventing more rejections and saving more patients? Will that help? Or you would rather wait for rejection to take place and then biopsy and then decide for treatment?
Recently, what I discussed it with the nephrologist as well, they did have they do have a follow-up, right? So when they're actually following the patients, they do see the clinical symptoms. So sometimes they are working on it, that maybe they can they have a certain criteria. But what usually happens in our system, because it's a third world country, and they don't usually have a standardized criteria that they're going to do it after 6 months or a year. So they do it according to the symptoms of the patient.
Do you have any specific biomarkers for rejection, which would detect rejection earlier than biopsy?
So what? Okay. So what for that they can do some for antibody mediated rejection, they have serum antigen levels. Certain for example, if they're also suspecting infection, they can also do a viral loads, and for certain there's certain they can also do some immunophenotyping, so that can also help them with the before biopsy they can do that as well.
Thank you.
You're welcome.
1 second, I just want to comment on the renal biopsy. I think, as she answered clearly, that clinical parameters are the guidelines. I mean, it's easier for the patient to draw blood rather than doing renal biopsy, because it's more basic renal biopsy, and it's kind of inconvenient for the patient. So I guess the follow-up, the clinicians, what they usually do, they follow up with the creatinine level, because if the creatinine doesn't go down, I mean, there's something wrong, or the kidney did not engraft correctly. But if the clinical parameters, I mean, the clinical condition of the patient, the lab, like antibody levels,
right, true,
all the blood or the serum level of these antibodies, or the creatinine, urea, and all the clinical parameters, it's easier to follow the patient with the clinical, with a simple blood draw, rather than doing an invasive renal biopsy procedure. So that's I guess that's why they only hold the biopsy unless there's a strong clinical suspicion of rejection. Okay, this patient creatinine is not going down, so we have to schedule him for a biopsy. So I don't think they do prophylactic kind of biopsy.
Yeah, there's no follow-up with the biopsy, right? So just as he said, the creatinine levels are very important, and even sometimes they still wait that for the creatinine levels to come down. They hydrate the patient, they see if they can do they do other tests, they see if it's an infection or something, and then if it's not responding to the other medical treatment, then they go for biopsy, because it's an invasive procedure, right?
Kidney donors, so there is a big list of waiting.
It's very difficult. Actually, what happens is that there's some institutes who only take related donors, right? So it's very difficult to actually there's a or otherwise it has to be, we don't have any donor, bank or something, so it's very difficult. So to find a related donor, and sometimes it's also on an exchange basis. So it's not easy to find a related, to find a donor in our country. Yes, yes, and I know a lot of people also from our country also go to India to get their transplant done. Yes, true. It's very important.
Okay, thank you for this nice presentation and this very important topic. I have a lot of practical questions. At first, regarding the antibody-mediated rejection, about C4D assessment, is it important to assess the C4D positivity only in tubular capillaries or in glomerular tuft also? This is number one.
Okay, so what happens, regarding C4D, there are two parameters. You can either see it via immunostain, which is just seen in the peritubular capillaries. The control usually is seen in the glomerular tuft, but the positivity in case of rejection is seen in peritubular capillaries only. Right, and also you can also see the C4D staining via immunofluorescence, right? So it also has that pattern of staining, a significant pattern of staining.
Oh, so in IHC just in peritubular capillaries.
Peritubular capillaries,
Oh,
yes.
And we neglect the glomerular.
That's a normal control.
Yes.
That's the normal control. You, in case if you want to do your quality assessment, you need to see that it's present in the glomerular tufts. So that's a normal control. If you don't have, that's an internal control.
Okay, regarding CD3 and CD20.
Okay.
Is it mandatory to stain?
Okay, so if it's, sometimes it's difficult to see the number of lymphocytes within the tubules. So only in those cases we do CD3, but not CD20. CD20 is just, to see atypical lymphoid B cell infiltrate. So we don't do CD3 in all the cases, and it is not even mandatory. We can also assess these lymphocytes on H&E sections, but sometimes it is very difficult to categorize it further if it's a T2, T1 versus T2 lesion, right? So in those cases, we can perform CD3 immunostain.
The question regarding the CMV and polyomavirus
Okay.
is routinely to stain IHC for CMV because it's sometimes suspicious in most of the cases of as a cause of rejection.
Yes, we do it. We do it, and for if I whatever in a renal transplant case, when I receive it, we need to have at least 5 H&E sections with 3 levels. Number one, we have SV40 immunostain as in that tray. We have a CMV stain, and then we have special stain, trichrome, silver, and BS stain. They are routinely done for renal transplant patients, because sometimes CMV, you can it's so hidden, and you can always miss it. So we routinely perform CMV and SV40 in our institute.
But there is no IHC for polyomavirus.
SV40. SV40, yes.
Okay.
SV40 is for polyoma. Yes, it is available. I've even shown you one of the slides. It is available. We routinely do it. It is a routine panel in our transplant biopsies.
Regarding the final categorization of the rejection, in your study, do you found there is a mixture of more than one category, like T-cell mediated rejection and delta?
Yes, there was. So even it was the T-cell mediated was also in those patients. We also saw that they also had antibody mediated rejection. They also have chronic changes. So they also have IFTA. There was a mixture as well.
So should we categorize as a special column?
No. According to BAF, it's not categorized as a special, as another category. But we do have to mention that in our reporting. While we are reporting it, we have to mention that it also shows IFTA and how much percentage of IFTA is that. But it does not have a special or a separate category that it's a mixture of T cell and IFTA. But we have to mention how the percentage of IFTA. You're welcome.
This is not a question, but I have been always listening to doctors from various specialty that biopsy is an invasive procedure. I think like as you have a BAF criteria of renal transplantation, we should have a UGC criteria now. We can establish that and let us declare that biopsy is not an invasive procedure, so that doctors will not hesitate to take biopsy before the disease occurs. Because this is a common problem with us. I'm a maxillofacial surgeon. General dental surgeons are avoiding taking biopsy from oral precancerous lesions, and therefore we have a more oral cancer incidences in Southeast Asian countries, including Pakistan.
Pakistan,
Yeah,
true,
you know
true.
that.
Yes, true.
Right? So can we similarly suppose clinicians don't wait for serum creatinine to go high? Using criteria, okay, renal transplant done in 2022, biopsy has to be done after 3 years or whatever. I mean, I'm not renal surgeon, so I don't have idea. So remove that thing from your mind that biopsy is an invasive procedure. It is not invasive. If you remove this thought from your minds, you will take more biopsies and possibly save more lives.
Yeah, but the problem is like I'm a hematopathologist, so I cannot talk much about the renal pathology.
Yeah.
But for example, the bone marrow biopsy, which is even worse than the renal biopsy, it's done kind of mandatory. We have protocol. So we just go by the protocols or the guidelines. So let's say the patient has a chemotherapy. We do regular, as you mentioned, there's guidelines like day 14 marrow, day 28 marrow. And after the transplant, we do follow up the patient by the marrow. Just based on the CBC might not be enough reflective of what's going on in the bone marrow. But I think they're thinking, that's my assumption, why they are doing or following up the patient with a clinical parameters and serum creatinine and other stuff, because I guess they feel they might be more reflective, or it's easier for the, not just easier and less invasive for the patient, might be as good as a renal biopsy, or they do not hesitate if there is a clinical suspicion. Clinical suspicion, the patient is not improving, the serum parameters are too bad. Like on the other hand, on the other side, the hematopathologist, we do a lot of bone marrow biopsy, but because there is no other way or no less invasive way to determine what's going on in the bone marrow without doing the biopsy. So I think this is the way they're thinking, and nephrologists, I guess they do have their own protocols.
Yes. So what is important is that you need to follow a step, you need to do things stepwise. You can't really jump to a biopsy. So yes, you're right. We really shouldn't think that it's a very, it's a very, but
Doctors, biopsy invasive, patients are going to believe us, isn't it? They say, oh, no, I don't want to undergo biopsy. I've seen patients with a frank malignant ulcer in the mouth, and they refuse biopsy because they think that we take biopsy so that we make more money.
so then it really depends
So
on
that this thought process has to be removed from not only doctor's mind, from patient's mind also.
It is labeled as like it's a potentially fatal procedure. Like renal biopsy rather has been well presented, and renal biopsy are an asset for the person. We take multiple levels, we try to get the diagnosis out of that single board. The reason behind that was
Biopsy being taken by nephrologist. Needle in the kidney, take it out. It's so simple.
So it's not that easy. And you have a pathologist who says the adequacy, so you might stick the needle 3 times, and you just.
This is just a light, don't be asking this. Just to.
maybe you're right. It shouldn't be considered as a taboo.
Use this statement that biopsy is the invasive procedure. that statement has to be removed from all the textbooks of medicine. If biopsy is necessary, it's necessary. Whether you have to open a heart, biopsy, or go into bone marrow, or go into the mouth, wherever you want to go, you have to go into. One saying is invasive. If it is, so in your mind, you avoid biopsy as a doctor, and then when you patients are going to avoid obviously, isn't it? So as a pathologist, you go and tell your nephrologist that biopsy is the must. It is not invasive. You agree, Mariam? She is also a pathologist from Iran. Do you agree?
Fears when you did. Biopsies.
Absolutely, if the biopsy is needed.
So whenever it is required, it should be done, right?
Yes.
You can't really, say that it's invasive, so you're not doing it, and you can't really always say that you have to do it. Just go for this at the first time, So it has to be done when it's required. That's it.
You uttered those words, and he also uttered. So I got a little bit emotional.
So what's so bad? No. So what's so bad in calling something invasive? I don't understand. It's not so bad in calling something invasive. Yes. So if it's invasive, you have to do it, right? When you have to do it, you have to do it.
Now we have last topic.
Diagnosing the lesion, and it is a pre-pregnant entity that can be swapped. So I agree with you that there should be the staff should be removed and it should be labeled like the patient should have a biopsy. He has a suspicious lesion in the oral cavity. But regarding the renal pathology, it has to be needed and absolutely needed. Because our important principle in this regard textbook is it's a potentially life-threatening procedure for renal biopsy. Because you have to insert the probe through the flank into the kidney, you have major vessels around. So the thing with oral pathology,
Die. When you take renal biopsy, that means you never understand. Tell them to bring something that's
So if biopsies are done so liberally, we will discover disorders.
Exclusive or not.
Pathology is not yes, we love the tissue. Every organ.
Yes.
We will tell the world more technology than is existing now. If we have tissues from every organ, that would be.
If pathologists are so well trained,
Yes, sir.
clinicians are not so well.
The clinicians, they take the decision according to the patient's requirements. That is up to them. But we will love tissues from every organ.
Okay.
Thank you very much.
Maria, you didn't say anything. One minister, she's our.
She wanna say something. Okay, you are VIP pathologist. Then this side, okay, got it.
And then about which organ you want to biopsy and what you face to which region. For example, maybe a surgeon asked me about why we biopsy STC. Patient come back with multiple STC with metastasis to other lymph nodes. When you use knife or biopsy, it's make a way for malignant cells to be made easier from lymph nodes, from blood cells. But I can't say with confidence. I agree with invasive or non-invasive biopsy.
Thank you very much.
It should be done.
Yes, true. Anything should be, you should have certain criteria for doing anything, whether it's a biopsy or, whatever. And I'm a pathologist, I'd love to receive biopsies, so never mind. Thank you.
Thank you very much.
Thank you.
So our first keynote speaker of today to now is Mr. Steve Barbie from the United States. I'm passing him to deliver stage and keynote speech on the future of pathology is digital, so it must be very interesting topic.
You can use Slido. You will do the here? Yes, you can do it because.
Which configuration का कर रही answer में
So I'm not sure if my presentation is going to be as exciting as the last one. But it's definitely from information technology side with respect to pathology. Myself personally, I've been involved in digital pathology as investor, owner, founder for the past 25 years. 22 years ago, we put in some early digital pathology systems in the region. One of the leaders was Dr. Salah at Kuwait Medical Center, working with Princess Margaret up in Toronto with Dr. Azza. There was also a big project I worked on with MODA in Saudi Arabia with the Ministry of Health, Ministry of Defense and Aviation. And then we also had a project with the National Guard in Saudi Arabia. And then in Abu Dhabi, we worked with Tawam Hospital, who was associated with Johns Hopkins in the US, so they were able to do consults back and forth. And then I heard somebody else say this group too, but with Sheikh Khalifa Hospital, who was affiliated with Cleveland Clinic, we also did early digital pathology projects there. And so what digital pathology is really the capability of digitizing a glass slide, either an H&E slide or an ISC slide, and then putting it somewhere in the cloud or a data system, and then somebody remotely could look at it. The number one example people used it for a long time ago was not everybody in pathology, from what my doctor friends have told me, are subspecialty trained. So the capability of using digital pathology is you might be a generalist pathologist somewhere in the world, and then you could access a subspecialty expertise in the bigger cities in the different countries and those kind of things. And then the second idea came out with respect to diagnosis with pharmaceutical treatment, specifically with Genentech and chemotherapy for breast cancer. So they needed a way to stratify the patient pool on who should take the Genentech product for breast cancer. And that's came up with the early digital pathology image scoring systems for IHC for breast cancer, where you score it 1, 2, 3. And then that dovetailed into percentage scoring with Ki-67 and other kind of IHC stands. So it's been around for about 25 years. I've been involved in it about 25 years. I'm 55 today. I started when I was 30. So I'm really passionate about this. And any questions you might have, I deal with clients throughout the world, and friends are my pathologists throughout the world. Reason I dress like this when I present is one of my best well-known friends. I think he has the most articles of digital pathology on PubMed, is Dr. Anil Parwani, whose heritage is from Pakistan and India, and his wife's Indian. And I met him when he was a resident at University of Pittsburgh Medical Center, and he sort of made a career out of digital pathology, and now is the chairman at Ohio State. And we're good friends. And so when he does presentations, he dresses like this because one of the advantages of digital pathology is you can dress like this and read cases, and you could be from home. So that's an advantage too. Oh, I got you. Okay. Okay, so the topics to talk today is a program called Digital Pathology Today, which I'm affiliated with, and we want to talk about implementing it in other regions, including India and the GCC countries. Sort of the trends of digital pathology and some case studies take away, and then usually towards the end, there's a lot of questions, so I'll save some room for questions. So Digital Pathology Today, if you look it up, digitalpathologytoday.com is a weekly series that focuses with interviews of people that have used digital pathology over the past 25 years. It started as the first sort of podcast website for digital pathology education, and now there's probably 10 other people that do the same sort of idea. And one of my ideas is to take what we've done in the US with Digital Pathology Today and have maybe Digital Pathology Saudi Arabia, Digital Pathology India, and have the guests and the moderators be local persons, sort of break it up, sort of send it out to the field and to the masses. What it does is it just brings, different ideas of people that have used it. It's run in the US by a doctor friend of mine. His name's Joe Anderson. He's a molecular pathologist, and he's been involved in digital pathology for many years. We've had over 200 guests, some internationally, and some of the folks up on the screen, you might know who they are just from their names and those kind of things. And our first guest was Dr. Parwani. And then our second guest was Dr. Parwani's partner at Pittsburgh, who now runs pathology at Michigan, a person by the name of Dr. Laurent Petenowitz. And so over time, more people have done it. It's focused both on industry and users, and also on economic models and budgeting. Because a lot of times, one of the first questions people ask is, it's too expensive. How could I afford it? How could I do it? Et cetera. And there are case studies and business models that the different doctor friends do. We've had listeners of approximately over the past 5 years, about 25,000 people, and they've ranged from almost every country in the world that participates in it. And that's where the idea comes. And if anybody's interested afterwards to talk about it separately, is how we could do one specifically in regions like the Gulf Coast countries and India, and partner with local folks to do that, because I do believe, I know pathologists are trained very similar, but there's different protocols and pricing and business models and things that work in other countries that are not US focused. And then at the same time, because it's a technology-based education system, we could get on experts from the US or England and Europe, and then partner them with people in India and the Gulf Coast. So if anybody's interested, let me do it. It's really cost effective, doesn't take a lot of money, if at all, because it's mostly free tools. And as I mentioned, Joe runs it, and there's the website. So don't hesitate to, I think we do it every Tuesday and Thursday live. So if you could join, that would be awesome. And if you want to join as a guest, just let me know. I have my email. You can send me an email. We could get you on as a guest, because that's another idea, to have guests by specific different countries to talk about the differences. So with respect to digital pathology itself, one of the things I wanted to talk about was the different trends. One of the trends that there are is people are doing what I call best in breed purchasing. So there's different components of digital pathology. There's slide handling machines. There's different cameras and systems that take pictures of the microscope slides. There's different ways to store it from a workflow standpoint. Clinical workflow and primary diagnosis is a different workflow than secondary or consultation diagnosis, which is definitely a different workflow than research programs. I'm not sure if you heard, but I read about this 2 days ago when I was flying out. Google approached the president in the US and Congress to fund $1 billion project to digitize and archive all the slides from AFIP and Armed Forces of Institute Pathology, which is now part of the Joint Pathology Center. And almost every pathologist in the world knows what that is, or did some training there, or knows people that have done training there. And so that data set will be sort of the first data set that'll be in the billions of categories. Now it's going to take 10 or 15 years, but it's taken 25 years to get Congress in the US to budget $1 billion. And now just getting the president to sign off on it. And from what Google says they're going to do is they're going to give it the images free with the appropriate diagnostic paperwork and the reports out to researchers, clinicians, whoever wants to use it. And so at least in my lifetime, which hopefully I'll have another 25 years, that should be done by then. Also, there's different kinds of AI software that's popping up right now. That's a big buzzword from an investment standpoint. I think I've counted about almost $1 billion in investment in pathology, artificial intelligence. One of the biggest investments was a group that's run by an investment bank in New York called Page. They raised somewhere in the neighborhood of about $450 million. And their idea is really twofold. It's one, to give pathologists tools to more quickly process slides. We all know pathologists, it only takes a second or two sometimes with the slides to read the slides. But if there was a way where you could have the slides categorized and you only have to read the slides that look like they have the most cancer within the slide itself, so it would be able to take out that 80% where they're mostly negative reads. And then you could have a higher throughput for pathologists. The also thing with AI too, which is a new thing, again, it changes every week. I read about it on the plane. There's a handful of groups, one in New York, a spinoff of Mount Sinai Cancer Center, one in Tel Aviv, spinoff, I forgot which hospital group it was a spinoff of. And then there's a group in Korea called Lunit, which is they're all thinking about and trying to work on how you take an H&E slide, run an image analysis program at it, and get the same information you would get from a molecular test. So what that would do is two things. One, molecular tests are really expensive and really controlled by only a certain amount of manufacturers. And an AI slide to read it from an H&E standpoint would cost, dollars, if not pennies. So that's something that's exciting. And I forgot there's a company in Pune doing that too, working on that. And then talk a little bit about the different hardware choices out there. A lot of people always have questions, and then regulations. So these are the main players on the software side to manage image management and workflow for digital pathology images. Some of the names you might know, some of the names you might not know. But if you were to look it up, they're sort of the leaders. The number one leader is Page, who I mentioned, which is out of New York. There's also a company in Korea that just came out about 2 weeks ago called Lunit that's sort of leading in this space. From an AI standpoint, this is oversimplification, but there's a couple of different ways to create an AI program. One is you do it supervised. So for example, I own an AI company. I employ a team of 125 pathologists based in Poland and in Japan, and they're annotating 50,000 slides in different subspecialties. So what it's done for pathologists and my friends is really twofold. One, it's given a new income for pathologists outside of just reading cases. And as I mentioned, there's been about $1 billion invested in AI companies. So, and the AI is only as good as the pathologist annotating the cases. And so it gives a lot of opportunity for people to do side work, or if they're in between jobs or those kind of things, to do it. It's the first time in my life about 2 years ago where pathologists were always my customers, and now pathologists actually work for me. So it's good to pay them back over time for the things that they've done well for me. And I have pathologists in the US, Europe, primarily in Poland, and in Japan. Then there's another kind that a couple of companies do it, and it hasn't really worked out yet, but it's this idea of unsupervised teaching of the AI, which is basically you take data, pictures, and reports, you put it into some computer system, and it generates an AI system. I believe that that can probably be done, but from a business economic standpoint, I believe since the people that would be using the AI are pathologists, that they wanted to know that a pathologist participated in it So for example, Dr. Epstein at Johns Hopkins and Dr. Mahul Amin, which are both leading uropathology or prostate pathologists, they're working with two individual software companies. So effectively, when somebody would be using the Gleason scoring for prostate cancer that was done by the Dr. Epstein team, it would be like Dr. Epstein reading the case. And with the Dr. Mahul Amin team, it'd be like Dr. Amin reading the case. Instead of having to put a case in the mail, ship it out, get broke, all that kind of stuff. So I really think from an AI standpoint, different parts of the world, you'll see AI software programs developed by Dr. blah, blah, blah. And I think that would really be beneficial. The other thing that happens, a lot of institutions outside of the US use this. If you want to take a picture, these are free sites and websites where you could get access to image analysis and AI programs that have been developed in different countries throughout the world. And the reason I bring that up is, not everybody could afford certain things depending on conferences, or if you need to do a prototype project so you could show your administration. Those are sort of groups that you could run small sample sets of 100 cases or 200 cases, and then you could give people feedback and do it. You also, when you go on PubMed, you'll see a lot of reports actually done with those software systems. So in the world of urology, these are sort of the main players in the world right now. A couple of them are in the US. IBEX is in Israel. Area Matrix is in Mumbai. Verisoft is in Istanbul. MindPeak is in Germany. Deep Bio is in Korea. Context Vision is in Sweden. And Lunit is in Korea. So you see the whole idea with digital pathology development, it's a global topic. It's not just US or just Europe or those kind of things. And so what you'll see is those institutions will have to do a couple of things that you might be able to help them with. One, they need to buy data sets. They need to buy data sets. They got to have the data. Two, they need the pathologist to read the data, to then put that into their computer system and create an accurate AI program. So even if AI doesn't look like long-term, it's going to make sense, short-term for pathologists and institutions that own data, this is an opportunity to make income. So for example, Page got affiliated with Memorial Sloan Kettering for their data sets in New York, and they paid Kettering $80 million to get access to their slides for 5 years, right? So if you think about that, so Area Matrix, who's in Pune, and there's another company called INDX in Pune, they're partnering, I think somebody said earlier, with Tata Memorial to access their data sets, and then Tata Memorial owns a piece of their companies. So there's just a lot of opportunities globally. And then on the derm side, it's a little bit smaller. When people ask my advice on which AI tissue types or disease states they should work on, is I always say derm, but for some reason, there's only one derm player right now. And the reason for that, in the US, derm has 15 to 20 million slides a year on biopsy cases for melanoma. 90% of it is negative. And the way economics work in the US, which if you know this, you probably laugh at it, I'm sort of embarrassed by it, but the way doctors are paid is by how many slides they read. So that's why there's so many dermatopathologists in the US reading those cases. And if you had a computer system doing it, you would actually be able to lower the cost of how much reimbursement goes for cases. So in the US, since I've been involved 25 years, doctors used to get paid $150 per slide. Today, they're down to like $15. And they expect it to go, like Dr. Parwani tells me, they expect it to go sub $5 within 5 years. And so as the reimbursement costs down, you got to be more efficient. You got to do that. And so some of the companies I mentioned, the only other one that jumps out is Mechanomind, which is an Israeli company is in this space, and then Pathology Watch is in Utah. But those other two companies I mentioned earlier. Okay, and then to run the image analysis, you need a piece of hardware that takes the slide and makes a picture. That picture tends to be 1 to 2 gigabytes in size. And so these are really the sort of the key players in the space in the world right now. I categorize them in 3 areas: high cost, medium cost, low cost. Just like all the AI companies, I've worked with all these companies and worked for them and helped them with their funding and business plans, including Philips. And Philips or Leica has been around the longest time because they acquired a company that started 25 years ago, which was out of San Diego. 3Histech, you're probably familiar with. It's in Istanbul, one of the early inventors of scanners. KF Bio is a leading scanner company. They sold 2,000 of them in China. And then Cellavision is based up in Sweden. Then on the medium size, there's 2 Indian companies, Afterscan, and that is based in Pune. And then there's another company, which I just found out about yesterday, called Morphle Labs, which is based in Pune too. And then Motic is based in China. Scopia Labs is in Israel, and then Microscan's in the US. And then these 2 lower ones, which I mentioned, Morphe and Grundium. One of the things I've been advocating for the past 25 years is for the cost of hardware to go down, because adoption won't happen if it costs more than a cell phone. It just won't from an economic standpoint. So when I first started, systems were sold for $200,000. These systems down here are sold for less than $7,000. And they do the same thing as the $200,000 system from 25 years ago. There was just an article written with Grundium. Grundium, within 5 years in the US, has digitized over 2,000 hospitals in the US. And then Morphe Labs is going to try to make the same play in India. And probably there, they sell it in India for about $2,000. So you can't really use all the AI tools from a research or a clinical standpoint until you digitize the slides. You can't really digitize the slides until a piece of hardware could do it cost effectively. And so those things have to happen, and they're starting to happen. And then these are just some other companies to watch that I tell people about that you might not have heard of. There's a company that received almost $1 billion in funding, which they're partly owned by LabCorp. If you know who LabCorp is in the US, LabCorp also, I think, still currently owns, no, they manage the laboratory here in Abu Dhabi, I think, the National Reference Lab. And then Kaiser Permanente is a large medical group in the US. And then there's companies in Finland, Denmark, the US, and then companies outside. The reason I put that up is when you go back home, or if you do a project in 2 or 3 years and you're like, what are the names of the companies I should look at? Those are them right now. Yeah. From a European standpoint, there's a group called the Digital Pathology Society. That's a group of pathologists, IT people that are focused on digital pathology issues in Europe. Just from my research and from what I know, some of the leading groups in Europe that have worked on digital pathology is, for example, University of Leeds was one of the first persons to go 100% all digital for pathology about 12 years ago, 15 years ago. And then in Spain, if you know this name, Dr. Rojo, he's been an advocate of digital pathology for many years. 10 hospitals in Paris are all digital now. There's a group in Morocco called Data Pathology that's looking to digitize digital imaging within the Middle East and then also in Africa. Again, the idea is all the costs go down lower, you're able to put in systems where then subspecialty experts could do the reads. either for a fee or for pro bono basis. And then there's a team in Portugal or Germany that is not adopting as quick, but there's a consortium that is trying to fund the government like they're doing in the US with Google to get the government to fund an entire digital network for the public hospitals. So then the next question always people ask is, so what does the FDA say about digital pathology, and what does the IVD CE group in Europe say about digital pathology? Before COVID, it was really a hard answer because they didn't say anything. But when COVID happened and the pathologists started to work from home, the FDA got lobbied by the College of American Pathology, and they wrote a waiver for digital pathology under the FDA rules. So now slide scanners, slide software, image analysis doesn't have to be reviewed by the FDA. It was sort of implemented underneath the FDA emergency rules that were caused by the pandemic. The IVD CE group in Europe has done the same thing to follow that. And if there's anybody in any other territories or parts of the world where they want more information about that, just let me know and I can email it to you, because then you could share it with your regulators or your people that run the hospital groups or the administration policies and those kind of things. And then there's two groups that you should really participate in if you're into digital pathology. One is called the Digital Pathology Alliance, and it's now a group of about 1,000 doctors and regulators throughout the world that have working groups specifically on different topics of digital pathology. And then there's also a group with a website called Pathology Innovation, which is trying to do a similar thing. If anybody's interested after looking at the Alliance, the doctor that put it together is at Mass General, Dr. Leonards, Joe Leonards. He's looking for individual countries who might want to take an advocate role in their countries or regions to do something similar. But it's good tools, especially if you have people on your team that don't know a lot about digital pathology or are new pathologists. It's really good education. And then the other thing is CAP has, with the support and the leadership of Dr. Perwani and Dr. Pantanowicz and a handful of other doctors from the Digital Pathology Alliance group, has lobbied CAP. And just recently, it was announced that CAP was able to lobby the US government. So potentially within 3 years, people that use digital pathology in the US will get paid more money than not using digital pathology. And so you can see that as a trend then to start to go into Europe and into other countries, because saying, look, this is what they do in the US. And the US came up with new CPT codes, and they came up with new ways, and they're going to eventually within 2 or 3 years get paid additional for it. So that was exciting news. So as costs go down, reimbursement for services go up, and then there's more competition. It makes digital pathology adoption in 5 to 10 years look really like a reality. So here's a couple of quick case studies to put together. One is AI-supported cancer diagnostic for prostate automated Gleason scoring. Second one for lung tumors. Third one, which we talked a little bit about, which is IHC scoring. This one for specifically PD-L1. And then also the H&E idea that I told you about, where you could analyze an H&E slide and get as much data as you would get from a molecular test. Then there's this idea led by a company out of Pune called, which I'll explain, called an Omnics multi-data platform, which I think is, Google is probably going to end up going there in that area too. But it's the idea to accumulate all in one place, all the different data points for a patient, and to be able to present that in a graphical / written way. So at least in the US and in Europe, from what I understand, doctors have to go to different systems to get different reports, and then they got to, staple those PDFs all together, in their emails, and it's complicated. Then there's some niche issues that I'm personally working on in pediatrics and transplant. And then there's another issue I'm working on, which is, I mentioned it earlier, it's a CRO for digital pathology companies. So this is just quickly and I could tell you more about it, and you can find it on PubMed. There's a company called IBEX, which was based in Tel Aviv. The chief medical officer of IBEX, you might know the name, is Judy Sandback, who works at Maccabee Health Systems, and she's been sort of a leader pathology technology in Israel for many years. Is they came out with the first software for different prostate, breast, and gastric diagnoses. And then their first client that did their first validation was actually a laboratory out of Puerto Rico. And you might be wondering why out of Puerto Rico, but you can think of Puerto Rico as a small state or a small country, because it doesn't really get managed by the US government. It's a territory in itself, and they manage themselves from a healthcare standpoint. And they're big into talking about, if you ever want to meet the doctors there that work on the system, is they're in front of 3 monitors looking at stuff, and they just think it's like, flying a plane. Then there's a company out of Finland called Iforia, who came up with a way to do automated scoring for the 4 different grades in different type of lung tumor caps. The reason I bring this one up is they're the first person that have worked in the area of lung. So for those people interested in that, this study is on PubMed itself. And if you want to meet the leadership of the team up there, just let me know and I can introduce you. This is a company out of Korea, which is doing the idea of reading H&E or PD-L1 slides to get as much data as doing a genomic test or a molecular test. I'm not sure how it is outside of Europe and the US, but the dominant provider in the US is a company called Genomic Health, who got bought by a company called Exact Sciences. The reason that's important is, from what I understand, those tests cost $2,000 or $3,000 to do it for the different patients. And they say by using this technology, you get the test down to $100. So you'd be able to get as much data of a $3,000 test as a test that's only $100. And so if it's $100 in the US, there's no reason why it can't be $5 or $10 in India, let's say, for example. Well, this is the multi-omics sort of research, and this is a group out of Pune, where they're taking a whole bunch of different image and data sets, throwing it into one image management system to create AI algorithms that are provide more data than just doing an AI application on an H&E slide or an H&E data set. This is probably 5 to 10 years away. But one of the reasons I do bring this up is I'm part of the advisory team on that. And the person that put it together was one of my old bosses at a company called Bioimaging, which got bought by Roche for $100 million. That was one of the earlier AI companies for immunohistochemistry. And he's a serial entrepreneur, has been in the pathology, histology space for years. So if he says he's going to be able to do this, it might take him 5 or 10 years, but he'll do it. And he's in Pune. This is a company I work with too, called MedPoint. Cool thing we came up with here was trying to look at what I call a niche AI pathology patient substrates, where the patient pool isn't as large as, let's say, breast cancer. So, breast cancer in the US, there's about 250,000 new cases every year. There's 5 slides per case. You could do the math, you got the reimbursement. So you know how to put a business together around that. But what I saw was people weren't really creating companies in other spaces. So the idea I came up with the doctor friend of mine out of the UK that lost his child to neuroblastoma, was to create some new tools for pediatric oncology and pediatric pathology, which globally, from what I've learned as we started to put this together, there's less experts in that field in comparison to other fields of pathology. And then for him personally, he lost a child. I lost a child not to cancer, but I lost a child to a different situation. So there's some internal focus and motivation on our part to make it work. Reason I bring that up is if there's anybody interested in that, we need to buy data sets and we need to hire doctors to do annotations. So if anybody's interested, just let me know, or if you go back home and your organization is interested, just email me. And then the other area, which is something that's been worked on with the University of Pittsburgh and their transplant pathologists for the longest time, specifically in liver, which has never really taken off, is there seems to be, if you look up through Google, a lot of investment in transplant pathology areas to make acceptance of organs from one body to another at a higher success rate. And so if they're able to do the pathology earlier on, when they take the liver out of the patient before it goes to the new patient, when it's in travel, and to do pathology in the field, there's a hypothesis that said you'll get more, and I'm not using the right medical word probably, but you get more acceptance of the transplanted organ in the transplant patient. And that's something they've worked on for years, but if you look now, there's a whole bunch of people working on it. Again, if you're interested in that, I could connect you with the people that I know that are in that space. And then what I did, since I've been in the space for about 22, 23 years, I know a lot of people, I travel throughout the world, is I decided to start a company just to help different people, groups, so laboratories. We've worked with Quest and LabCorp, and then a big laboratory out of Australia called Sonic Healthcare that owns pathology labs in Australia, the US, Germany. I've been talking to the folks in India at SRL Diagnostics to work with them. They're owned by a company out of Singapore now. And to just give my expertise with me and my team on how to implement and look at digital pathology and the benefits for that, both from a patient side, a doctor side, but also an IT side. And then I work with manufacturers to help them build products that are more cost effective. And then there's like a website called openslide.org that I started 12 years ago. And it was this whole idea for open source code for digital pathology, because people were selling software applications really expensively. And I knew how much it really cost to build a software application. So that's where I came up with the idea. I no longer participate in it, but if you go to openslide.org, there's a whole bunch of different tools to look at, to use for images, image analysis, databases that are free. And then I've got a small investor group where we invest in different companies throughout the world in this space. So I'll open up the questions. Usually the questions come down to what scanners do I like the best? What software do I like the best? And what AI I like the best? So I just throw those questions up there, but anybody have any questions?
Well, I wouldn't want to ask a question of what you like, but I was thinking, can we develop digital biopsy?
There's actually a couple of companies working on that have been funded on that. So could you define just so I don't define it wrong?
Actually, I was witness to a very piping hot and very spicy discussion that took place just before you on invasive biopsy
Sure.
and non-invasive biopsy and when it should be done,
Sure.
it should not be done. And I remember teaching my pathology class students about excisional biopsy. It's like cutting a, or incisional biopsy, like cutting a cake and taking a piece. I was wondering if we could replace in future, especially in my field, which is in oral cancers. Because I've started awareness, prevention and screening programs. So number one, I should be discussing with you in private personally about the screenings. But I would just wanted to have an idea and thoughts about of the audience on the digital biopsy.
Yeah, so the current state of everything.
Yeah, I'm sorry. Would it be possible from your hardware side?
Not from the hardware that's out there today. There are, I think, 5 or 6 companies I know of that, and again, I'm not a doctor, so I don't want to say this the wrong way, but the patient gets opened up, macro pictures get taken of the organs and those kind of things, and then some software program gets run behind it such that you don't have to do a biopsy. It's taken us 25 years in the US to get the FDA to say it's okay to use a computer on a microscope slide. I mean, it's just taken that long. Right? And so this idea that you're talking about, I would think would be 25 years from now. No, not from a technology side, but from a regulation approval side, right? Because one of the things I've learned, because I'm, again, not a pathologist, but I've learned how we went through this area of digital pathology where pathologists privately were not acceptive of the idea to lose their jobs.
I agree with
I mean, they just, but they were all private. It was like, you know, top secret kind of, and there was never any intention to, I mean, me, I'm an IT application software person, so I'm just building tools. And as I tell my pathologist friends back then, when they said that, I was like, you know, when they digitized radiology, all of a sudden there's a lot more radiology jobs today than there were before. And I learned that from pathologist friends, right? And then, but an idea like that, I think it's there technology wise, but from an adoption standpoint, I would think it would take a long time. This whole idea with reading an H&E slide to then give you the same data set that you would get from a molecular test, they just started on it now. So that would tell me from a technology side, it'll take 5 or 10 years. And then, you know, Genomic Health, who's owned by Exact Sciences, they're going to lobby and try to prevent from that getting approved by the FDA. Because that business that they have in molecular testing is huge. Right. So you got different factors of acceptance.
So in oral cancers, we already have Wellscope, which has
Right. Yeah.
Wellscope, which is being used by a company developed from Canada. So and Wellscope has not been very popular. I think the mindset is changing. I do not want to comment on any political situation in any country because I'm an apolitical man and I'm as we said, Vasudeva Kutumbam, I'm a global citizen. But having seen Mr. Narendra Modi ji working, I'm sure a person like me, a stubborn Taurian like me, would be able to sell this idea to him, and not only able to sell, to get it implemented at least in the next 5 years.
Sure.
This is my hopeful thinking. We are in 22nd December. Say by 27 December, I think it should be implemented in India because Tata Memorial, where I'm associated with, is the hub of cancer center for Southeast Asia.
Of course, sure.
So all countries are coming over there.
Sure.
Having said that about well-class.
Just to say one thing really quick before I lose it, and I forgot the name, but I'll get your email. So in the US, there's statistics that come out every year on pathology procedures and how many are done by organ type and this, that, and the other. And the number one type, and it's in the millions of digital imaging with AI, is for oral cancer. And it's like a monopoly with one company in the US. And I could send you that, and I never really understood, because I've never really worked in oral pathology, why that would be, but it is a fact that it's one of the biggest users of it. Sorry.
I'm sad to share. India happens to be the leader in oral cancers.
Yeah.
And the state that I represent, Maharashtra, happens to be number one in oral cancers. And my anti-tobacco mission started when I was only 10 years old in 1970, and I stopped my father's cigarette. I'm very passionate. Now that I have lost all my family members, I've decided my life will aim in fighting oral cancers, and I give you a promise. I shall show you in next 5 years the rates in oral cancers in India should go down. Because of my dedication, and I'm not going to leave oral cancers. Talking about your presentation, you spoke about artificial intelligence. So I just wanted you to throw a little light on using artificial intelligence from your scientific point on predicting malignancy.
I would say in the space of AI, the investment's been, like I mentioned, almost $1 billion with different companies throughout the world. But the investments in AI and other areas of healthcare, like radiology, for example, is $3 to $5 billion. Based on my 25 years of experience, again, so if the investment's taking place now, there'll be some tool that'll be validated clinically, supported by the pathologist associations, and then reimbursed in 10 years. But those companies that start today are looking down the road for 10 or 15 years, right? Because that's just how startup money comes into the business and stuff. Do I think it'll get done? I mean, I would say yes. I think every country I've visited throughout the world, they have certain expertise in different kinds of AI, and they all have different kind of data sets. So like a data set in India, I say this to people all the time, based on what my mentor Mohan taught me, like the data set in India is potentially different than a data set in Southern California, right? And so you gotta provide applications for your data sets, right? And to do that costs money because you got to hire the people and all that kind of stuff. But on that idea, I would say, yeah, maybe, but I still think it's 10 or 15 years. Yeah.
I have two questions actually.
Sure.
One is the business from the business standpoint. So let's speak as a consumer, a customer, and I'm gonna buy software from you to implement the digital pathology in my hospital. The two questions that come in mind, first of all, how much would you charge me, right? This is the first question that comes in mind. So the first question, you will tell me, okay, this digital slide scanner is not really expensive, and I'm easy to, like, it's very easy to be affording such price for such machine. The problem is the storage. It's like the phones, like the cell phones, says you, it says every time for you, your storage is full. You want to purchase iCloud storage and pay how much money per month for this storage. So storage is extremely expensive. How? What are your thoughts about cost effectiveness as a hospital or as an academic institution or a private lab? How do you decrease the price for storage of the images? Because we are going to send you millions of images, and we want to have a digital archive for our images for the last 10 years. And this is kind of the cap requirement. Each hospital has to keep their slides for a certain number of years. How much would you charge me for keeping digital slides on the system for 5 years, for 10 years, for 20 years? That's one question.
Okay. So there's a question, what I can answer today, and now with Google coming into space, what I think is going to happen. Okay, because this Google announcement just came out, right? A couple of days ago. To store stuff, I know of companies that do it anywhere between 50 cents a slide per year up to $2 per slide per year. And that includes the scanner to scan it, the internet bandwidth to upload it, and then this cloud storage. So anywhere between 50 cents to $2. And the people I know that are doing that are in Europe, India, Japan, China, and US. In the future, I think Google is going to make it free. I believe that. And we're going to all have the same issues that we have today with how many times do I give Google my credit card? And how many times does Google know absolutely everything I do? Or Google on my Android phone knows where I'm walking. And all of a sudden, oh, you know, you're in Dubai, go visit this place, right? So there's going to be this uproar, I think, from patients that'll be like, I don't want Google to know what my family has, but it's all de-identified information anyway. But Google will win within 10 years because, I mean, Google started, for the pathologists out there, does anybody know who Dr. Mahul Amin is?
Yeah.
Yeah, okay. So Dr. Mahul was a consultant in me and him with Google 6 years ago. Because Google was like, why would we want to be into healthcare? And all I kept saying to them is the slides are 2 gigabytes, right? There's no other image in medical imaging where the data storage is so huge. but they will come up with a way to monetize it. I mean, I believe that. So today, 50 cents to $2 a slide per year. Future, going to be free. I believe free to the extent you get an advertisement on the bottom or something like that.
Because if you are like dealing with a huge hospital with millions of slides every year, you want the cost effectiveness, one of the issues they are looking for.
But the other thing too is the vendors. So look at your Roche, look at your microscope companies, look at Agilent, right? These people will pay money to get their ad in front of a pathologist. And that's who Google needs, right? If you're looking at a slide and, you know, every day you see the Roche logo, you're like, oh, I forgot to call Roche or something. I mean, digital marketing could come into healthcare.
So the second question is actually a question or from the legal standpoint as a pathologist. So let's say I'm working in Southern California and I get slides from New York, right? I have to get New York license to be able to sign out this case.
That's what they told. That's what they tell me.
The big lab, like LabCorp, Quest.
They manage all that.
All these ones, you have like at least 20 licenses to be able to join their team, and they would help you with this. But how about internationally? Since you days we have digital pathology will make the whole world as like small village, right? And I'm in the United States might be able to sign out kids from England, from Dubai, from other countries. How about the license? Let's say I get a consultation service from Dubai. Okay, do I have to get license, Dubai license, to be able to sign out this kids or not? What are the thoughts about mitigation of this or decreasing the loads and the paperwork? And because you would get consulted one day for sure, you get consultation from all over the world, cases coming to you from South Korea, cases coming from Middle East, and you have to sign out these cases. How about the malpractice insurance? I mean, you are affiliated with a certain hospital, right? And you are only eligible to or licensed to practice in this particular state. You know, I've practiced in multiple states in the US, and I know each state, they have their strict requirement. You have to go through a lot of procedures to get licensed. And I do hold a couple of licenses in the US. How about if I sign out international cases?
So based on the knowledge I have in the US, which is from my pathologist friends, because it's not like I'm an expert. I ask them questions and they give me answers on what they deal with. In the US, it depends on, are you getting paid for it or not getting paid? That's the first question. So if you're not getting paid for the slide or to do the read or do the case, licensing is not an issue from what they've told me. And then if you are getting paid, then you not only need to get licensed in that state, but you also have to have insurance in that state. And the insurance, they've told me, is more complicated than the license, because insurance is like, what is digital pathology? What do you mean? Internationally, outside of Europe and the US, I don't know of any rules. I mean, my suggestion would be, if there's a pathology advocacy group, let's say in the region, and you get a working group to work on that issue by states or by region. Because that's what they did at College of American Pathology. They put together this digital pathology imaging group, and they have one group on licensing, one group on reimbursement, one group on blah, blah, blah. And they're focused on it. And so when the College of American Pathology goes to the regulators, you know, that means something. So I don't know the answer to this, but is there something similar in other regions of the world?
So we are working on this.
Like in UK, got the Royal College, right? And Australia got the Royal College, but you know, what's similar to the group? The other thing I would tell you too is, from a regulatory standpoint and a business standpoint, you have to really get mentors and sponsors that want to be involved. And like, for example, on this pediatric pathology company I started with a fellow pathologist friend a couple of months ago, we have a passion, right? His child died from cancer, my child died from suicide. So just, we have a passion. So what I tell people in new areas, find people that have a passion. Like, find somebody where they lost their wife or mother to breast cancer or prostate. I mean, you find those people And and I'm talking now from the regulatory side, the government side, the politics side. And you could usually find out who those are, right? Because you know the people from Dubai that fly to Tata to get, you know who they are, so they're probably important in the region. That would be my advice. And anything I could do to support it, I got a lot of friends. Like I know when Neil has come out to Dubai and talked to the healthcare system a couple of times, or the Ministry of Health, sorry, SIHA, I think. And so there's other experts that come out to have these kind of discussions. That isn't, you know, coming from a business person doesn't usually make sense. It's more of it comes from a doctor. I really appreciate the opportunity. I'm passionate about digital pathology, as you can tell. Anything else?
One last. I think this question about license, Dr. Mewar asked you. In future, I think we can have a global digital pathology license.
That'd be cool, yeah.
So you appear for an exam, and if you pass that exam, you can receive slide from anywhere in the world and give your license.
Like a driver's license.
And get paid for it.
I hope so, actually, because that would save a lot of money paying for different licenses, like I paid thousands and thousands of dollars to that
Very
state
costly.
license.
And it takes a lot of time to manage those licenses, right?
Patient for everything.
I mean, start this initiative of
Sure,
global digital pathology license.
Sure.
something like that.
Get paid in your own currency. So not much time. Crazy about, sir, one good news is, my passion was always there. On 5th of December, last year I was detected with abdominal cancer, abdominal tumor, I'm sorry.
sure.
That happened to be transverse colon. On 5th of December, the other sonologist said it's pancreatic tumor. So I'm waiting on for Christmas to get over to know whether it's malignant or benign. That doesn't matter. Before I go up, my passion doesn't allow me to keep quiet, and my not a question, but a note is to all the pathologists is how about making the stains obsolete? Use the technique. Just now I got a crazy idea. Combine the sound, the ultrasound and the chroma, like the rays we are getting from the sun. Somebody must have developed hematoxylin in eosine stage. So that's how we started it. Why can't the future world, we can develop now, and the pathologists won't go out of the job. Let me tell you, we dentists never went out of the job.
That's
doing
right.
scaling and polishing.
That's
We
right.
were trying to save the team, and we have enough business. So if I'm just putting this germ of an idea in the audience's mind, because that may stimulate many young minds, and not an old mind like me. Combine sound technology and the chroma technology to send sound waves to the malignant cells and the non-malignant or the normal cells. Also the colors that they emit, and tomorrow we could totally replace glass sliding, and it would only be digital pathology.
Sure.
Just an idea. It's for your scientific.
Yeah, just two quick things in closing. So there's a company in Lund, Sweden, which is in between Copenhagen and Stockholm, where they've created a replacement for staining using nanoparticles. And it's 5% the cost of staining. Their main issue they're dealing with is upsetting Agilent and Roche. Because we need to, if you look at the annual reports, that's $1 billion business for those companies. Right?
What's the name of that?
Name of the company in Sweden is called Lumito. LUMITO.se. But there are entrepreneurs coming up with these ideas. But my advice to them is try to do it in hiding and under cloak, because the big guys will, I mean, the big guys have the FDA people in their pockets. You know, they take them to dinner, they golf. Yeah. Okay, I appreciate the time. I'd like to hear everybody's excited about digital pathology too. And if you, I'll be here, just come up if you have any other questions.
Thank you very much.
And here's my info.
I want to write this.
Thank you so much.
Mr. Steve. It's a target for you. In 1 minute, I just want to take down a number. I have to get those fun. 1, 2, 5. 5, 8, 6 to see. It's very interesting. Thank you.
Is it done?
Yeah, I'm done. Thank you. And you can sure. 6 to 6.
I think now it's time for our next speaker is Ms. Krishna Himani from United States. I'm passing her the stage to deliver a speech on beyond the conventional approach investigation.
You can use sliders. I'll use this. This is easier. Just look. Can you see me? Do I use this for the next? No, you need to do this. Okay, yeah. And I can.
Like 15 years. It wasn't real.
Good morning, everyone.
We can talk about.
And since we are all from such diverse subsectors of healthcare and pharmaceuticals, I was just having a conversation in the morning with one of my newfound friends, Sidra, that, you know, it feels really different for somebody from a manufacturing pharmaceutical background to be here with the professionals from healthcare and discussing about things that, you know, I might not completely understand, but then finally, you know, it just made sense to me because it looks like we're all, you know, connected with our vision to create healthier and happier lives ultimately, and that's what matters. So yes, now it, you know, instantly made sense to me, and I really am glad to have been invited on this stage in this beautiful futuristic city of Dubai amongst all you amazing professionals from healthcare and pharmaceuticals and allied industry. Before I introduce myself and what we do as a company, I'd like you to imagine something. So imagine this, it's 9 a.m. on a busy Monday morning. And I don't know how many of you use your laptops, computers immediately after you start your work, but just imagine this, you're ready to plow through the mountain of emails in your inboxes, as you always do in the Monday mornings, the first of which appears to be an email from Microsoft. And it says that your password is going to expire. So it says, click here to confirm your account. And you do so without a second thought, because, you know, there's no second thoughts here. It's important. So you do it. You click on the link and you're taken to a page that asks you to input your account details, your password and username. And you do so again. Why? Because without Microsoft, you cannot, you know, imagine your work nowadays. It's like your emails, your calendar schedules, your tasks, and everything is so related to Microsoft. It's all condensed there. So you do that, and naturally, because you don't want to lose access. Now, moments later, if you're working in a company, imagine this, that your company's IT team follow up with a trail of questions and back-to-back calls. Why? Why do you think are they calling you? Because without knowing it, you have unintentionally given hackers access to the company's network, and that might have serious implications, not just technical, but financial implications, because there's a lot of confidential data in there. Now, believe me or not, any human error like the one that I just described, where you did not identify whether the male was a phishing male or a real male, are very common. How common are these errors? Research suggests that regardless of the task that you're conducting or the work that you're doing, humans make an average of 3 to 6 errors per hour, which is somewhere around 50 errors per day, or at least per work shift. What you look here in the slide is also some really important and staggering statistics. The FDA receives more than 100,000 reports of medication errors every year in the US, and there are about 400,000 drug-related injuries that happen in the hospitals every year because of medication errors. Worldwide, human errors are the third leading cause of death, only after heart diseases and cancer. When we talk of the pharmaceutical manufacturing subsector, you see that around 80% of the product quality related issues are somehow related to human errors. And the cost of poor quality in the pharmaceutical manufacturing, it is around 30% of the overall sales and 40% of the operational expenses of the entire firm that is working to manufacture drugs. And possibly this is something which leads to a lot of impact, not just in terms of financial losses, but also regulatory compliances. And irrespective of what we practice and what we believe, we've come to see that human error has been a real problem for the past 2 decades in the pharmaceutical manufacturing. What I'll be talking about today is the conceptual, the foundational basis of changing the approach to investigate and reduce human errors in pharmaceuticals. But you may well apply it to any area because humans are everywhere and so are the errors. So before I move ahead, I'll just give you a background of what we'll be looking at. So these are the points that we'll be highlighting. My name is Karishma Kimani, as you already know. I'm the founder and CEO of Heuristics Pharma Perceptions Private Limited, a company with a vision to create error-free pharmaceutical environments and allied industries. And how do we do that? We compile together the concepts of human factors, human reliability, and cognitive science, along with the modern technological tools like AI, predictive analytics, and machine learning into contemporary quality systems. And we make sure that every element into investigating and reducing human errors is included in this methodology so that we get the real results. And today we are not just going to be talking about these issues that I just said about investigating and reducing human errors, but also we'll be trying to break a few myths around the human tendencies about human errors so that we have a solution to this long-standing problem with a new approach to ensure productive systems, regulatory compliance, as well as patient safety. So if you see here, we know that organizations, especially the pharmaceutical companies, have an uphill battle tackling human errors. And when talking specifically about the drug manufacturing, it has been found that the regulatory observations and warnings have been making it to the top 2 reasons of non-compliances for the past 23 years, and it has remained that way. So there's definitely something that we are doing wrong. And while we talk about the approach that we need to really start doing something about when it comes to human errors, here's the first myth that we are going to break today. When we say a zero error system, Does it sound impossible to you? It does, right? Why? Because we have tried everything. We have made our people competent. We have trained them again and again to, reduce errors, but everything in vain. Somehow we have started believing that, and we call it the belief bias, that we are over relying on our understanding of human errors and how people respond in certain situations. Because what we have seen is, in spite of training people, giving them experience and making them competent, they go on making mistakes. So it's possibly never going to stop. That's what we think. And that's a bias, I say, because when we talk of the zero error system or a zero error environment, it's not just about avoiding or eliminating human errors. It's a philosophy. And it's a funny thing that, more often when we have a first interaction with the clients for human error reduction projects, they say, what, perhaps what you're doing is, really significant and all, but here's the thing, we've tried everything and there is no way you can eliminate human errors. And it isn't surprising, I would say, because we know this fact that humans are made to make errors. We are hardwired to be working like that. And we look at a few human tendencies and how it is really important to make errors, and how we can, actually overcome them by understanding human behavior. But I make this a point to ask people when they tell me this, rather, or, there are a few who are not as suspicious as the ones who believe that, it's just the performance and it's dependent on the person who is doing things. I ask them that, do they remember the movie Meet Joe Black? How many of you have seen that movie? Meet Joe Black. Okay, so it's a movie with Brad Pitt as the major protagonist, and he plays the part of the Death Devil. And he comes to, take the life of a business mogul. It is played by Anthony Hopkins. And the important part here is there's a scene in the movie where Pitt's character reminds all the earthly people that there are just two certain things in life. The first is, of course, death. And the second is taxes. That's what he says. But what Mr. Pitt forgets here is there is a third most certain thing in human life, and that is human errors. So you cannot deny the fact that, irrespective of what you practice, what you believe, we know that it's an undeniable fact and a gospel truth that we are made to make errors, right? So what is zero error system then? As you can see here that, the first question is, can humans stop making errors? No, you cannot avoid humans making errors. You cannot stop them from making errors. But what you can do is prevent what's preventable. And when we talk of the zero error possibility, we say that 100% of human errors are preventable. It's just a change in approach that's required, moving and shifting your focus from person to the system. And we'll see that how it's done. So, a zero error philosophy is a way of thinking and doing things that reinforces the idea that errors are preventable if you take the right actions, and that everyone must strive to achieve it. And once you break that belief bias, somebody just said that, it's all about your mindset. So it is true and in fact possible that you can prevent. We have seen systems where we have tried to create almost zero error environments, and it's possible when you think that it is possible, right? And you need to bring about that shift in mindset. So you're not talking about eliminating errors, you're not talking about avoiding errors, you're talking about preventing them. You're talking about preventing errors that have severe repercussions on your system, your process, your business, and the entire quality of the products that go out, right? So to prove this, we're just going to do an activity here. A few of them have gone for the coffee break, but the remaining ones, I think we are going to do this. Now, here's something that I want you all to do. I'm going to give you 20 seconds and a paragraph here, and I want you to count the number of times letter F for fish appears in this paragraph. And you're not going to tell the number to anyone around you, okay? You're going to keep it to yourself. So as soon as you can see the paragraph, the time starts. And then once I say stop, you need to stop counting. And possibly I'm going to change the slide so you won't be able to see it too. So you have 20 seconds. We're all trained. We're all competent people. I want you to count the number of times the letter F appears here. Your time starts now. Time's up. Okay, so what was the count? How many of you got 10? 10, okay. 11? None. 12? Good. More than 12, anyone? How much? 14. Anyone less than 14, more than 14? Any other number?
7.
Oh, that's one. 7. You could not read the entire thing. Okay, so nobody got anywhere around 14, 15. Okay, I want you to do it again, but now with a slight difference. Yeah, so what's the correct answer?
18.
18. So what possibly happened? Why nobody moved beyond 15? Anyone?
They are same colors.
Okay, that's one.
Stand out.
Couldn't stand out. Okay, don't think about the second slide that I showed you. Why couldn't you read the correct number of Fs? One factor is time, yes, what else? Okay, so here's the thing, your brain often doesn't process the word off for a particular reason that you have read this word so many times in your life that your brain processes it without any effort, overlooking the second letter or sound. It just makes things easier for you. That's how your brain is designed. And that's why if you count, there are 3 Fs with the off, and the others are, obviously you were the closest, but you're not a superhuman though, because you wouldn't get all the 18, right? And this shows us, irrespective of the fact that, these are the letters we've been looking at since kindergarten. And what age are we now? We still did make a mistake here. And the second part is the stress. You change the condition and you see how your performance changes. So if you want humans to make lesser errors or stop making errors, which is, as we said, not possible, what you can do is remove those factors from the system that is allowing people to make mistake. In this case, as Nan said, it was stress of completing it in 20 seconds. Had it been more than 1 minute, of course, you would have, gone through the OFs as well. So there are these conditions that change the way humans respond in a particular situation that's coming from the external stimulus. And there's also the second part, which is, the first is we know that we are going to make errors. The second is we can prevent it by altering the condition, the system. In this case, what was the change in the second part? We highlighted the Fs. So what happened was, it was easier for you to identify, right? So we are not going to focus on the person, train them again, make them more competent to read this, but rather you're focusing on the system, changing it so that anyone can, read it correctly. Now, when we talk of, so this was the first myth of zero error system. Second is the myth that everything that's in your surrounding, when we talk of the system approach in human errors, we say that you have to focus on the system. That's how you can, change the external, internal factors affecting human behavior. But there is a slight difference when it comes to what the regulatories like the FDA, MHRA, and EMA expect and what the industries practice. And the difference is that we as an industry take human errors as the first line of response. So whatever happens, whatever goes wrong, the first thing that comes to your mind and the first thing that you say is, oh, okay, so it was a careless mistake. it was probably an attention lapse. Okay, so if this is the person approach where you focus on the person, what went wrong by him. The regulatory perspective, if you see the EMA and the US FDA, the 21 CFR, it has a guideline which says that when you want to actually get to the root cause of issues, especially the ones that are related with human errors, you need to first rule out system, processes, and procedure related issues. Only after that you can move to human errors, because possibly what is apparently looking as human error is something deep within your system. So this gap in understanding of the guidances by the regulatories to the industry is the mismatch. That's the mismatch which is leading to human errors and repeated observations since the past 2 decades. And also one of the reasons why they think the person approach is the easiest is because there are CAPAs, the corrective and preventive actions as we call them. The easiest thing that you can do when you have an error, especially in the manufacturing firms, is you want to get everything done really quick. You're profit oriented, you are all related to production and stuff. It's less about, although we keep on talking about quality, that is the essence of our CGMP norms, we often fail to keep that as a priority. What we focus on is deliverance. So when that happens, you find the easiest way out. And what is the easiest way out when there are errors, there are failures? To blame a person, train them. That's all, your work is sorted. So the best CAPAs taken in such cases of person approach is you retrain them, you discipline them, counsel them, or sometimes even get rid of them if they're repeatedly making errors. But there's something wrong with this approach or the picture here, because when you say that the performance of your system is directly related to the performance of your people, you have a people-dependent system. And why is people-dependent system not the right thing here? Because we know that people are flawed. And when you believe that, you're going to have systems with zero possibility of zero errors. So you don't want that. And that's why the dependency of people performance from a system needs to go down if you really want zero error systems. We're also going to look at one more example here to prove this. And I'm going to show you this video really quick. Let the ad pass by. Is the volume up? Just check. Why? Because you cannot correct that. Can you change gravity? No. But what you can do is, what can you do to prevent the glass falling down? You can change the ergonomics of the place. You can use a steel glass instead of a glass, right? So if you change the system, human errors can be prevented. Yeah, I'll do it. Thank you. So this is exactly what we are talking about when we talk of system approach. It's human factors engineering. It's the science of designing systems by considering the human limitations and capabilities both. So you design considering the way our brains have been designed, so that when you implement those systems, the possibilities of human errors drastically go down, and in some cases it even vanishes. A lot of industries like the nuclear industry, the oil and gas, maritime, space industry, medical device industry, which is a sibling industry, you can say, aviation industries, have been using the human reliability concept, the human factors concept of designing as per the limitations of the human brain, and they have been successfully doing it since ages. And with our company, we are also trying to inculcate the philosophy behind human factors engineering, human reliability and cognitive science, to bring all those into the pharma manufacturing and allied industries, so that we can effectively reduce human errors and change the graphs and statistics when it comes to the FDA observations. So the next myth is about the system approach. So the people who say that, and it's mostly through theories and academics that we hear, that when you're opting for a system approach in human errors, it's just the system approach. You just focus on things around people, forget about the person. So that's a myth, because human error reduction and control has a little to do with managing acquired behaviors too. So if you really want to reduce human errors and control them, you might, in certain cases, find it difficult to implement human factors principles. And that's exactly where you use the cognitive science, behavioral science, understand how people make mistakes, why it made sense to them at that moment, and then work on managing the acquired behaviors. So the myth that you exclusively have to go for either system approach or person approach is wrong, because you have to optimize it and use it as per the situation. Most of the time, studies have found, especially with the cases that we have run in the past few years, we've seen that 80% of the times, human factors engineering does work, and a few remnant cases, that's exactly where you apply the cognitive science and behavioral science into reducing human errors. There's a concept called performance influencing factors or performance influencers, which is really important when you want to understand human factors and reduce human errors. Now, performance influencers are all those factors around a person performing a task that may impact his performance during the process. So it could be something related to the task, like time pressure, as you got to know through the test that we did, that stress, time, pressure does create a lot of internal chaos, and that's where you're not able to focus on things. And it might sound trivial, but it is true. And most of the times you find that where root cause is not identifiable in human errors, it has a lot to do with that internal chaos. So the work environment, workarounds, what kind of personality conflicts you have, whether there are distractions and interruptions in your surrounding, individual capabilities like a person's attitude to work, cognitive abilities, some tasks that may be new. Okay, all these individual capabilities affect the outcome and human errors. Human nature, this is something which is in that 20% segment, and it's really difficult to handle these, but there are workarounds. So habit patterns, complacency, assumptions, these are the things that come under human nature, and these are all various performance influencers that are present in your system. These performance inferences that I just showed you can be categorized into these three parts, basically. Some have to do with the organization, so they are known as organizational factors like management, resource availability. Then there are task factors that are specific to a particular process, and then there are individual factors. These three are directly, indirectly, in a way, categorized as internal and external factors. So those that are related to the individual person, like psychological profile, situational awareness, brain functions, motor abilities, cognitive abilities, these are all internal factors. And anything other than the internal factors in the vicinity of the person, exterior to his body and mind, they all come under external factors like the environment, procedures, processes, operating systems, your equipments, and both these together result in optimum human performance. We're going to skip this. Okay, there's another factor which really impacts the way you control errors in your system, and it has been seen.
Excuse me. Way we can running out.
Yes, I skipped 2 slides already. We're just closing now. So the error management culture has to do with how you respond to errors. So it's not just working on or investigating errors. The mindset also has to change in a way where you create a culture in your workspaces where you know how to handle the errors that happen, how you respond to them, what actions you take, whether you blame others, are people comfortable to talk about them? Because once they start speaking about them, that's exactly where you start solving them. So the solution-oriented approach is really important here, and the leadership plays a vital role here. So how you communicate about errors, how you share error knowledge, how you learn from them, all that comprises the error management culture, which is equally important to investigating human errors. And now a few myths that we've broken. Now you see the bigger picture and you connect the dots when you really want to switch to that mindset of zero error system and the possibility of error reduction in the pharmaceutical arena. You have to include these contemporary concepts of human factors, human reliability, performance influencing factors, and obviously culture enhancement, the system approach. Once you have all these, it just becomes easier for you to, think about a zero error state. So I'm not going to go through each one of these. This is exactly what we went through in the slides. So this is an outline of a product, SECA, which we have created using AI, predictive analytics, and machine learning. It has 5 modules using all the concepts that I just described, and it is curated as a 5-integrant solution to human error-related problems in pharmaceuticals as well as healthcare. I'm going to play this for a minute. There's no sound. So possibly you can have a look at it on my profile. I'd be glad to connect on LinkedIn. This is the QR code that I have. If you're interested in this area of work, this is what I do exclusively. So you can always reach out, and yeah, that's all. Thank you for having me, and I hope to connect with each one of you personally during networking. Thank you.
Thank you very much. Thank you. Yes. Thank you. And now we are going to move to our next speaker, Dr. Iskander Shawbi from Royal Blackburn University Hospital. I'm passing on the stage.
He was there.
So Dr. Shadi will give us a talk about implementing digital pathology workflow in a leading private lab in the kingdom. Thank you. Yeah, nice.
Thank you, everyone. So I'm Iskander Chowdhury. I work at the Royal Blackburn Hospital. You'll have to mind, I've got a cold at the moment, so I brought it all the way from the UK. Hopefully I won't be sharing it with you. I've actually put two presentations into one. So I'm going to run through two areas. I will talk about the overall landscape in terms of digital pathology in the UK, and then I'll also talk about specifically some of the work we're doing with private labs and also NHS labs. My only conflict of interest would be that the software that we implement in the private labs is a software that I built. So it's a licensed product that I distribute in the UK and also in other countries. So we'll tackle a number of sections, and I'm going to run through this part, and then I'll run through the example of the private lab. So you're all hopefully very familiar with the digital pathology workflow. Part of it's conventional, so electronic request, booking in, macro grossing, and then the area of interest is the digital scanning and that generating the final report. Now, there's quite a lot of data out there in terms of digital pathology, and I'm going to make some reference to that. And essentially, we come under the EMEA, and that data is collated for that part of the world, and I'll go through that in a bit more detail. The landscape in the UK is rapidly changing. The amount of investment into digital pathology is vast, and I'll be able to give you some examples of that in my presentation. In the UK, there's two main frameworks, the Digital Pathology Framework from HTE, and then the Gateshead Technology Framework. And what that means is that certain suppliers are listed under the framework, and then tenders are then put out, and they can then bid for those tenders. The sources of funding in the UK are partly government, so Innovate UK has put vast amounts of money into this, and I'll give you an example. And then we can get digital technology funding, Cancer Alliance funding, and funding from individual hospitals. So this is dating back to 2018, this government website, you can see that they put in 50 million into 3 centres in the UK, one in the Path Lake project in Coventry, the second one in Leeds for 100% digitisation of their slides, and the third one was to Oxford and London. The PathLake project, you can go onto the PathLake website, and essentially you can see there, it states that here PathLake is one of a network of 5 new centers of excellence in digital pathology and medical imaging. And what they're doing is they're just digitizing images from across the country and then creating a massive library of cases that they can then do AI work on and then guide others around diagnosis. This is the Oxford group, and again, they were awarded 10 million in funding. And as a consequence, you've got kind of very familiar organizations that are now moving into this sector. So if I just show you here, so this graph here, if you look at the purple areas, you'll be able to see that within the European group, the European, it says Middle East and Africa, you can see which companies have the share of that particular business in those regions and their customer base and their products. So 3D histech scanners, especially the P1000, has a strong influence in the UK. If you compare that with Hamamatsu, their Zumo scanners, you'll see that they've also got a sizable part of the market, and then the Hurren Digital has a lot smaller influence in the UK. People are very familiar with Leica and its influence in the UK, and there's whole areas. For instance, Wales is the Leica scanners are in Wales and other parts of the UK. Less familiar with is Microscan, and also Roche has some influence, but more within Northern Ireland. Then less familiar scanner companies have a smaller proportion of influence in the UK. Then there's a whole list of well-known companies like Philips. Their scanners are in central London, they're in Liverpool, they're in the Merseyside region, etc. And they've got, you can see their revenue for 2020 and 2021. They've made a fairly substantial footprint in the UK. Well-known PACS company, Sectra, is making very significant kind of influence in the UK. So the Peninsula region, which is in the south of the UK, has been awarded to Sectra. The Black Country has been awarded to Sectra, and they're using their PACS technology to be able to deliver digital pathology. A presentation wouldn't go ahead without talking about AI, and I4, based in Helsinki, has a strong footprint in AI with new clinical suites coming out every month. Then you've got, again, others in the market, Carista, Inspirata, which joined Fujifilm, and they were awarded Nottingham and Derby, and also Bath and the South. They were awarded digital pathology footprint. IBEX has gained a lot of interest, mainly around prostate algorithms and using those in the diagnosis of prostate cancer. And then Indica have started moving into the UK, so real big footprint within research, but in the UK, they have now got a digital solution, again, for the west of the country. And then here's some other names that you'll be familiar with. And all of those are making a influence in the UK. So really, just as a very kind of quick summary, digital transformation in the UK, there's huge amounts of investment by local trusts, by cancer alliances, and also by the government. There's very few hospitals that are 100% digitized, and a lot of that is pathologists not being certain, validation issues, just inertia that's preventing that. One area which we will address in the second part is the integration between a LIMS system and and an IMS system, and the problems that that can face. How do you validate people within digital pathology and people embracing change? So really, where radiology was 10 years ago, pathology is now. So, in 10 years' time, I think you'll start, seeing very little in terms of glass slides, a lot more digitization. You never see a plain film anymore. It's very rare. So that transition across has taken a while. And I think it's happened in radiology because in many respects, it's easier because of the digital size of the image is a lot smaller. And then radiologists are kind of accepting it. Pathology is a more difficult challenge, but certainly is going in the same direction. I think one of the big factors that's limiting that is the cost. So, a typical hospital installation would be, no less than $500,000 per annum on setting up these scanners and the infrastructure that you'd require. So I just want to give you one example. So a project I've been involved in, which is installing a digital pathology platform for a well-recognized lab in the UK. So this is just things that you already know, benefits of digital pathology, second opinion, multiple centers, integration, communication, etc. And this would be the pathway. So you get cases that are booked in, e-ordered, then they're tracked through here. It's the software, PathHub software, and then scanned and then shared. The final reports are shared out. The ability of a software to be able to integrate with other EPR systems, with scanners, being kind of vendor agnostic, is a very strong selling point. So originally, the iSyntax format for Philips was proprietary, and then they realized, well, actually, people want to be able to use different softwares to be able to view it, and they've opened up the code access to allow people to do that. So a good system would have end-to-end specimen tracking and lab alerts, and really that flow really fits in well with a digital pathology system. So you really want to know where the specimen is at any one point, and then you get the digital slide. And these things have been kept completely separate through the LIMS system being kind of one component and the IMS system being another. And the reason for that is the drivers behind IMS have focused on the scan and the scanned images, and that's become their interest. And then the LIMS side of it, because it's quite complex, it's got to interface with EPR systems, people have stayed away from, particularly the digital scan companies. So this would be an example of LIMS integration. You would have laboratories, software in the middle, and then it would interface, so either FHIR or HL7 messaging, Mirth Connect, etc., to get the reports back to the clinicians. So this is the software that was used in this reference lab. And this just gives you some indication of some screenshots of the software. So because it was built by pathologists, the whole workflow is how I, as a pathologist, would want it. So it's not a IT person trying to build a database and then create some forms and connect those together. It's really looking at efficiencies through a software system. So it's based upon dashboards, different user groups. So here, whether it's secretaries, general members, pathologists, clinicians, etc. It uses web technology to visually look pleasing, and also have the function of being able to recognize different users at a glance. And the system then, through HL7, would bring orders into the system, and then these would be connected to laboratory tests. And through the system, you can do a lot more than just look at images. So here you can create projects, and those projects can be service improvement projects. So here an example is macro dissection, another will be lab tracking, and you can invite different groups to those projects. When it comes to digital pathology, so scanning is the easy part, but say if you're working across multi-centers, then how do you workload distribute? So how do you create efficiencies within a network? So the system actually uses an allocator that would allocate based upon preference, so your subspecialty preference, and also based upon your workload arrangements. So say if I just work 3 days a week, it only allocate those cases to me on those days of the week. Or if I'm given a set number of cases or set points, you can, different places work in different ways. The digitized work would follow those algorithms. So there's a lot of work and emphasis on AI, but actually workload efficiencies people are not really focusing on. And actually, if you have good efficiencies, then the pathologist is doing their job rather than somebody else's job, and you can get greater productivity. So a well thought out system can save a pathologist maybe half a day a week, potentially a day a week, where you're kind of running around trying to find cases, get cases allocated to you, etc. So the system would generate these little squares here, talk about variances. So blue one would be kind of cases that have been allocated, how many you should have got, and how many you've actually covered. And certainly in the UK, we base all our activity based on workload points. And we're allocated a number of points or number of cases per week, and then that's kind of knocked back per week. Lab specimen tracking. So now there's Cerebro and Roche have their own system. So what you end up having in a classic NHS environment would be 10 software trying to do different components of a lab workflow. But the PathHub system actually builds it all into one. So it'll track the specimens through the process. It will list the records, and it lists them according to different clinics, the priorities, a flag system, how old the cases are. And it does it according to specialties. And then this is a bit people like to see, so they want to see the digital image. But often what you have to do, you've got to get your LIMS system, then you've got to copy the lab number or the digi number into your IMS system and then look at it. So purely from a governance point of view, there's a risk. You put the wrong number into the wrong case, you're in trouble. And so this is an integrated system. And then these kind of views that I'm sure you're very familiar with. So this would be the digital view, and then that's the actual report itself, and etc. But the point here is that it's all built into the one system. So if you want to then allocate to tumor board or MDT, you do it through the one system. So if I just go forward, it's got a tumor board area, different specialties, dates for those specialties, and the cases automatically get assigned to them. Other elements of it are voice recognition built into the system, support center, videos, etc. And the software allows you to, it's best of breed. So you can have multiple scanners, etc. And you can connect all those scanners into a single PathHub viewer. So workstations are just part of the whole workflow, and this is where the system is at the moment. So it's kind of real interest is where new clinics and labs new labs that set up and don't have a LIMS system, then you can put a digital pathology LIMS system in one fell swoop. You don't have to have the two, or at a regional level where you're trying to distribute work when pressure in one hospital where you've got subspecialty interest within the region, how would you achieve that? You could use the workload function. So this is just a whistle-stop tour of the software. It links in with iForia's clinical suite, so you can do AI. And these are some examples of implementations in the UK. So sites that are actually using the software and some of our partners, etc., as well. So that is the end of my presentation. That's just a breakdown. Just about us. These are the company names, but these are some of the badges of honor that you have to be able to run these systems. So being on the NHS supply chain, part of HTE framework, have the various ISO accreditations and cybersecurity accreditations as well. So that's the end of my presentation. So I'm open to any questions, etc. Thank you.
Thank you very much for your presentation. Any questions? Thank you.
Thank you.
Your certificate.
Oh, sorry.
Thank you very much.
Thank you.
Thank you.
Thank you.
So now moving on to our next speaker, Dr. Abdulhamid Al-Fadl, from King Fahd Specialist Hospital, Dammam. He's going to give us a talk about pediatric acute liver failure in Saudi Arabia, prognostic indicators, outcome, and the role of genetic testing. I'm looking at
Good afternoon, everybody. My name is Dr. Abdulhamid Al-Fadl. I'm a pediatric gastroenterologist. I will thank the organizing committee just to give me this opportunity to present our abstract titled Pediatric Acute Liver Failure in Saudi Arabia, Prognostic Indicator, Outcome, and Role. Here is my team. Our abstract has been recently accepted for publication in the European Journal of Gastroenterology and Diabetes.
You need to speak a little bit louder.
I'll come across. It has been accepted recently for publication in the European Journal of Gastroenterology and Diabetes. Here is my team. We are from 3 departments: pediatric gastroenterology, multi-organ transplant, in addition to pediatric. This is an ethical consideration. This study was approved by the pediatric by the Institutional Research Ethics Board at KFSHD. Just a small introduction regarding the acute liver failure in pediatric. It's a rare clinical syndrome that characterized by severe deterioration of the liver function in addition to rapidly evolving encephalopathy and death. Despite the diagnosis of the adult acute liver failure requiring presence of encephalopathy, but in pediatric it's not necessary because it's difficult sometimes to categorize your pediatric patient in which grade in pediatric hepatic encephalopathy. So diagnosis requiring the adult encephalopathy, but in pediatric is not necessary. So they must adopt the criteria for diagnosing pediatric acute liver failures, the BOLF criteria, which is patient younger than 18 years without any pre-existent liver disease, in addition to biochemical evidence of acute liver failures, such as the increase in the transaminases in addition to bilirubin, with hepatic base encephalopathy. Either if there is a encephalopathy, you need only more than 1.5 INR, but in absence of encephalopathy, 2 or more is sufficient to diagnose your patient with acute liver failure. You may see different classifications or categorization of the etiology of acute liver failure in pediatric, but this is the most adopted classification: infectious, genetic, metabolic, autoimmune liver disease, in addition to vascular, such as vascular occlusion, malignancy, and unknown. Despite all the effort that has been made for the last years to improve the diagnostic effort for those patients. Still, the unknown cause or undetermined cause for those patients ranging from 40 to 50%. They have a poor prognosis if you don't take them seriously and you transfer them to a hepatic transplant center. The outcome will be poor and devastating, though, especially for those patients with advanced hepatic encephalopathy, high INR at admission or at peak, in addition to high Build score and King's College criteria. Only 15 to 25% of them who present with advanced encephalopathy will recover spontaneously or with supportive management, necessitating that for any patient who presented severely should be transferred urgently to any pediatric liver transplant center. So there is no specific treatment till now for acute liver failure, except in few cases, similar, for example, those with autoimmune liver disease. You might start steroid in addition to other modality of treatment. Few metabolic diseases, especially for those neonate or the infantile hepatic liver disease, you may start specific formula for galactosemia or other medication like NTCP for proctosemia. Asthma and infant toxicity, still there is a treatment like NAC. So beside or apart from few available medications, still the transplant is the main definitive treatment for those patients. The idea for our research in KFSHD and DEMA, because there was a lack of data in our area, not only in Saudi Arabia, but also in the Middle East. Based on our quick search in PubMed and other literature, we don't see any evidence of any previous study, except in one study done in Saudi Arabia a few years ago, but it's account only for those early infantile liver failure. Our aim just to identify the etiology, prognostic factor, and the outcome and for our patient, in addition to assess the role of genetic testing. The liver transplant in our center has been established since 2010, and for the first few years we have few cases, but despite the efforts, still our the diagnostic yield is still low. So we have a high rate of undetermined cause or unknown cause. So by 2014 and 15, we start to implement genetic testing in addition to metabolic workup for those patients. We don't have any clear cause for those for their liver disease. So our aim just to assess the role of genetic and the yield of the genetic testing. So the method it's a retrospective study, included all the patients or pediatric patients less than 16, because the cutoff limit for age in our patients is 16. Those who admitted to KFSHD, liver transplant center in Saudi Arabia from January 14 to December 2021. We excluded those before 14 because, as I mentioned, less number of cases, they don't have the full laboratory testing in addition to genetic testing also. The inclusion criteria, we excluded those neonate because the neonate, they have a specific etiology and not only the etiology and behavior. And as I mentioned, 16 is because our aim. For those who met the criteria that I have mentioned before initially. So we excluded the neonate, and we excluded also those patients who admitted to the ICU and who developed acute deterioration or derangement in their liver failure secondary to multisystemic disease, such as those with sepsis or patient with any other diseases that end up in the ICU. So we excluded them. So we extracted the patient characteristic from the files, including their age, the time of the initial symptoms, the day they presented to our hospital, to see how the duration of the symptoms before they arrive to the transplant center, because many previous studies have mentioned that there is a delay in the transferring those patients may affect their prognosis. In addition to that, we take a special attention to their symptoms, especially fever, because they might raise the suspicion of infection in addition to vomiting, jaundice, also other symptoms. Regarding the indicators, we collected laboratory data in addition to we calculated some scores, the important score. Regarding the laboratory data, in addition to their hemoglobin, platelet, INR, liver function, we included lactate, bicarbonate, albumin, and kidney function because it's important for those patients who presented with acute kidney injury in at early stage of their disease as compared to those who presented with normal kidney function. We calculated their score, King's College score. We know that PILD and MELD score, it's used for a chronic liver disease, but it's our aim just to see which indicator can help us to categorize our patients. So we included this score and we calculated their score at admission, but we don't calculate it at the progression of the disease. Etiology, we categorize our etiology as I've mentioned to you, and I will mention to you in the later slide what investigation we had done before we run the analysis. So just to see, we included 46 patients. All of them have met the criteria of pediatric acute liver failure. We can see, let's start with the age. Those patients who just I have mentioned that we grouped our patient two groups. The first group is recovery, those who recovered either by specific treatment such as NAC or spontaneous recovery with the supportive treatment, like in the ICU treatment, and those who underwent either transplant or death. So two groups. You can see that the age is numerically different between the two groups, but was not significant. But when you subgroup the age and the talk those who less than 1 year, you will see a difference. For those patients less than 1 year have high mortality rate as compared to younger, older age group. Those who presented with low glucose score, again, they have a poor prognosis in addition to those who presented with high grade of hepatic encephalopathy as compared to those patients who presented with low grade or non-hepatic encephalopathy. This is the whole the characteristic criteria we had calculated before we run the analysis. As I mentioned, if you can see also the duration of illness was there was a difference between those patients, but was numerically, but in the p-value was not significant. Those patients who ended up by death or transplant, they have almost 11 days of symptoms before they appear to our center as compared to 7 days only. There was no significant difference between the etiology. Symptoms, again, there is not any difference between all symptoms for the two groups, and renal impairment also, there was no any significant difference. What we have got from regarding the etiology, despite all the effort we had made to lessen or to decrease the number of undetermined cause, still we were able just only to define the etiology only for almost 54%, while 46% still the undetermined cause. Apart from the undetermined cause, the genetic and metabolic, in addition to drug-induced liver failure, was the main cause of acute liver failure in our community, or now at least in our center. Other causes and infections account only for 4%, while the Wilson disease and autoimmune hepatitis account for 7% and 4% respectively. These are just the table just to show you the details of the etiology we have done. Despite the all effort and family education regarding the dangers of acetaminophen, still acetaminophen, the paracetamol, one of the major drugs that can cause death, and you have lost one patient who presented late, despite all the effort that had made before he died. Other medications, because we are a transplant center, because we are an oncology and transplant center, we have two patients who had the chemotherapy-induced liver disease after we rolling all the other etiology and going with the temporal correlation between the drug and the clinical symptoms, we diagnosed them as drug-induced liver failure. And even after the patient recovered, when the oncologist they reintroduced the medication again, he developed again a liver failure. Those are the secondary causes, like secondary HLH, fever associated with herbal medication, autoimmune account for 2 patients, Wilson 3 patients, undetermined. There was no significant differences between them. The most interesting thing the thing that we're looking for to see what's our yield for those patients. Before we thought that the main cause of the acute liver failure in pediatric is the metabolic, but nowadays there is a new genetic diseases that lead to acute not only but acute recurrent liver failure. We had determined, apart from the the mitochondrial diseases that's well known for all the hepatologists and gastroenterologists that can cause acute liver failure, like MPV17 and DJOG related mitochondrial depletion syndrome. There is 3 or 4 syndromes newly coming in the literature that can cause acute recurrent liver failure, starting from the homozygous mutation in SCYL1. 2 patients have the similar presentation. They presented with a recurrent attack of acute liver failure with the ataxia symptoms and short stature and neurodegenerative symptoms. Fortunately, both of them had recovered, but the second patient still, he presented to our hospital after we diagnosed him from the first time. He presented for the second and the third time. Interestingly, again, Wolcotralis-son, the patient with the neonatal diabetes, they haven't, again, they present with a recurrent attack of acute liver failure with the mutation in EIF2AOK3. And finally, the patient with infantile hepatic news syndrome has been discovered that have a gene mutation which named as an neuroblastoma amplifier sequence gene. They have a problem in their trafficking system intracellular in the reticular endoplasmic reticulum. Infectious, again, still hepatitis A virus and EB virus associated infection are the cause. These are the indicators that we had collected before we run our analysis. We have taken the hemoglobin at admission and peak, platelet, ALT, AST, bilirubin, INR, ammonia, and other laboratory data, in addition to the two scores, Gold and King's College criteria, and we have found that there is significant differences for the peak albumin, peak platelet. Peak for platelet and albumin mean the lowest, while INR, bilirubin, ammonia mean the highest. There was significant differences between the 2 groups. So to see if these differences are significant and can be taken in consideration as a poor prognostic, we run the Cox analysis, univariant Cox analysis regression to determine which one is the significantly related to death, or at least for poor prognosis. Starting from the INR, we have found that not only the INR at admission or at peak, also how sharp is the rise in the INR. It's significantly related to death. We just measure the median ratio for the INR between the highest one with the peak and the admission, and we compare the rise between the 2 groups, and it was significant. So not only the how high the INR at admission, just only while you are monitoring your patient after admission, how high the increase or an increment in the figure of the INR is also differ from the 2 groups. Again, ammonia at admission was not significant, but ammonia at peak was significant. But again, also the difference, how high the ammonia will increase, it's again related, seen higher for those who ended up with death and transplant as compared to those patients who had recovered. Again, also in the total bilirubin, these are the parameter or indicator we have calculated just to see the differences between the 2 groups. Again, for those patients who presented with good, glycoma score at admission as compared to those patients who presented with lower glycoma score and day of hospitalization, and we compared also those who presented with non-encephalopathy to those who presented with either grade 1 or grade 4 encephalopathy, and all the differences were significant also. Just to control the confounder, we run the multi-variant analysis Cox regression just to see which the most indicator that's correlated to poor prognosis or death. So the 3 indicators were the age, glycoma score, and INR. The higher the age, as you can see, the higher the age, the more favor for survival. While the vice versa, the lower the age, the younger the age of the patient is more favor for mortality. Again, the higher the glycoma score, the favor of survival, as and vice versa, the lower the glycoma score, favor of mortality and transplant group. And finally, also the INR is the opposite. The higher the INR, the higher the INR, more favor for mortality and transplant. So this is the most three indicator for death and for a transplant for our patient group. As I've mentioned initially, that King's College score is a well-known score used for acute liver failure, but we implemented the Bell and Melda score for our patient, and we run the ROC curve just to determine which is the figure of the Bell score that can have a highest specificity and sensitivity. And we have found the figure 20 for those patients who presented with a 20 or less have a high favorable outcome as compared to those patients who presented higher than 20. Here's the outcome. Despite all the efforts, we had the death rate of 34%. Recovery, all in all, for those patients to recover either spontaneously or with a specific treatment and transplant, almost 60%, which is again going with the previous what has mentioned in the previous literature. And it also indicates the high mortality disease. Yani, despite all efforts and all the investigation and management that has been done for those patients, almost 30% will die, and almost, let's say, 10% they will need transplant. That almost 40% of them they end up of death if they not transferred to a transplant center. Coming to the discussion, so just a few minutes. So spontaneous recovery before the era of liver transplant for transplant was only 28%, while after improvement of intensive care unit management and liver transplant has increased their survival rate to reach up to 50 and 60. The recent Pulse data showed that 65% of spontaneous recovery for those patients in 2006, and there is a recent United States Pediatric Health Information reported increase in spontaneous survival to up to 73% with extensive management and addition to liver transplant. So I have mentioned before, we need to accurate prognostic indicator that we can differentiate which patient will recover spontaneously, so he will not unnecessarily go for liver transplant, or unnecessarily will keep following him and will not offer for him the liver transplant. So early listing for liver transplant to improve the outcome and optimize the use of organ resources. So the success of reducing the number of unknown causes of liver failure has remained challenging for not only for one center, all over the world and all over the kingdom, let's say in Saudi Arabia, because of many reasons. Difficulty in reaching the diagnosis, rapidly fatal cause. Some of the patient will end up within 7 days, or they will have rapid deteriorating disease that you cannot do all the required investigation, and also some investigation you cannot do it in your center. So rapidly fatal deteriorating disease, difficulty in reaching diagnosis, and limited time and to complete all diagnostic tests, and lack for diagnostic resources. So in the past few years, there's many reports have been published showing the evidence that there is a new the role of NGS, which is the next generation sequences, to identify those patients with the acute liver failure who had they call it monogenic genetic diseases. So recently, many monogenic diseases linked to acute and recurrent liver failure. Example for this gene, as I mentioned before, and there is also another gene which we don't find in our patient. So genetic testing in our patient, and out of 46 patients, we had 31 patients, which almost 67.3 in order to find the final diagnosis. 7 patients of them, which is mean almost quarter of them had the homozygous variant or varying pathogenicity, as I've mentioned before. So it's mean that these findings indicate that the genetic study are helpful in diagnostic some cases of pediatric acute liver failure. Not only for diagnosis for us, also for the family, which lead to better understanding of the inheritance pattern if they are planning for the next pregnancy, in addition to can explain the risk, determine the carrier status, and review the available testing options for the family. So it subsequently this approach has reduced the parental anxiety and levels as shown also in the previous study. So the limitation of our report or our study, it's small sample size for this only 46 patients. We know it's a rare disease, but for 46 patients did not allow us to do more comparison, especially for those patients with the X diagnosis to other patients. So patient did not undergo the same diagnostic workup. In addition to generalization of our study, results need further research. So just these two tables will show you the how frequency we did each test. For example, hepatitis A has been done in all majority of the patients except 3, while hepatitis E viruses had been done only on 42 patients. These are the other tests like Wilson screening. You know that Wilson will not screen all patients for Wilson, especially those less than 1 or less than 3 years. Autoimmune hepatitis in addition to metabolic screen and genetic testing. So what's the future perspectives? Still, there is a new discussion regarding those patients with acute liver failure or undetermined cause, that there is an area showing that those patients may have an pathophysiological basis of role of abnormal immune dysregulation or hyperfunction for them, like hyperinflammatory immune response, viral infection that we are not able to explore in addition to environmental exposure. There is a new, there's an ongoing multi-center trial treatment of immune-mediated pathophysiology, randomized controlled trial for immunosuppressive therapy for those children with acute liver failure that underway, that is underway, and we are waiting for the result. They are treating them with the immunosuppressive medication just to see the outcome. So better understanding for pediatric acute liver failure in our region, particularly the etiology and prognostic indicator. Conclusion. So this single retrospective study of 46 patients with the pediatric acute liver failure confirmed that this disease associated with high mortality rate in our region. Its most common etiology in our area is different from West Asia and more similar to the West, showing that undetermined metabolic and drug are the most common cause, while the infectious causes are less. Genetic testing improve our diagnostic rate and assist the clinician to make therapeutic decision in addition to support the affected families. The main prognostic indicator are 3: younger the age, the low Kurosaki score, the high INR at presentation, the most 3 poor prognostic indicator for our patient. Finally, increasing the risk of mortality in this patient admitted with advanced encephalopathy may raise the awareness for all the hepatology across the kingdom that those patients, when present in advanced encephalopathy, or once they show the evidence of encephalopathy, they should be transferred to a transplant center for better allocation for liver transplant, including PILD and MILD score, and the comprehensive evaluation for both may help to predict the outcome and guide us for liver transplant decision. Thank you.
Thank you very much for this nice presentation. Excellent research work.
Questions? I don't have time, just like 1 minute.
Yes, yeah, one main question.
So, I'm a GI dermatologist, and I have extensive exposure to pediatric cases in sick and toddler. So just my question, what is the role? I didn't see any role of histology and liver cancer diagnosis and all this country.
Yeah, there is, but we have what we did. We just had those those who are acute liver failure with histopathology. We excluded them from the paper because the paper was only full of the information, so it was difficult to include. So they will be included in another paper. So out of the 46 patients, almost 20 patients underwent the histopathology evaluation. It helped us a little, not that much, because majority of liver biopsies were the extensive necrosis and was not helpful. But for 3 or 4, at least for those 2 patients with the autoimmune hepatitis, it gave us the diagnosis. 1 patient with Wilson's, we did the Wilson amount, which came 1,700 microgram per gram. The 3rd patient, I think 2 autoimmune, 1 Wilson, and the 3rd patient with a drug just with herbal. They have mentioned that there is a neutrophil and other stuff that say better of support of the diagnosis. but not all patients, only 20 they underwent, because you know that they will present with high INR, difficult, it will be sick on uninterrupted, so difficult to go for liver biopsies, and the yield it will be sometimes difficult. So I think it will be another paper just to publish the histopathological feature for them. But we have the data also, and we have also the MRI data for the hepatic encephalopathy and EEG, but it will be just difficult to put all these information in a single paper.
Thank you very much. Thank you very much. So now our last speaker before lunchtime, I know everyone is getting exhausted right now. So is Dr. Olavi Buyan from India. She is gonna give us a talk about inflammatory bowel disease and intestinal tuberculosis pathology.
This one. And this one.
A very good afternoon. Namaste from India. I'm really honored to share this platform with my work experience. Well, that's about inflammatory bowel disease, which includes ulcerative colitis and Crohn's disease, and intestinal tuberculosis. This is a pathologist's tale of gastrointestinal triplets. Well, IBD, it is a tale of two diseases, Crohn's and ulcerative colitis. Inflammatory bowel disease is a chronic condition resulting from complex interactions between intestinal microbiota and host immunity in genetically predisposed individuals that leads to inappropriate mucosal immune activation. So the salient features between Crohn's and ulcerative colitis. Well, Crohn's is basically involves the ileum and the colon as well, whereas ulcerative colitis starts from the colon in a carpet-like fashion. There are skin lesions in Crohn's disease, whereas there will be a continuous involvement of the intestine in ulcerative colitis. And there are certain salient features like transmural inflammation in Crohn's, whereas there will be mucosal inflammation, mostly in ulcerative colitis. To start with, the pathologist can see various lesions which begins as athos ulcers or which proceed on to form linear ulcers. These are the linear ulcers and there will be intervening areas. So there will be edema of the intervening normal mucosa, which gives a cobblestone appearance, where diseased part appears depressed and normal mucosa is raised. So what we get is a cobblestone appearance. The fat, mesenteric fat, tries to protect the ulcerated areas, so there will be creeping fat. And then this is what we find, that is segmental involvement with transmural spread. So also there another feature, that is fissure. These are the gross pathology of disease. Ulcerative colitis can be the acute form, that there will be marked hyperemia, and there will be in the chronic form, there will be mucosal ulceration with residual foci of elevated and hyperemic mucosa. In later stages, we'll see total mucosal atrophy, and this is a case complicated with toxic megacolon. Other features include pseudopolyps and mucosal ridges. So this is the gross pathology of ulcerative colitis. So what we are dealing with is that Crohn's and ulcerative mucosal, because we get biopsies, so we have to look for those features in biopsies. Ulcerative colitis mucosal and Crohn's disease transmural. Okay, what do we see in biopsy? The chronic features of chronicity, which includes basal plasmacytosis, crypt branching, architectural disturbances. We see surface irregularity, we see palate cell metaplasia, and along with those features of chronicity, we also see features of activity. Activity means neutrophils. Where are the neutrophils? Either in the crypts forming cryptitis or inside the lumen forming abscess. These neutrophils may infiltrate the softest enterocytes or they may cause ulceration. So that side is the normal colonic mucosa where we know the tubular, it's like test tube like gland sitting over the muscularis, and here we find the disturbance in the biopsies. So there is surface villus, there is architectural disturbance, and there is surface villus transformation. There is increase in the lamina propria mononuclear cells. There is basal astrocytosis. These are features of chronicity along with cells. If you find them, well and good, they are a feature mostly in the left side colon, not exactly of significance in the right side colon. So these are features of chronicity. There's a plasmacytosis, crypt branching, and crypt loss, surface irregularity, panels. Then what is activity? Neutrophils. Okay, neutrophils in the crypts, that is called as cryptitis. Inside the lumen, that is the abscess, and if they can infiltrate into the surface epithelium. So these are features of activity. So the next picture shows a chronic active lesion. It is mostly a diffuse process, whereas the right side picture shows a convex. So we'll call it as, if this goes in favor of ulcerative colitis, that goes more in favor of Crohn's disease. So, and the second feature is granulomas. Well, granulomas, where we find the difficulties in granulomas occurring less than 50% of resected specimens and less than 5% of biopsies. And what are these granulomas? They are just collections of epithelioid histiocytes. Well, this is the cryptolytic granuloma. Granulomas can also occur when the crypts rupture, the mucin is poured out, there will be granulomas. But what we want to see is a pericryptal granuloma, which is a feature of Crohn's colitis. This is an architectural disturbance summarizing the microsection in Crohn's disease. These are the architectural disturbance. This is section B, showing the granulomas, not caseating. This C is showing the ulceration, purulent exudate, and T is showing transmural involvement with granulomas on throughout. Well, this is a summarizing case of microscopic picture of ulcerative colitis, where we find yes, architectural disturbance, then cryptitis, then crypt abscess, then there will be mucosal inflammation, not much of transmural inflammation. So salient points to remember here: ulcerative colitis is a diffuse process which is ciliary-sided and mucosal. Crohn's disease is a the skin lesions mostly right-sided, and there is transmural inflammation. Granulomas, feature of Crohn's disease. And when we find it difficult to give a definite opinion, there we have a term indeterminate colitis. And of course, biopsy should have mentioned whether there is dysplasia, low-grade or high. And I always give a score to help the clinician for their future follow-up of cases. That is the GBOS score. Well, and this is a simplified GBOS score, which is just proposed. There is robust histological activity score as well, and it have NATS index. So a lot of things to report about. Then, clinician wants features of histologic remission. What is in any way can a pathologist help? Yes, in maybe absence of intraepithelial neutrophils, erosions, and ulcerations. These can be used as follow-up markers. Diagnostic challenge next is Crohn's versus intestinal tuberculosis. Why? Because tuberculosis of the GIT and Crohn's disease are both chronic granulomatous disorders with similarities in their clinical presentation and pathology. Limitation is that rat tumors, which are the primary differentiating features of TB from Crohn's, are found in only 50 to 80% of intestinal mucosal biopsies of TB patients and 15 to 65% of mucosal biopsies from patients with Crohn's disease. Caseation is a feature found in only 18 to 33% and acidic bacilli are found in as low as 5% of cases. So this is some features which helps a pathologist at the size of a granuloma, which is small, less than 200 microns in cross. Larger the size, more than 400 favors tuberculosis. Caseation of coarse tuberculosis. Confluence granulomas features of tuberculosis. So this is what is called as a confluent granuloma. This is a large granuloma favoring features of tuberculosis, small granuloma, and the penetrant granuloma location of the granuloma is also important, which says cross. Then tuberculosis. This is central caseation necrosis, and these are surrounded by Langerhans histiocytes and epithelial histiocytes. And this is the acid-fast bacilli. So this is the gross picture, which of tubercles on the surface and the transverse ulcers in the intestine. What we have reported is the histomorphological evaluation of a non-neoplastic colonoscopic biopsies in lower GI diarrhea. It's a 2 years experience in our center. What we found is the age distribution, mostly 30 to 50 years, and more females and males almost equally affected. And then we have IBD, yes, almost 23%, mostly non-specific, but the features of IBD. And we also found tuberculosis, granulomatous colitis in 2 cases. And what we concentrated upon is the histomorphological patterns. We studied all these very meticulously, and we compared our study with others, and we find that IBD, yes, it is almost comparable, almost similar to other studies as well. So what we found in our study, ulcers, lymphocytic colitis, collagenous colitis, and eosinophilic colitis, amoebic, this is amoebic colitis, and picture of melanosis coli. So when a gastroenterologist, if the clinician doesn't give me total information, then my answer is that of the young sage where who learns to talk and says, let it be known to the birth giver that my stomach consists of emptiness. And mother says, oh, for heaven's sake, Shakespeare just say, Mom, I'm hungry. Thank you. Yes.
Thank you for this presentation. Any questions?
Yes, please.
So, TB versus Crohn's.
Yes.
I think there are always the features that are mandatory for Crohn's, the chronic colitis is not present in the TB, even though if there was a problem like differentiating granulomas, the main core, which is the chronic colitis, is not in TB versus Crohn's.
Yeah. That will be nice if we have a good chunk of specimen. We are only getting biopsies where most of the things are may or may not be present. If they are present, the architectural distortion, all these features, if they are present, they really help us. But in India, in our setup, we have a lot of tuberculosis as well. And the presentation is very, very difficult to distinguish between Crohn's and TB. And the treatment is also so different. So it's a pathologist has to give a very risky report. We take all the risk to give out because the treatment is again different.
Based on my practice, like if we have granulomas without chronic active colitis, we will never say Crohn's, even if it was.
Okay.
And yeah, so because as you said, the treatment is totally different. And it's happened that a mistake happened and a patient ended up after 2 years in the ICU with the abscesses everywhere because there was a disseminated TB.
Yeah.
And he was treated as Crohn's.
Exactly, same case. So with 17-year-old girl presented with cachexia and all that, they thought in line of IBD and they were saying, give us a report of IBD, give us a report. But finally, in later stage, we found it was tuberculosis everywhere. So that was like, some cases do happen. It's a very diagnostic. That is why I said it's a pathologist triplets we deal with, as it is a triplet we are having. So we have to distinguish all three in a very delicate way.
Thank you so much for the great
Thank you. Yeah.
at the same time, you make comment and To correlate with PCR, rule out TB, it is very fast. All culture, it is standard, but PCR is very fast, and for BC, for tuberculosis, so out. And correlate with
Yeah, that is being done, that we try to take all steps here. It's only intestinal TB because the X-ray may or may not help. Yeah, of course, yeah, PCR, yes, of course. They are doing it, the clinicians, they try all the measures. And pathologists, reporters, like they rely on it. So, well.
Excuse me. Thank you for this important topic. We are also endemic in TB. The most confusing sample really is the idiopsychial area, which is common.
Yes.
Disease, Crohn's and
Yes.
yes. So the difficulty is the limitation of the samples, as you say. I don't know if you have an experience with the PCR result in the endoscopic samples. It can exclude the presence of TB in this endoscopy. Endoscopy can take only superficial layer.
Yes.
Is it? We can depend on this results only.
Right, the biopsy is superficial, so maybe PCR may be able to catch it up, but biopsies, I don't know. Ileocecal region, it's a really diagnostic challenge because that area itself is so busy. And to find those tubercles, small tubercles and micro tuberculosis may also evolve from a microgranuloma to a larger granuloma. So if we get the biopsy early, that may be a microgranuloma, and microgranuloma is a feature of Crohn's disease. So it is very difficult from the ileocecal region. It is really difficult to give away straight diagnosis of TB. Of course, we get caseation. Sometimes we really get caseation, good amount of caseation. Sometimes that is a helpful event. But when we don't, then it is really difficult.
I saw a lot of cases of TB in India. Really, the cases of TB in our countries in the Middle East, a little bit have not so obvious granulomas or causation like that in your country. So it's still difficult to depends only on the type of granuloma to differentiate between Crohn's and TB, as you know. So the granuloma, most cases, as you mentioned, less than 5% of endoscopic biopsy we can see or face such a granuloma. When you see the granuloma, can make the diagnosis a little bit easier. But you depend on blood samples for PCR.
PCR, yeah, blood samples for PCR.
Thank you very much.
Thanks.
Thank you very much. One more question? Okay, so we're going to take the last question for lunch, man.
For your presentation. I'm an oral digital pathologist, and we see frequent cases of Crohn's disease in young patients. They come to the clinic with a couple of stony with oral mucosa and linear ulceration. My question is, have you ever been across a case with Behçet disease and Crohn's disease?
Behçet's disease, not yet.
This patient, she came with severe oral ulceration and she had this couple stone on her tongue. She is being treated for Behçet disease because she had also genital ulcers. So when she came, the tongue looked very like classic cobblestone. But I said, okay, let's treat the ulcers and then we will see if the cobblestone resolved or not. So I started her on topical steroids. All the ulceration was resolved. Okay, it was a linear ulceration, so it's more of a cause than Behçet disease. But anyway, so all the ulceration was resolved and she was left with the cobblestone on the tongue. So I asked her if she had abdominal cramps. She said yes. Frequent diarrhea, she said yes. So for me, this is a classic representation of Crohn's disease. I asked her that I'm going to take a biopsy from the tongue. She refused and she never came back because she was traumatized. She had a biopsy before. She was traumatized. She never showed up. About 2 months. So you've never been across a case with like both.
Yeah, no, we have not come across, and biopsy, as sir has already put it in the morning, a lot of people.
Yeah, we don't want to be in place twice either.
Yeah.
And it's good appointment. Yeah. It's really a really yes, she does. She wouldn't write us. Yes, she does. She has a general ulcers, yes, but I'm not really sure about the word. Yes.
Yeah, more channel, we
We took up the dinner hour, so that.
Thank you very much. Thank you. 850 and also come back.
Thank you.
Thank you, Dubai.
Connectivity on the email. Meeting group. If you want to, yeah, I'll share everything. See, usually we should. Big one. Yeah, that's great. I have made this. So at home, I didn't have to come for it. Initially, biopsy, of course, it can handle it. Yes.
So, hope everybody had lunch. We're gonna start the afternoon session now. I wanted to introduce our first speaker of the afternoon, Dr. Mariam from Morocco.
I just have to click in cutting. Hi, I'm very happy to be with you today. I would start by introducing myself. I'm Dr. Zariouhi Mariam. I'm a resident physician in the Department of Pathology in Morocco, and I will present a case report about an entity rarely described in literature. It's about the differentiated leiomyosarcoma with heterologous osteosarcoma components. In order to discuss this topic, I will adopt the following plan. I will start by presenting the case, the element of the clinical examination, the results of the radiology, and then I will proceed to an analysis of the pathological features. Then I will move to a discussion where I will review the data from literature before concluding. So it's about a 43-year-old woman who came to the consultation for a painful mass of the upper left thigh, progressively enlarging over the past 2 years. Her past medical history includes a pleomorphic leiomyosarcoma of the upper left thigh, surgically treated 3 times. She also had 33 sessions of radiation therapy. The physical examination showed a visible mass with scars and mild inflammatory signs. There was no palpable regional adenopathy, and the remainder of the physical examination was unremarkable. Our patient underwent a CT scan, which showed a lesion centered on the posteromedial muscular compartment of the left thigh, irregular in shape, polylobed contours, heterogeneously enhanced after contrast, containing central hypodense area of necrosis, as well as calcification in their surrounding fats. Thereby, the imaging findings were mostly compatible with the diagnosis of a local recurrence made of a subcutaneous permeation nodule on the posterior compartment of the left thigh. Now we will move to the pathology analysis. Our page, so a surgical excision of the tumor was performed and sent to our department of pathology. The soft tissue specimen has been received fresh and consisted of 5 centimeter thin white mass with cystic area and hemorrhage. Histomorphological features of this nodule revealed a biphasic pattern. The first one is made of typical spindle cells arranged in short fascicles, focally pleomorphic with an ovoid hyperchromatic nucleus and eosinophilic cytoplasm, typical of leiomyosarcoma. Mitosis were frequent, with 16/10 high power file, and there was no necrosis in the whole area. And the other components has a distinctive calcified osteoid laid down by malignant cells. An immunochemistry study was performed, and it shows a positive immunoreactivity for smooth muscle actin in the area of leiomyosarcomatous appearance, whereas the osteosarcomatous component was negative. Desmin and Ashkal-Desmon were negative in both components. A fish study was as well performed to rule out the differentiated liposarcoma with osteosarcomatous component, and it came back negative. There was no amplification of MDM2 gene. There is 1 copy of the gene in each chromosome. So, based on these findings and the patient history, the lesion is favored to represent a leiomyosarcoma grade 3 according to FNCLCC grading system with area of osteosarcoma, so-called differentiated leiomyosarcoma. Leiomyosarcomas represent 10 to 20% of soft tissue sarcomas, most commonly involving the retroperitoneum and commonly the lambs. The terminology, the differentiated leiomyosarcoma, was first introduced by Schmuckler, and it refers to a tumor characterized by an abrupt transition from a classical leiomyosarcoma to a high-grade sarcoma, which does not express muscle markers. Diagnosing sarcomas can be challenging, especially for specific subtypes, and this rare phenomenon of the differentiation can mislead to inappropriate diagnosis and treatments. They are aggressive neoplasm with poor outcome, with 50 to 65% of mortality and 89 metastasis incidence. And there is no exact mechanism proven to be the direct cause of the differentiation. Up to now, 6 cases were described with heterologous osteosarcomatous differentiation. Considering all the cases reported, the average age is 56 years with a male predominance. It is very important to note that all patients had local recurrence despite the treatments. In three patients, the outcome was fatal, and the due to the progression of the disease, while no clinical follow-up was detailed for the remaining 2 patients. And our patient is still alive, and she has also another recurrence. This data confirms that the differentiated leiomyosarcoma of the soft parts is highly aggressive tumor. The main differential diagnosis is differentiated liposarcoma with heterologous smooth muscle and osteochondroblastic differentiation. This was excluded because there was no evidence of a well-differentiated liposarcoma component in both first specimen and the recurrence. And in addition, there was no amplification of MDM2 gene in fish study. The diagnosis of leiomyosarcoma grade 3 of FNC-LCC with heterologous osteosarcomatous components is appropriate for this case because the tumor gained the osteosarcomatous portion during the progression and did not show the two lines of differentiation from the beginning. So the differentiated leiomyosarcoma is a rare disease with poor outcome. Extensive sampling is important in leiomyosarcoma to reveal the different components, and looking of amplification of MDM2 gene by fish is recommended in diagnosis and approach. And thank you for your attention. Here is the final poster.
Did you do any other markers?
We do. Excuse me? Can you repeat, please?
Yes. Do you need other markers, immunomarkers, to rule out other substitution tumor?
Yes.
Because you do, it's in as I mentioned, and listening, of course, it's a positive.
Positive.
It is how.
All other. Yes, all other markers were negative. We have done CD 34, 31. Let me think what we do else. Myogenin. It came back negative. And what else? I am. Yes, nerve markers were negative. Yes, yes. Oh, yes. Nerve markers were negative. 34 and 31, they were negative. And the PS 100 was negative.
Molecular or something scientific?
No, we did just fish. Yes, and it was it came back negative. MDM2, yes, liposarcoma, the differentiated liposarcoma. No.
Any other questions? Well, thank you so much for your presentation. I'm a rookie, I forgot. Guess I should have dressed up.
Thank you.
So for our next presenter, speaker would be Dr. Sumaya from Pakistan.
If you want, you can use you can use pointer to yeah.
Good afternoon, everyone. I am Dr. Sumaya Suhail. I am here. I have come here all the way from Pakistan to represent what is happening in our part of the world. And my topic today is very much in a sense of the topic that was discussed in the morning. It is artificial intelligence in categorization of breast pathologists. Just for a brief introduction, I did my MD in histopathology like about 4 years back, and I'm a diplomat of Royal College of Pathologists. Currently, I'm working as a consultant histopathologist in Pakistan Institute of Medical Sciences, which is one of the tertiary care hospital and one of the leading hospitals in Islamabad, centered in the main capital of Pakistan. The topic of my discussion today is what I am getting out of the discussions which I heard in the morning is a pathologist perspective of developing artificial intelligence and the digital platforms in countries, and what are the hurdles which we face in the developing countries, and as we grow our journey in this field, what are the basic issues that we have to face and we have to overcome in order to adopt it successfully in our settings. The layout of my presentation will begin with a brief introduction. I will go with the study design and the results, conclusion, what are the future avenues and the limitations. To start with, as pathologist or as histopathologist, when we hear these terms, artificial and then intelligent, these are rather paradoxical terms, which have required a very pivotal role in this era of personalized medicine. What I comprehended out of it is artificial intelligence is basically a branch of computer sciences that deals with the task that normally requires human intelligence. It is the simulation of human mind in computer system that is programmed to think like a human and mimic the actions such as learning and problem solving. Digital pathology, on the other hand, is the acquisition, management, sharing, and interpretation of pathology information, including the slides as well as data in the digital platform. It is, in my opinion, essentially a merge of medicine and technology. Digital pathology has acquired this much hype and this much growing power because of the increasing utilization of the digital imaging technologies and the emergence of artificial intelligence in medical imaging. We all are already working with static images, like all the presentations which we make, the teleconsultations we are doing, the MDT we are doing, we are working with static digital images. And whole slide imaging scanner, in some parts of the world, I have seen like it is really good to know that there are many people who are working on it for more than 4 decades. And in our part of the world, whole slide imaging techniques are really novel to introduce. And if they have been introduced, they are in the process of validation. The other very important aspect of developing a digital platform is developing a digital, annotated, authenticated digital library, which Mr. Steve also highlighted in the morning. The main thing is that we need an annotated digital platform that has to be implemented and that has to be authentic enough to be applied to our patients. The rationale of my study was the deployment of artificial intelligence tools in digital pathology, specifically in binary categorization of breast pathology. By binary categorization, I mean the labeling of the biopsy as benign and malignant. We started, as our country is a resource-restricted community, we started with static images. And interestingly, when I explored this option, I came to know that there are many attractive things that we very little know about. Like we all are using AI in our basic use. Huh? Every one of us uses Google Photos. We wake up, the Google asks us, do you know this person? Do you know this? Can you help me find your daughter or your mother? And they can scan the whole gallery of your mobile and they help you find that specific person. This is what implication of AI in digital pathology or in pattern recognition on the slide. This is the rationale which we used in our study, that is the image recognition or the pattern recognition. The image or the pattern recognition, like labeling a case as a malignant or a benign focus, labeling a particular focus as a malignant or benign focus. This is what was the rationale behind our study. Now, what the pathologist's job is here. We can acquire images, we can label it, but we have to tell the computer, we have to tell the algorithms that this focus is benign and this focus is malignant. And this is basically what is the concept of annotation. As pathologists, we very little know what is annotation. Annotation is the process of labeling digital data by a pathologist, by a qualified pathologist, that can be kept for a digital record, and later it can be shared on a digital platform so that it can be utilized at a remote site. The materials and methods which we employed, this is basically a prospective study that was conducted in my department, Pakistan Institute of Medical Sciences, Pathology Department, in collaboration with ComSats University between April 2020 to October 2022. Just for the sake of introduction, PIMS is a tertiary care teaching hospital, university associated, and we are providing diagnosis to about 10,000 and more patients per year, not only histopathology, but also cytopathology, immunohistochemistry, frozen section. And we have very specialized surgical teams that provides influx to our histopathology concepts. That includes oral pathologies. One-stop breast clinic is one of the best that comes in the umbrella of PIMS Hospital. We also cater the neurosurgery team, the cardiac surgery, pediatric surgery, and the burn center units. These are all the random influx of histopathology that we are receiving in our department. ComSats University, on the other hand, that was the AI partner of our study. It is one of the leading universities in Pakistan Institute of Medical Sciences, and it is providing faculties not only limited to engineering, information science and technology and AI, and they are well recognized for their trend-setting approach, allowing interdisciplinary approach and methodologies for learning and research purposes. What we did, we included all the patients undergoing a breast biopsy. Age ranged from 16 to 70 years, and they were done with the biopsy that was done for a clinically detectable lesion. We included all the true-cut biopsies, mastectomies, and excisional biopsies, but we excluded all the autolyzed and non-compromised and non-representative samples from the study. Now, coming to the acquisition of images. Acquisition of images, we labeled the slide with particular foci of benign and malignant foci on the glass slide. The pathologist labeled the benign and malignant foci and asked the resident to take the static picture images of this using an infinity analyzer that has been installed in our department. It is basically a camera that is linked to that digital microscope, and it can be assessed on an internet-based laptop, and then later can be shared by Google Drive with the AI team. These are a few images that we took. This, as we can very well see, that this is a benign focus and a malignant focus. And further, the workflow, the AI team, what they did, they applied Python programming language, deep learning methodology, TensorFlow, NVIDIA, Tesla, that is funded by Higher Education Commission in ComSats University. And they employ deep learning methods for the binary categorization into benign and malignant foci by using the deep learning methodology. The AI team used the image recognition technology. Like I already mentioned, the image recognition or the pattern recognition, they employ deep learning methods, which is a sophisticated form of machine learning. Initially, they pre-processed each of the image by image normalization, H&E segregation, and cropping or resizing the images to 224 into 224 pixels. And then they fed all these data to train the deep learning models. They approached the stochastic gradient descent with an initial learning rate of 1E4 and a momentum of 0.49. A batch size of 32 was used, and binary cross entropy and loss of function were calculated. What our AI team told us that they found that the Inception V3 was the best and the most reliable model when they were assessing the microscopic images, because it helped to maintain the architecture and save many parameters as compared to that of other deep learning models that can be used. They evaluated the data for sensitivity, specificity, accuracy, and used it to evaluate the AI-based system. Out of this, we fed them about 992 histopathology images, benign and malignant. And after employing the AI or deep learning methods, they found out that there was an 87% accuracy, 90% sensitivity, and 80% specificity that was achieved by this model only with the static images. Like the basic static images, the AI algorithm was able to categorize it. Later at a predictive analysis, they could call it the benign focus as benign and malignant as malignant. So what we found that these results are promising even at the very start. They are, in fact, the need of the hour. Why is this the need of the hour? Because as we all know that breast cancer is one of the most prevalent breast cancer in the world, and especially in underdeveloped part of the country where there is a lack of expert consultation. So why I chose breast cancer is keeping in view the international statistics. We all know that out of every 8, the prevalence rate of breast cancer is this frequent. Like every 8 women, 1 will have a breast cancer. And in Pakistan, there has been very important discussions as well as cancer registries and initiatives. And there is a very interesting article saying breast cancer in Pakistan, a looming epidemic. And this is because it is a very underreported and delayed treatment that is offered to the patient only because either the patients are not aware of it, or if they are aware of it, they don't know where to go. So what we employed, we assessed what are the avoidable delays, and I corresponded them with the international data. And there's a very important Cancer Research UK. They say that the avoidable delays can be at the health professional level or hospital level, and then at the patient level. The patient may delay the reporting to the specialized unit, but what we can avoid is this 50 to 60% population, there's 60% delay. How AI can help us is in the timely triage. They can, what the minimum out of this study or out of these parameters we can do is that we can have a histopathology biopsy done at a periphery and a static image being shared with any of the concerned or expert pathologists that can either label it as benign or malignant, and the patient can be triaged to the specialized center so that she can get a timely treatment. Now, an important aspect of this, like we all have been discussing in the morning, AI is it has attracted a lot of attention and it has a lot of potential to grow. And we realize that only working on static images and a small number of patients, this is a small population, and we have to work more in order to consolidate it. So we are ongoing this process, and we have included other parameters from the breast cancer, including the histopathological type, the grading, lymphovascular invasion, and perineural. We annotate the slide, and we are teaching the algorithms so that they can further at least annotate this focus is to be reviewed by the pathologist. An extra plotted plan of this is the incorporation of whole slide scanner images. And just before I came to Pakistan, luckily we have got a collaborative effort at a national level, and we will be adding all these data to the whole slide imaging scanner, and the scanner further can use the annotated data so that it can further qualify that this focus is to be reviewed by the pathologist. A few simultaneous and other potentials that we can employ out of this study is we all as pathologists know what we have to go through serial deepers and deepers to find a single ganglion cell in Hirschsprung disease, where whoever is reporting histopathology, they will know like 40 deeper levels is the standard which we have to examine and find that one single cell that has to be annotated, and that one single cell will prevent the patient from going to colectomy and colostomy throughout his life. Like, it's a very big decision for the patient. So to implement this thing, we extrapolated it to this, like we started with the study that recognizes the ganglion cells, and we are taking multiple static images and incorporating it into the whole slide imaging as well as the static imaging deep learning algorithm, so that at least the computer can assess that single focus and annotate that focus to be reviewed by the pathologist. The idea what we planned was basically from one of the leading digital pathologists, like sir mentioned in the morning, Dr. Anil Pipani, and he presented this concept of working with static images about 10 years back. He developed an app of Pocket Pathologist, and this is a very interesting article in which he says that at that time, static cameras and whole slide imaging scanner, he quotes it was $6,000, and the whole slide imaging scanner is about $300,000 at that time of the study. And they worked with the static images, and they found that it was quite promising, and they could provide a platform for teleconsultation, which we all are already doing it and have been practicing it in some basic form. Now, what AI can do with this? AI, as I mentioned already in the morning, AI worked either supervised or unsupervised. In supervised learning, the basic concept which we have employed is just to classify, just to binary categorize whether it's a tumorous condition or it's not a tumor, or it's a malignant condition or not a malignant condition. The AI algorithms now developed so much that they can classify, they can generate, they can segment, and then they can search. The search aspect is basically what can be employed in finding the malignant focus, because as one of the speakers in the morning, she was saying that human error is always there. Finding a single one focus in the slide that can easily be missed, and that is something that we all read as histopathologists. Developing these digital platforms will definitely be an adjunct in missing small foci in the long. It will help us in decreasing the time what we spend in searching of single blast cell, single ganglion cell, a single malarial parasite. These are the things which we pathologists do every day, and we spend hours and hours on reporting this. So the future implication is working with static images and then incorporating whole slide imaging scanner and then the AI. Just for those who don't know the basic concept behind whole slide imaging scanner, it is basically now considered synonymous with virtual microscopy. We are using Leeds Pathology. We are using all the digital platform that is an educational resource, a research retrieval data. And these are the platforms that are in random use. And truly speaking, what I realized is that COVID made us more familiar with all these digital platforms. The basic concept behind the whole slide imaging is Z-stacking. Z-stacking is basically integrating all the data into small pixels, or generating the data into the small pixels that can later be reviewed at a remote site, so that it can be used for teleconsultation, for reporting, as well as for annotation. Now, as pathologists, the basic concern of switching to digital platform. The foremost, we all work with microscope, and what we want to know is the resolution and the picture quality. And believe me, the digital platforms which I have worked with, they are equivalent to what we use in our microscopes. And each day there are advancements, as we have already seen. And these digital platforms are really helping us in not only diagnostics, but at the very basic level as a research resource, as an educational resource. We all never knew about virtual mocks. We never knew about educational platforms. We never knew about webinars. The webinars, the virtual mocks, the teleconsultation, these all have been attributed to the deployment of artificial intelligence and digital pathology into the pathology networks. Some of the future studies which we are planning in our part of the world, Hersprung and incorporating other parameters of cancer diagnosis are the few which I have already mentioned. Other few are which was conducted in Karachi. It was in for the identification of blast cells, like as hematopathologists, we know identifying blast cells and malarial parasite throughout the slide is always time taking, and it can be easily ignored as well. So these are the important parameters that can be explored for the application of digital pathology into our network. Now coming to the discussion that we all also had some part of it in the morning, the challenges and limitations. The first and the foremost for me is the validation. The second is obviously the cost. And third is how much the pathologist will be comfortable with working with the digital platform. And the last but not the least, like we discussed in the morning, pathologist versus AI pathologist. Is it a competition or is it a collaboration? I'll be taking up all these challenges one by one. Now, the basic concern. As a histopathologist, we all know when we render a diagnosis on the slide, we know that behind every slide, there is a human being. We have to know that that human being is a pivotal part of his family, and we are rendering the diagnosis for that human being. That last slide is basically our patients, which we work daily. Supervised learning of the software is the answer to this, of validating these platforms for our patients. What I mean by supervised learning is that all the digital information that is being incorporated into various parts of the world, it has to be annotated and it has to be validated, not only for the patients in that part of the world, but there should be specific legislation and because we cannot apply all the statistics throughout the globe. We all know that there are molecular heterogeneity throughout the globe. The tumor, which I see in my part of the world, it's very much aggressive as compared to the other part of the world. So we have to validate these data for our local setup. We have to validate them for our setup and then further incorporate them for an international and global level. Luckily, where these things are being practiced, the College of American Pathologists, they issued Digital Pathology Resource Guide in about 2017. And the Royal College of Pathologists, they also have very authentic and validated guidelines. They have a specific protocol for saying to start with, a pathologist should report on the digital platform and then counter confirm it with the glass slide and then further give feedback so that these platforms can be validated. So the successful implementation needs a feedback-based revision of the guidance. Now coming to the second and most important part, which we were discussing in the morning as well, is the cost. The available options which I explored are national collaborations. We also have had some collaborative efforts with International Massachusetts General Hospital, and there are some research funds that are being granted to the institute as well as nationally as well as internationally. This is one of the important paper which I was a part of about 5 years back, and the Holy Family Hospital and Massachusetts General Hospital, they had a collaborative effort of developing whole site imaging based digital pathology network. And we were having teleconsultation with Dr. David Wilbur in Massachusetts General Hospital. And we presented this in our Pakistan Association of Pathologists, and it was one of the award-winning posters in about 2015. And the jury there was Dr. Shafiq Gill from NHS. But the basic part, like the scanner which we were using at that time, it was about 2015, 8 years back. It was a small base scanner, and the specification it had were mainly for teleconsultation, and for the sake, it took a whole day to scan that one single slide. The third very important task that we as pathologists, while incorporating the digital platform, is we need a good digital library. We have to get used to the digital platform. Like we, in our training part, we see the glass slides. We say you can see 1,000 slides, and after that you can report it independently or something like that. The same applies for digital platforms. We have to validate it, we have to authenticate it, and we have to employ it in a definite infrastructure, and above that, after validating it on our local population as well. And there's a very interesting article, if anyone wants, it says not just digital pathology, we need intelligent digital pathology. So it has to be validated, it has to be labeled, and it has to be very well annotated by a qualified pathologist so that we can incorporate it for our local networks. Where these systems are developed, what people are doing is the pathologist-centric pathology AI system. What I mean by pathologist-centric pathology AI system is that cell recognition mechanism, cell classification, and then the pathologist reviews it. And this information is to be integrated into the digital platform. The pathologist verifies it, whether what the computer has said is correct or not. And then this data is incorporated into the digital platform, and this can be validated and further incorporated for future learning and teaching purposes. Now, the most important part, like we discussed in the morning, just on a lighter note, there was an article and it said that pathologist versus AI pathologist. Is it a collaboration or is it a competition? And when I started these research projects in my part of the world, everyone was like, are we going jobless after it? And like we said in the morning. So I was a bit worried too, and I explored the data, and there were many good documentaries as well as very good answers to this. And I like to quote one of the documentaries, and it says, like the famous notion goes, fire is a good servant, but a bad master. We must realize that these digital platforms are already a necessity. We cannot deny it. And COVID has compelled us to adopt it and to incorporate it. And COVID told us that we can do it. We have to incorporate it. It's high time that we train these platforms for our adjuncts, not our replacements. So the answer to this question, is it a collaboration or is it a competition? It is definitely a collaboration. Digital pathology and artificial intelligence are our adjuncts. They are not our replacements. And definitely it's a collaboration, and we have to make sure that it stays a healthy collaboration. Thank you very much. And in the end, I'd like to say the sense of this platform was, like, I realized that man and machine, they both complete each other. So we cannot live without the digital platforms right now.
Well, thank you. Any questions? Go once, go twice. Okay. Well, thank you.
Thank you.
Coming up, next speaker is Dr. Hopefully I'll say this right, Yanni. What? Jody. From the Philippines.
So good afternoon, everyone. I am Dr. Joni Faye-Teves, an internal medicine resident from the National Kidney and Transplant Institute of the Philippines. I'm in behalf of my advisors, Dr. Jade Hamyas, who's a gastroenterologist and hepatologist, and also Pepito de la Peña, who's an endocrinologist, both of whom are highly esteemed specialists in our country. I'm here to present our case report entitled Hypoglycemia as a Manifestation of Neoplastic Disease and its Progression. So I aim to present 2 cases of tumor-induced hypoglycemia, to discuss its pathophysiology, to present case reports or publications of diagnostic principles and management of non-islet cell tumor-induced hypoglycemia. So to start off with our first case, we have TDS, a 32-year-old male who came in with a chief complaint of unresponsiveness. History started 2 weeks prior to admission when the patient presented with profuse sweating. However, there were no other accompanying signs and symptoms. This prompted consult at a local health clinic wherein thyroid function was done, which revealed a normal TSH and a slightly elevated FT4. He was then referred to an endocrinologist and was managed as a case of hyperthyroidism. He was prescribed methimazole 5 milligrams, 1 tablet once daily. However, in the interim, there was no relief of symptoms, and this was now associated with generalized body weakness, easy fatigability, and shortness of breath. Until 3 days prior to admission, there was already slurring of speech and unresponsiveness. This prompted consult at a local hospital, and thereby he was admitted. Initial workup revealed a low capillary blood glucose level of 18, for which he regained consciousness only after 1 vial of administration of D50/50 solution. Other pertinent workup was done and revealed a pertinent here is hypokalemia, and thyroid function was now normal. Furthermore, whole abdominal ultrasound was done, which revealed hepatomegaly with right hepatic lobe mass. And further on, CT scan was done, which revealed a huge hepatic mass with arterial enhancement, measuring about 17 by 11 by 13 centimeters, and this was again enhancing. Tumor markers was done. Pertinent was elevation in the AFP and normal SEA. With these findings, the patients and family prompted second opinion in our institution. On review of systems, it was unremarkable except for easy fatigability. And on past medical history, there was pulmonary tuberculosis for which he completed treatment 6 months in the 1990s, and the rest was unremarkable. Family history was significant for liver cancer and hepatitis B, as well as lung cancer. He was an occasional alcohol beverage drinker and was a 5-pack-year smoker and had history of tattoo administration in the year 2000s. He arrived at our institution awake, alert, but diaphoretic, not in distress, but had saturations at room air. The rest of the systemic PE was unremarkable except for a palpable liver edge, 3 centimeters below the right costal margin, which was extending laterally. However, that was non-tender. He was admitted as a case of hepatocellular carcinoma, hepatitis B related, and type B hypoglycemia, for which I will expound later on. For our second case, we have DPB, a 68-year-old female who came in with a chief complaint of decrease in sensorium. History started 2 months prior when the patient presented with generalized body weakness and decreased appetite, as well as loose stools. In the interim, this was already associated with undocumented weight loss, jaundice, and vague right upper quadrant pain. Because of the persistence of symptoms, the patient consulted with doctor and workup was done. For example, a whole abdominal ultrasound, which revealed hepatomegaly with diffuse parenchymal disease and echogenicity lesions, measuring around 7 by 6 centimeters and 5 centimeters. CT scan was also done, which revealed a multiple heterogeneously enhancing nodules and masses in both liver lobes and in the caudate lobe. Largest is seen as ill-defined conglomerate of masses at segments 4A to 8, which measures around 11.5 by 10.8 centimeters. Biochemical workup was done, which revealed AST predominant transaminitis and a tremendously elevated AFP of more than 10,000. He was then admitted, and then further metastatic surveillance was done, and chest CT scan revealed progression with lung metastasis. And then he was managed as a case of multifocal hepatocellular carcinoma, for which she was started on nipatinib.
Eventually, she was discharged stable and had improving CBGs, but at home, there was recurrence of hypoglycemic episodes, and there was now association of decrease in sensorium, hence she was readmitted. On review of systems, the patient had easy fatigability and dry cough, and past medical history is significant for hypertension, diabetes. Family history was significant for liver cancer and hypertension and diabetes as well. Personal social history was unremarkable, and she arrived at her emergency room drowsy, roused about to name calling. However, she was already gasping. The blood pressure was elevated, and that's typographical error. There's no desaturations, 97%. However, in the systemic P, it's remarkable for icteria, the decreased breath sounds on the left base and rales at the right base. And the abdominal P was remarkable for a palpable liver edge 4 centimeters below the subcostal line. And there was grade 2 bipedal edema. The patient was also jaundiced. A neurological exam revealed a drowsy but disoriented patient, but there were no lateralizing signs. She was admitted to the case of acute respiratory failure, multifactorial, probably from encephalopathy, metabolic from hypoglycemia, hepatic and septic, probably from pneumonia, and multifocal hepatocellular carcinoma, hepatitis B related, and type 2 diabetes, non-insulin requiring poor control. With these take-off cases, we asked these clinical questions. What causes recurrent hypoglycemia in a non-diabetic individual? What is the diagnostic approach to hypoglycemia and non-islet cell tumor hypoglycemia? And what are its treatment options? Hypoglycemia are common. Symptoms of hypoglycemia are common and relatively non-specific, but hypoglycemia itself is relatively uncommon and usually occurs in the setting of the treatment of glucose-lowering agents such as your insulin and sulfonylureas. Neurogenic symptoms include sweating, warmth, anxiety, tremor, nausea, palpitations, tachycardia, and hunger. And for the neuroglycopenic symptoms, we have behavioral or visual changes, cognitive impairment, for example, confusion, difficulty in speaking, lightheadedness, and in lower plasma glucose concentrations, we'll have lethargy, seizure, loss of consciousness, and coma. So its diagnosis requires the fulfillment of the Whipple's triad, namely the hypoglycemic symptoms, the low levels of your plasma glucose, and the relief of symptoms after the administration after plasma glucose level is raised. So maintaining normal glucose concentrations is accomplished by a network of hormones and neuroendocrine mechanisms. So for example, here, as you can see in the figure, insulin plays a dominant role, such that with decrease in glucose, you have a corresponding decrease in insulin level. Second line of defense would be an increase in your glucagon. The third would be an increase in epinephrine acting on the beta-2 adrenergic receptors, which leads to the increase in delivery of your gluconeogenic substrates in the periphery, and also decreased glucose tolerance, leading overall to increase in arterial glucose level. Cortisol does not play a role because it has a slow onset of action and does not have any effect on acutely rising your plasma glucose levels. So let me guide you through this figure. As the arterial plasma glucose concentration falls below the physiologic range, as you can see, that bold line is the glycemic threshold. So it is activated when the plasma glucose level reaches around 80 to 85 milligrams per deciliter. However, if there's recurrent hypoglycemia, this glycemic threshold is further decreased, as you can see in the dashed line. However, this counter-regulatory mechanism becomes extremely dangerous when there is recurrent hypoglycemia, because it might push the patient to hypoglycemic unawareness, and this leads to a vicious cycle of hypoglycemia and impaired mechanisms to counter that. So what is the approach to hypoglycemia? This is a basic overview, and as you can see, the easiest would be the diabetic individuals. You have to just modify your medications. But in patients who are seemingly well and present with the first onset of hypoglycemia, you have to seek clues, for example, of alcohol abuse or drug ingestion. It becomes complicated when it comes to non-diabetic individuals. So we need to seek clues pointing to specific disorders, for example, critical illness of sepsis, congestive heart failure, acute liver failure. And if there's persistence of hypoglycemia and recurrence of hypoglycemia, you already have to consider probably insulin secreting tumors or increased insulin growth activity. So we classify hypoglycemia into two types. There's fasting state, wherein it already occurs around beyond 5 hours after food intake. It happens wherein there's decreased plasma glucose, and a corresponding effect would be a decrease in insulin secretion. A second type would be the postprandial or reactive type, which occurs around 2 to 5 hours after food intake. So what happens is that after you eat your food, there's a transient increase in your glucose, thereby leading to a consequent increase in your insulin, leading to hypoglycemia. So again, we work this up with screening for oral anti-hyperglycemic agents. And if it will not still be evident, then we have to seek for biochemical values or work up. For example, again, your plasma glucose, your insulin, and your other insulin precursors. If, for example, spontaneous hypoglycemia can't be possible, you have to formally recreate it by giving IV injection of 1 milligram of glucagon, or simulating it with 72 hours fast after a fixed meal. So it was outlined here by the study of Cryer that in endogenous hyperinsulinism, you have low glucose, and after administration of glucose, you have increase in level of your glucagon, and also you have increased levels of your insulin and its precursors, as well as decrease in beta-hydroxybutyrate. So from those values, we can further classify the type of hyperinsulinism our patients have. So this figure shows you the different pathophysiological classifications of tumor-related hypoglycemia, primarily hinging on tumor production of autoantibodies, which impairs insulin binding and its clearance, leading to increased plasma insulin. So non-islet cell tumors are rare and serious complications of malignancy, and this is caused by aberrant production of your pro-IGF or your big IGF-2, and this leads to persistent insulin-like activity. This can be caused by benign or malignant solid tumors of mesenchymal epithelial hematopoietic lineage. Again, 41% as seen in this study would be from mesenchymal origin, while 43% are of epithelial origin, hepatocellular carcinoma at 16% being the most common. So why is there an increase in insulin activity despite a decrease in insulin production? This can be explained by an understanding of insulin-like growth factor. So as you can see, both IGF-1 and IGF-2 are proteins that are structurally and functionally related to your insulin, but they have 10 times lower glucose lowering effect. As you can also see in the figure, IGF-2 has a higher binding capacity to your IGF receptors. And once that complex is made, then it can activate your insulin. And it has been postulated that IGF-2 is more predominant in NICTH tumors. So in non-islet cell tumors and a failing liver, there is their effect of decreased glycogenolysis and gluconeogenesis, and also decreased lipolysis in your adipose tissue. And also because it's a hypercatabolic state and increased consumption of glucose by the tumor, there's increased peripheral glucose consumption, overall leading to hypoglycemia. NICTH is thought to be secondary to an abnormality in your IGF-2. So normally, pro-IGF-2 just constitutes around 10 to 20% of your total IGF-2, but with your NICTH, it makes up tremendously to 60 to 70%. So in NICTH tumors, you have here decreased serum glucose again, and then, however, your insulin and your other insulin precursors are already decreased in contrast to your insulin secreting tumors. So this table just shows you that comparing the different etiologies of hyperinsulinism, your NICTH only has the type which has increased IGF. Hypoglycemia may be the first presenting symptom of malignancy, as seen in the study, 48% of cases prior to the manifestation of hypoglycemia. So this figure shows you the levels of IGF-2 and your IGF-1 in patients with NICTH tumors. So in the black circles, those are IGF-2 values. And in your open circles, that's IGF-1, while in the square figure, that's the normal subject. So it basically shows you that in hypoinsulinemic hypoglycemia, the combination of a tumor burden plus hypoglycemia should warrant the suspicion of IGF prevalence of hypoglycemia as a paraneoplastic manifestation was shown to be at 4.27% and shows you that's relatively rare. Again, the findings of biochemical parameters would give you a low serum glucose and low C-peptide and insulin and high AFP as well. Furthermore, if we do further test ratio, again, I'd like to highlight our case. In case 2, there's a massive tumor burden shown in the right hepatic lobe, measuring around 11.7 by 19 by 11 centimeters. So in this study by Fukuda in 2006, it was shown that 70% of most IGF-2 tumors had large size of beyond 10 centimeters as seen in this graph. And now we move on to the types of hypoglycemia in our hepatocellular carcinoma patients. In type A, these are rapidly growing tumors, and hypoglycemia manifests the late course of the disease. The mechanism of action is the tumor consumption of glucose. And again, the biochemical markers would reveal suppressed insulin, C-peptide. However, there's increased glucagon. However, in your type B, it constitutes just 5 to 13% of HCA patients, and hypoglycemia occurs early in the course of disease, and the mechanism of action being defective processing of pro-IGF-2 by the hepatocytes. So again, to diagnose that, we have to satisfy the Whipple's triad, satisfy the different biochemical parameters. There must be a rapid response to your glucocorticoid therapy. And again, IGF-2 versus IGF-1 ratio is increased. After diagnosing, however, in some patients, however, we can do further testing of IGF-2, but the measurement of total or free or big IGF-2 is not routinely available and is unnecessary for establishing the diagnosis. Hence, when you have low insulin and tumor burden, this should already raise the suspicion for NICTH. So in both our cases, they had low insulin, and in our second case, C-peptide was done, which was also low. And after that, we can now identify the presence and location of the tumor to address hypoglycemia. In the first case, imaging was done, and in CT scan, there was again a right hepatic mass, which was enhancing on the arterial phase, measuring around 17 by 11 by 13 centimeters, and AFP was again elevated. We sought the help of our pathologist, and stainings were done. Initially, it was diagnosed as HCCA, but the negative HepR1 and positive CK7, albeit the negative CK20, would not lean towards the HCCA diagnosis. Hence, further stainings were done. And we did CK19 and TTF1. TTF1 was negative and CK19 was positive. Hence, we lean towards the diagnosis of primary adenocarcinoma of the pancreatobiliary tree. However, there's a question because the CA19-9 was normal. And again, we place it side by side with the finding of an elevated AFP. Hence, we entertain the question whether this was a combined case of hepatocellular cholangiocarcinoma. And true enough, when this is just a sweeping overview of the immunohistochemical stains expected in the HCCA are shown in the left part of the column of the table, and in cholangiocarcinoma in the second part of the table. In our case, it seems to be it satisfied both. No, there's a combination of HCCA and CCA features. Hence, we diagnose this case as a possible combined hepatocellular cholangiocarcinoma. This type of malignancy is a rare and aggressive primary hepatic malignancy, which just occurs in 1 to 4.7% of all primary liver cancers. And it has limited treatment decisions and unfavorable outcomes with systemic therapy. Risk factors include HBV infection, HCV infection, in some cases, type 2 diabetes mellitus, which is an inconsistent finding. So what is the management of NICTH? First, this just shows you the basic outline or algorithm of NICTH management. So after its diagnosis, we had to do prompt resection. Full resection should cure the hypoglycemia, but in some patients, there is massive tumor burden or the structures would be intimately related to vascular structures. So this would limit our option to subtotal resection, also probably patient preference. So subtotal resection often ameliorates or postpones hypoglycemia. In our first case, it was in both cases, though both were considered non-surgical. So in the first case, the patient was referred to oncology and had undergone one cycle of chemotherapy. For a second case, it was diagnosed as multifocal hepatocellular carcinoma and was started on lenvatinib. With the administration of chemotherapy, it was found that the patient had improved CBG levels and had decreasing requirements for steroid and glucose infusions. Hence, discharge was being contemplated. In our second case, however, with a massive tumor, persistently the sugars were labile and the patient was acidotic and required sustained low efficiency dialysis. If those measures are not feasible or resection is not feasible, then we must provide our patient with alternative anti-tumor modalities. And also nutrition is a must. However, if all those won't work out, then we can give glucagon infusion, glucocorticoids, recombinant in IGH, or in other cases, octreotide. Steroids are the mainstay of treatment for NICTH. We start at the lowest possible dose because it has been found that steroids can suppress big IGF-2 in a dose-dependent manner. So in some studies, it has been found that continuous rather than intermittent infusion of dextrose is more effective because it can lead to less frequent hypoglycemic attacks. So in this figure, you see the levels of our CBG. After the administration of chemotherapy, it relatively increased. And then at the 15th hospital day, glucose infusion was decreased and the patient was shifted already to oral steroids. However, on day 20 of hospital day, the patient acquired acquired infections and also had recurrence of symptomatic hypoglycemia. The patient also had persistent diarrhea. So we needed to shift back to IV steroids by then. Other forms of medical management include octreotide, which is a somatostatin analog and inhibits the release of growth hormone and insulin. Another modality would be your recombinant growth hormone, which increases insulin growth factor binding proteins. And this can lead to an increase in your ternary complex and prevent interaction with the insulin receptor, thereby not fully activating the action of insulin. However, in other studies, these have not been proven effective. So again, in our first case, there was persistent diarrhea. In both cases, there was continuous D50-50 infusions. However, because as an effect of diarrhea, the patient had intractable hypokalemia and was already prone to infection. Also, in our second case, there was infection and intractable acidosis. Hence, both succumbed to demise. Hence, in the first case, it was signed out as septic shock secondary to infectious diarrhea and hospital-acquired pneumonia, while in the second case, it was signed out as severe metabolic acidosis secondary to acute and chronic liver failure and acute renal failure, multifocal hepatocellular carcinoma, stage 4 in progression. So overall, this case report, I would like to summarize this as a hypoglycemia manifestation should warrant workup. When it will not be evident, then we have to question whether the increase in insulin activity jives with the insulin production. If it does not jive with that, or both won't align, we have to already entertain the possibility of insulin growth factor. And when we diagnose NICTH, we have to address the underlying cause of hypoglycemia, which is the tumor itself, through resection and systemic therapy, and also provide supportive treatment of glucose infusions and steroids. These are my references. Thank you. If you have questions. I have questions.
Well, thank you for that presentation. Do we have any questions? Okay. Oh, we have one. Good.
Ma'am,
Yes.
do you want to establish any relationship between persistent hypoglycemia in a non-diabetic patient and neoplastic disease?
In non-diabetic patients, even if we address the medications or adjust the medications, if it will still be recalcitrant to your modification, you also have to entertain the possibility of a malignancy through this finding. And if by having that suspicion, we have to further work it up through the biochemical parameters that I discussed.
What about a diabetic non-diabetic? Suppose the patient diabetic is there for 20 years.
Yes.
How would you read it? How would you correlate it?
in some cases, type 2 diabetes is also an inconsistent finding. No? So even if we have addressed the hypoglycemia and diabetic individuals by modifying the medications or totally stopping the medications, if the hypoglycemia is persistent, then we have to further work up with biochemical parameters. Thank you.
Any other questions? Thank you, yes.
Thank you.
I don't know. For our next speaker, I'd like to introduce Professor Nadia.
Thank you. Good afternoon, everyone. I had to present this conference yesterday, but for medical reasons, I do it now. Thank you for following me to present it today. My presentation, I'm Professor Nadia. I'm a psychologist and communication and pathology and language pathologist at University of Breda II. I'm also director of laboratory of language cognition and interaction. My presentation is about the impact of cognitive flexibility on narrative in Algerian aphasic patients. Communication is not only to name or to exchange ideas, to express emotion, but it's also to convey, to tell, and to narrate. However, this ability can be disrupted for different reasons, such as aphasia. Aphasia is a language disorder that affects person's ability to understand and or express oral and written language following a brain damage, which is caused by a severe head injury, as a stroke, a tumor, an infection, degenerative process. In these cases, the aphasia usually occurs with other types of cognitive problems, such as memory and attentional problems or confusion. Several language disorders are described in aphasia, such as phonetic, phonologic, semantic, syntactic, pragmatic deficits, but the most deficit studied is the word missing in different tasks, especially naming tests, contrary to the narration. For that, we see that narration is also important in communication, since we tell our daily facts, hence our interest in this study. However, we underline that the Algerians are at least bilingual, that it's why we wish to study the narration in both monolingual and bilingual aphasic subjects. We know that switching to a language to another one requires the intervention of a number of cognitive processes we group under the label of cognitive flexibility. Thus, our aim consists to study the impact of the cognitive flexibility on the narration. In Algerian aphasic subjects. Methodology and procedure. In order to realize our study, we choose the clinical method, which allows us an exhaustive description of linguistic and cognitive disorders. The participants Our population is composed by 12 Algerian aphasic subjects. 6 of them are monolingual. They speak Algerian Arabic or Kabylian spoken. 6 others are bilingual. They speak Algerian Arabic or Kabylian spoken, Algerian Arabic and French, Kabylian spoken and French, Algerian Arabic, Kabylian spoken and French. The tools we use in this study, the following tools: the test of unusual stories. We elaborated this test in 1989 when I was a student, and we revised it in Arabic and Arabic spoken, also and in Kabylian and French, because the test was elaborated in 3 languages, French, Arabic, Algerian, and Kabylian. It includes the 5 unusual stories inspired by the Algerian socio-cultural reality. Initially, we proposed 11, but when we revised the test, we found that only 5 were statistically valid and reliable. Sorry, my English is not very good because we don't practice English in Algeria. Then I try to. This story, every unusual story proposed here answers to the following questions: where, when, who, what, why, how. And in the answer of the how, we can detect the unusual event, and the objective of this item is to push the patient to criticize the story. In order to study narrative, we opted for a cognition neuropsychological approach. This stage takes place in 3 stages. At first, we tell the story, then we ask the patient to narrate it without support, because the stories are told with comic pictures. After 3 minutes, we ask him to tell it in a second time according to pictures, and we ask him, sorry, but it's not the good PowerPoint I sent the last, but they put me for the draft. The two for explain and interpret the language deficits, we proposed a model because the stories, when we tell the story for the patient, he have to listen and to see the pictures for understanding. Then for that, the first modalities are visual and auditory modalities. Then perception and attentional functions are very important because they connect between the utterance and the content of every story. And there is a syntactic analysis and thematic interpretation allow the comprehension of the text. In expression, we have also attentional, auditory, and visual working memory, lexical and semantic systems, syntactic systems, sentences planification, and production of the narration of the And for testing cognitive flexibility, we choose only verbal working memory, and we use the test of Majerus. Visual working memory, ray pictures, la figure de ray, forme A, and the dam test, le test des barrages. The results showed that selective attentional, the presence of selective attentional deficits, working memory deficits, especially in auditory modality, word missing or the lack of words as paraphasias, semantic and phonemic paraphasias, syntactic and pragmatic deficits, agrammatis, simplification of the sentences, problem of tenses. The results showed that the narratives of people with aphasia, when they are monolingual, the quality of the narrative is less if we compare with bilingual aphasic. And the impact, and the linguistic deficits were more important than in monolingual patients. But according to Amstad and all, although GAS data suggests that bilingual spoken exhibit higher inhibition and monitoring abilities, the impact of the volume of the impact of selective attention is less when we compare with memory deficits. Then the impact is not very important. In conclusion, although this is a preliminary study, this research show an impact of cognitive flexibility on the narrative of aphasia subject. And thank you for your attention.
Do you have any questions? Okay. Thank you. Thank you.
Thank you.
And for our next poster presenter, we have Dr. Yu from South Korea.
Good afternoon. I'm Jae Yu, working in Department of Physical Therapy in Seoul University, South Korea. Today's lecture I'd like to talk is the use of the augmented reality based conditioning program for pain, muscle strength, and muscle endurance in young adults. It's a case study trial. First, background. Augmented reality, which is integration of digital information in real environment, has become a valuable and emerging technology used in the rehabilitation field to improve recovery following several health conditions. The present case study was carried out to assess the short-term impact of using an AR-based conditioning program on shoulder pain, muscle strength, and muscle endurance in active male students suffering from mild shoulder pain. Method: 8 young adult men with shoulder pain were recruited for this pre-test post-test case study trial. All participants were included in the AR-based conditioning program, which is consisted of warm-up and cooling down exercise, which is focused on stretching. And main exercise was strengthening that targets the following muscle group: deltoid, trapezius muscle, rhomboid muscle, thoracic muscle, and supraspinatus, infraspinatus, subscapularis, bicep brachii, and tricep brachii. Pain, muscle strength, and muscle endurance were measured before and after the intervention, followed by statistical analysis. We used the Wilcoxon sign rank test to compare the change in variables. I read quite the result. As a result, significant improvement was observed in pain, which decreased after intervention in all patients. P-value was under 0.05. Additionally, muscle strength and muscle endurance as well increased in all participants after the intervention, which was set under 0.05. Participants expressed their satisfaction with the exercise content and the feedback received through the performance of AR-based training. Moreover, participants expressed their desire to continue with program for the long term. The conclusion: as a result, we found that strength the muscle that support the shoulder movement help to keep the shoulder joint stable. While keeping these muscles strong can relieve shoulder pain and prevent further injury. More interestingly, gently stretching after strengthening exercise can help reduce muscle soreness and keep your muscle warm and flexible. Using AR-based training may provide sufficient motivation along with adequate feedback for better progression and an effective self-training program. Thanks for your attention.
Do you have any questions? Okay, I have it.
Yeah.
How would you relate with frozen shoulders in diabetic patients?
Yes, in case of our country, we always use several kinds of exercises. One of the most important exercises is pendulum. Yeah, pendulum exercise is like we shake the shake our shoulder because in case of the frozen shoulder, convex and concave between the joint is audition, mental audition. We want to make to relieve the audition. It is important. So we always use the exercise we like to pendulum, and sometimes we use the mobilization to prevent the audition. Yeah, it is our country's case.
Thank you.
Any other questions? Thank you.
I got it. I said, enough to speak with them. Yes, I after a more heat or the light. I actually, because I'm trying to play with topic something that's like
For our next presentation, we're going to have Dr. Ashmore from India.
That I sent today. No, it's not this one. I sent a presentation and PowerPoint. I think this other one. Today, email. And this one,
Hello, good evening, everyone. Yeah, first of all, I would like to appreciate and thank you for the invitation to give this small talk, or let's say it was a poster, but they requested us to do like a small presentation. I will go ahead with that. Well, I'm consultant gastroenterology and hepatology in Cleveland Clinic, Abu Dhabi. We're presenting here 6 years experience of POIM and PO for the management of gastrointestinal motility disorders, the United Arab Emirates experience. So, well, as you probably are aware, the perioral esophageotomy, POIM, or perioral pyromyotomy with PO have emerged as a new endoscopy procedures for the management of gastrointestinal motility disorders. There's existing evidence all over the world about the effectiveness of this procedure all over the world. But we wanted to evaluate what is the evidence in our region. In MENA population remains limited. So that's why our objective was to evaluate the feasibility and safety of POIM and PO for gastrointestinal motility disorders in the UAE. So what kind of methods we used? We used a retrospective study, including patients who underwent POIM and PO for gastrointestinal motility disorders from January 2016 to September 2022. Demographics and surgical outcomes were analyzed. Well, these are all results, which despite it's not a huge number, it's not a very common technique done all over the world. So that's why we could collect in all these almost 7 years, 97 patients. We underwent 71 for them were under POIM and 26 under PO. The mean overage of age of the patient was 40.7. There's a few common comorbidities, and it's very curious in some of these patients, like GERD, which is gastroesophageal reflux disease, hypertension, hyperlipidemia, diabetes, constipation. All these cases, as you can guess, we're doing endoscopically. The median operative time was very considerable lower in the PO rather than the POIM. For the POIM, we expect to spend around 74 minutes, and the PO around 30 minutes. Basically, what was the etiology for different kind of these gastrointestinal motility disorders, and which we performed the POIM was echinacea type 1, the most common one, type 2, the 72%, type 3, 7%. Type 2 was the most common one that we performed POIM. There is a certain kind of patient that we could not identify with esophageal manometry and classify them in echinacea type 1, type 2, type 3. So that way we classified them and specified. Etiology for patients who underwent both. For gastroparesis, idiopathic was the most common one, 73%. Diabetic patients, which, as you are aware, is very common in our region, combined with obesity, and 11% of post-surgical intervention. Well, these kind of patients usually have received or undergone different kind of procedures before undergoing to POIM or PO. In case of POIM, these patients, most of them, they went before under endoscopy balloon dilation was 30%, botulinum toxin injection, which is around 14%, and Heller-Dome operations, which is around 10%. Previous interventions for pyromyotomy included intrapyloric botulinum toxin, which is the same dose and same drug that we're using for echinacea in the esophagus. And all cases that we discussed before were performed endoscopically. And when we discuss again the timing about the procedures, around 74 minutes for POIM and 30 minutes for PO. What about the complications? When in general it's a very safe procedure, of course, it's an invasive procedure. There's always, some small window that we may face some complications. There are more often in POIM rather than PO. Basically in POIM, nausea and vomiting can happen to both of them, but the leaking and the pneumopathy thing is more common in the POIM rather than the PO. And well, all these patients were treated conservative for the complications, and then in case of the leak, we might use the partial cover stent just to fix the problem and be removed after 4 weeks. And again, these procedures will help us a lot to keep the patient, let's say, in a very, very, very mild admission in the hospital. Is very short, it's within 1 to 3 days. We have been following this patient up to 7 months after the procedure. 4 of them, unfortunately, they died, but nothing to be related, fortunately for us, with the procedure we performed. So as conclusion, we could say that the POIM and PO both of them are extremely safe and technically feasible, with low complication rates, as we have been discussing before. It takes, we discussed before, 74 minutes longer the point than POP. And our studies, are in the same line and the other procedure, already performed worldwide in terms of indication, feasibility, and results. Thank you so much.
Any questions?
What stage of gastric paresis you give injection?
Sorry?
At what stage of gastric paresis you inject this?
Well, we usually, first of all, realize and study, which is gastric emptying study. So initially, we try to treat this patient initially with conservative treatment with medications, prosynetic medication. If it's not working, we go ahead with toxin botulinum injections. If it's not working, refractory, which we already know is going to happen after the average is 6 months. And the patient is still complaining of symptoms despite doing conservative treatment with medication, endoscopy treatment with toxic botulinum injection, we go ahead with both. It's the last stage of treatment for patients with gastroparesis, but all of our patients has already been objectively diagnosed with gastroparesis, not only based on clinical evidence, which is very important. Sometimes expectations of the patient that they think that we're going to do something is going to help them if we're not. Well, I mean, we're working very closely with all the endocrinology because, as you know, the long-term surveillance of this procedure might be, again, not happening despite that we have been doing this procedure if the diabetes is not very well controlled. So actually have been developed very close loop working with endocrinology, and we have created already a fatty liver unit and obesity just to close all this multidisciplinary approach of this patient. We are aware that it's not only based on doing endoscopy procedure, is multifactorial factors which are involved in this problem. So we are always be sure that it's going to be followed by endocrinology pre and post procedure. Do this on the HbA1c always is around 5.4, maximum 6. Thank you so much for attention. Thank you. Thank you so much, Mr. Thank you.
Second is activity or troubles to be done by the market. Yeah, then there was not my I'm running the online. We get the online examination.
Okay. Our next presenter will be here in about 10 minutes, but we wanted to handle the certificate of participations for the event. I'm not sure who everybody is. Okay, start with it's improv to the event, so yeah, get a certain dance. Yeah, yes. Oh, no, I just gotta get me to
Taking if everyone is here, and give the certificate who all are present.
Oh, okay, yes.
That will be.
Gotcha.
You know the names, so you don't.
And I'm gonna say the name first. Okay.
Then I take picture.
Mr. Schubert?
This is excuse me, sir. Sorry about this. Yeah.
You will call me.
I just did, Mr. Schubert.
Yeah.
Dr. Nanavati. Yeah. I'm not big at the microphone. Right, I'm in the wrong place again. Oh, I have. Yeah. Turn it on then. Yes.
Thank you.
Thank you. Dr. Mansour. Going once. Mail that. Dr. Yasmin. Come on. She came to me. Gotta get the sign. Yes. We need tape on the floor. Yes, ready. Dr. Raymond. Thank you, Chairman. No, but that's very confident. Would like to appreciate that. So immense maybe. You, okay, who is it? What? Oh, okay. Chairperson. Oh, I got you. We're supposed to stand behind us. In front of this. Okay.
Congratulations, Steve.
Thank you.
For your nice presentation.
Certificate.
Hope to see you again.
Thank you. Thank you for the privilege.
Thank you.
Should we? Coffee break right now.
Okay.
Because I need some coffee. I'm kind of got jet lag.
Could there any presentation after this?
One more.
One more.
Okay.
Yes.
Are we having fun? That's the key, have fun. Life is too serious.
Yeah. Yeah, participation, that itself, great. अरे आगे नहीं दोनों में आगे Dr. Rizmi Mansoor. Dr. Rizmi Mansoor.
Yes. Thank you.
Yeah. Yeah. One participant is given to another participant.
Yeah, just having it coffee.
Thank you. Thank you. Thank you. One more. Yes.
I'm listening to
You stand on my right. So they should see the board.
Yeah, that's it.
Hold your right hand. This.
Yeah, very good. There we go. Yes.
We're going to be able to be mutual communication. That's we need. Okay. Over to you. Okay, where's Chairman? Yeah. Thank you. It was really nice. Thank you.
Professor who is currently working at the Institute of the Department of Gastroenterology and Hepatobiliary Sciences, Emirates Hospital, Bhubaneswar. So, first of all, I'm paying my happy gratitude to the organizing committee of the Utilitarian conference for giving the opportunity to present before all. Because of some important issues, I could not present it physically. So, I'm presenting my poster in platform and online. So, I am sorry for that, but kindly accept, kindly go to my posters. That is presented in the form of PPT. So, kindly go to it. If you have any queries, I'm happy to answer. Professor, platelet count are better, they don't operate. That's why there is an urgent need that we should how much modalities, which is actually efficient. Platelets with ratio of Mr. Presentation is platelets plane ratio of non-invasive predictor of tissue varices in cirrhosis. So coming to background, the background is it is the prevalence of cirrhosis, which is related to morbidities and mortalities, which is gradually increasing day by day. So that's why we should be very much worried about that. Almost around 1/3 of the patient usually expand because of the bleeding from gastroesophageal varices, which is usually related to the cirrhosis related to portal hypertension. Currently, what the diagnosis and consensus states all COPD patients should be screened for variants of gastroesophageal varices. Although imaging endoscopy is the most standard to diagnose the varices, but it is invasive and costly. And sometimes few patients usually, because of fear factor, they don't opt for it. That's why there is an urgent need that we should find out some non-invasive marker which can accurately diagnose the gastroesophageal varices. So what the GLD study says, this is that platelet count versus bleeding diameter ratio measurement can prevent unnecessary endoscopies in all the patients with a cutoff 909. That is the study conducted in the gut and published in gut 2003. So three of the non-invasive techniques, the PS square line, three of the non-invasive techniques, that is platelet count versus splenic diameter ratio, fibro test and fibroscan described in the literature. How to be the former, that is platelet count versus splenic diameter. And all super and says or teeth for surgery. A hypertension patient having fever associated with cytopenia, patient warrant which can cause pharmaceutical. So all patients underwent biochemical tests like LFT, complete blood count, renal function test, and USC approach is to control the patient by time. endoscopy was carried out in all patients to find out presence of varices and also to study for grading. The platelet count, splenic diameter, and platelet count, splenic diameter ratio were compared between the two groups of patients who are to be esophageal varices coming into the varices. So the statistical method we use, the results on the continuous measurement is presented a mean plus minus standard deviation and medians, interquartile range, wherever the non-parametric data is there. And also a categorical measurement expressed in number and percent. Independent student T test was used for non-skewed data or parametric data. Net quality used for the skewed or parametric. So the receiver of generated. Find out platelet count, splenic diameter ratio, split diameter to know the cutoff value and respective sensitivity and specificity. So what are the regions? This is the baseline demographic data. We found that the mean is mean platelet count is 1 by 3. The mean splenic diameter is 16.83 millimeter, and the platelet count versus splenic diameter is 1195 plus minus 86.10. So, the show if male number of females usually 4:1. 99 cases of esophageal varices, whereas 26 cases did not have any varices. Distribution of patients basing on the basis of grading of varices, we found 35% cases having grade 1 varices, whereas 39% having grade 2 and 26% having grade 3 varices. So when we compare the various parameters between the two groups, we found that there is significant difference in the hemoglobin values between the two groups. And there is lower hemoglobin values in patients with varices. And it is significant. Of esophageal varices of swelling, 26 patient of esophageal varices of and good evening, everyone. This is Dr. Sambit Kumar Behra. I am currently working as assistant professor in the Department of Gastroenterology, Medical College Hospital, Cuttack, Odisha, in India. This was a study which we did 4 years back. There's a one study, and it was regarding the assessment of awareness of hepatitis B and C among community barbers in coastal Odisha. As we know, 240 million people are HBsAg, that is hepatitis surface antigen carriers. A significant proportion of these individuals are at considerable risk of chronic active hepatitis or cirrhosis or hepatocellular carcinoma. And despite the immense progress that has been made with the availability of vaccines of hepatitis B and the development of antiviral agents, the global burden of hepatitis B related disease is still substantial. Even the universal vaccination programs, they have not been able to completely prevent this acute case of hepatitis B virus, especially the high-risk populations. There is also a significant gap in the knowledge, the attitudes, practice of the patients and the general public also regarding the various aspects of hepatitis infections. In India, the widespread traditional practice of saving at barber shops or from the roadside barbers might be a root of this blood-borne viral disease transmission. In a research study that is estimating the prevalence of occurred hepatitis B viral infections, knowledge and preventive practice against hepatitis B virus in barbers serving in armed forces in North India, the prevalence of hepatitis B was found to be 3.79%. And all the barbers were total anti-HBC positive had a replicative hepatitis B viral status. And not a single barber was aware of the existence of modes of hepatitis B viral transmission and had never been screened for hepatitis virus. Why? An overwhelming majority of barbers didn't follow the proper disinfection cross practices. Among barbers in coastal Odisha, that is our state, also has not been evaluated. As barbers are potential reservoirs infection, it is important that the knowledge and the practices of barbers regarding the risk factors and the modes of transmission of hepatitis B are evaluated in order to intervene and to break the transmission chain. With this background knowledge, we evaluated the awareness of barbers regarding the transmission of hepatitis B and hepatitis C, and we assessed the risk factors which were associated with this transmission among the barbers. It was conducted in Cuttack district of coastal Odisha, India, which has a population of over 2 million people. The sample size of 730. And the informed consent was obtained from each participant in the study and were assumed that the information will be kept confidential. A simple questionnaire was designed, which was structured into 3 parts. The first part of this questionnaire included personal information like the name, the educational status, age, experience in hair cutting and shaving, and the marital status. Second part included questions regarding the practices, blade changing, washing the razor with water or antiseptic solutions, disposing of blades, use of as alarm on facial shaving cuts, and changing of approach for every customer. The third part included questions which evaluated their awareness regarding the hepatitis B virus and hepatitis C virus. And these results were statistically analyzed. You can see that the majority of barbers were in between 26 years to 35 years of age, that is around the age groups. 21% of these barbers are between 18 to 25%, while 19% of barbers were between 36 to 45 years. So, majority of barbers were young Regarding the qualification of these barbers, 64% of barbers they studied up to higher secondary, while 16% had studied up to secondary education and 14% had only primary study. Regarding the experience of barbers, 34% of barbers had experience of 6 to 10 years, while 41% of that is majority had more than 10 years of experience. Regarding training of barbers, almost 74% of barbers were trained. Regarding hepatitis B vaccination, among those barbers, you can see that 97% of barbers were not vaccinated at all. And regarding the common practice, almost 90% they use blade and hair disposal in they throw the blade and hair in municipality dustbin, while new blades were used in almost 100% of individuals. Regarding the using the hand for disposing foam during shaving, almost 90% of barbers they use hand for disposing foam. Regarding the use of alum for facial cuts, more than 95% of barbers they use alums. And while the washing the razor with water, only almost 90% of barbers they use and they wash razor. Regarding the awareness of hepatitis B and C, whether it is a viral disease, 2 points, only 2.7% of barbers they know that it is a viral disease. Whether it can be transmitted, only 7.6%. So much of ignorance regarding this hepatitis B and C among these barbers. Whether it is a fatal, almost 14% do not know that is a fatal. And whether they themselves they might play a role in the transmission of the disease, almost 90% do not know. Regarding sharing of blades, whether hepatitis B and C can be transmitted, 85% their barbers do not know about anything. Regarding blood transfusion, whether it can be transmitted, 87% do not know. Regarding sexual contact, whether B and C can be transmitted, 82% do not. But when we compare the barbers, different type of barbers who are working in salons or who are working in open air shops, we can see a significant difference in between the earnings among those barbers also. So, the majority of barbers are poor regarding this hepatitis B and hepatitis C. And most barbers do not know about the vaccination of hepatitis B, and none of them have any knowledge about hepatitis C. And most of the barbers do not adopt appropriate infection control measures. So there is a need for regulations to be put in place regarding the adoption of safe practice in our country. Use of alums at salons should be strictly discouraged. And despite being an exhaustive guideline, the unknown statements of INASL, that is the Indian National for Study of Liver Diseases, do not have any recommendations concerning the barbers and the transmission of hepatitis B and C. So to conclude, this must be educated regarding the safe hair cutting and shaving, and must be aware of the seriousness of the problem of hepatitis, viral hepatitis, and the various modes of transmission. And we strongly feel that INASL needs to take cognizance of these hard facts and incorporate recommendations in its guidelines on the prevention of hepatitis B Thank you.
Hi, we are done with the meeting. So tomorrow we'll be joining via Zoom only. So please be there on time. Thank you. Bye-bye.